Paul, thank you very much for the very kind introduction. In the United States, if you go to a comedy club for stand-up comedy, you never sit in the front row. It's a thing. People will call on you and ask you to say things, but I won't this time. Okay, so why care about psychiatric genetics? And the stuff that Paul mentioned is exactly the case. I think the one thing that'll be a theme in this talk is just how much cooperation internationally has actually made a difference. And certainly that sort of ethos is everywhere now. So a couple of years ago, we needed some replication data. I sent 5 emails. 2 days later, I had replication data from many thousands of subjects. That's how easy it is right now. There's a culture of cooperation. People work together nearly universally. And that's the thing that's made progress really, really start to take off. So what I hope to tell you about today is really why you should care about psychiatric genomics. It's been a bit of a sinking ship, as Paul had on his slide. There's been some real problems. There's been a lot of high-profile difficulties. But the too long didn't read aspect of this is that, first, there are several things that are ready for the clinical utility right now, first. And second, I think a bunch of other things are coming fairly soon. Now, in saying this, I'm effectively predicting the future, or trying to. And if there's anything that's very difficult to do if you're a human being, the temptation, of course, is unavoidable. We all want to predict the future in one way, shape, or form. And of course, when it comes to technology, people have done this a lot. And clearly, this is from 1951. I've yet to see my personal helicopter or my personal jetpack. And I don't know if you've been to Los Angeles lately, but that definitely is not what it looks like. So some of these things actually have come through. This was somebody back in the 40s talking about a TV set you could hang on your wall. Okay, that happened. Star Trek communicator. Most of us have cell phones. Not the same thing, but it's pretty close. Still waiting for the jetpack, and of course, we're a little worried about what ultimate intelligences are going to do to us at some point. So the U.S. baseball scribe and Zen master, Yogi Berra, has this famous quote, "It's tough to make predictions, especially about the future." And if you think about that really hard, your brain might break, but it's probably true. And medicine, of course, is full of similar things, where here we have, I think this person was the personal physician to Queen Victoria. The abdomen, the chest, and the brain will be forever shut from the intrusion of the wise and humane surgeon. Not so much. This quote I really like by the president of IBM. I think there's a world market for maybe 5 computers. I'm going to bet there are people in this room who have 5 computers on their person right now. And finally, of course, there's no reason why anyone would want a computer in their home. And this was a guy who was head of a computer company, which of course doesn't exist anymore, because I think they kind of missed this particular boat. But I think computers are actually a sensible sort of analogy for what we're talking about here. Because if we think about how computers have changed in the course of the lives of many of us, from 1980 through to 2015, Cindy and I got one of these in 1983, in fact. This was one of the first Macs that came out, the 125K, because who needs more than 125K of RAM? It's a reasonable analogy in terms of how things have progressed. Some things that people have talked about with computers have actually gone completely wrong. Some things, in fact, have become completely right. And a lot of things that were not anticipated at any point are, in fact, part of people's daily lives right now. And so this comes down to technology development and knowledge and experience with the tech. And let's not forget Amara. Amara's got something called the first law of technology, which is you have a level of excitement and time, you have an idea, enthusiasm, it becomes overhyped, it crashes to this disillusionment pit, and then eventually we start to move toward true progress. I would argue we are there, that whatever disillusionment and hype we've had in the past, I think people are really starting to really think hard about what this might do and how it might be important clinically. So let's go over the technology relatively briefly. The first thing up here is the sort of the latest, greatest from Illumina, one of the big sequencing companies. This is called the HiSeq X10. To my knowledge, there are 10 machines, and this is the thing you buy if you want to sequence whole genome sequence populations. It can do something like 18,000 genomes per year. SciLife Lab, split between here and Uppsala, has one of these. I think it's one of 4 in Europe that does. I don't know how many are in China, but it's way more than that. This is another technology that's kind of popular. It's sort of a benchtop sequencer from Ion Torrent. And the one, of course, which is just spectacular, it doesn't work yet, but it's kind of spectacular to think about, is this USB one from Oxford Nanopore. And so eventually what people are, and people are actually trying to develop handheld sequencers, where in theory you could actually take a blood sample from somebody in an ICU with sepsis, just put a drop in and figure out, it's sort of more or less real time what the offending organism might be. So again, this is just like computers, isn't it? Where we heard such great things are just around the corner. And sometimes some version of that comes, but often it doesn't. This technology is established, ready to go. This is actually in labs right now. This is still on the horizon. Now, if we look at I think this graph is really kind of cool because the y-axis is a log scale. And so this is the cost to sequence one genome. And when the Human Genome Project was done, completed around the year 2000, the cost to do one genome took years, and it was like $100 million. And as you can see, there was a slow development with the same technology, a sharp drop, and again, this is on a log scale, so this is a huge decrease. New technology, plateaued a bit, but there's been a recent drop as well. So if you wanted to sequence one genome right now, the cost is a bit over $1,000 US, takes a couple of weeks. It's feasible to do at very large scale. And so this is with sequencing, which is rereading the genome. And then, of course, the other technology we have is genotyping, which typically looks at a fixed set of genetic markers. Sequencing means you actually go back, and for a person, you redetermine every single base, or as many of them as you can, in that person from scratch. This is one where you have basically a fixed set of markers, and you genotype them. Like McDonald's, if you get the same thing over and over again, you can do it cheaply and quickly. If you actually do it at scale, when you're trying to recreate everything, it's more expensive. And so the costs here have fallen as well. So about 15 years ago, to do 500 odd markers would be about $2 each. It would take months, literally. The price when the first big chip came out was about this. And then currently, they're very inexpensive, and the cost per genotype is very, very small. In fact, we do these for quality control and cell line experiments. That's how relatively straightforward it is. To do 5,000 people, many labs can do 5,000 of these per week, and large labs can do even more than that. Now, this was not supposed to pop up quite yet. Oh well, so much for that. If the genomic results that we use in schizophrenia were scaled to be 1 pixel, the raw data would be on the order of 1,200 kilometers, or approximately Stockholm to Vienna. So just to give you an idea, there's this enormous big data funnel, right, where you have to handle every single step with impeccable care in order to have anything valid at the end. But the scale is actually really quite huge. So that's so the whole thing about technology could in fact be an entire 1-hour thing in terms of what's happened, what's developing. But let's just leave it like there and come back and answer questions on this if people would like in the Q&A. Now, genetic studies for humans are typically, not always, but typically are variants on case control studies or are directly case control studies. And a lot of it is very straightforward. So the typical thing that we do revolves around logistic regression, which of course is the workhorse of epidemiology and clinical research. You're trying to predict case versus control status. So the dependent variable is schizophrenia, yes or no, autism, yes or no, cardiovascular disease, yes or no. You have many issues, as always, in terms of how you define a case, whether your controls are appropriate, whether there's some bias in your ascertainment or selection. And those are always as important for a genetic study as they are for anything else. The key predictor, the independent variable, is one genetic marker. So you've measured something, and you're trying to see whether the frequency of that marker is similar or different in cases and controls. And the cool thing about genetic markers is that almost by definition, exposure precedes the development of the disease. We're exposed to our genomes from the moment of conception onward. I mean, yes, there are some models in which that assumption doesn't hold, but generally what I just said is true. Exposure precedes disease onset, and so we don't play guesses about reverse causation. Next, we typically include covariates, particularly ancestry, to avoid a common bias. We have to take into account multiple comparisons. A typical study might cast 1 million different tests, and you can't use 0.05 as a level of significance. You can't use 10 to the minus 4 as your level of significance. You have to adjust for the fact you've done 1 million comparisons. Power, of course, is quite important as well, especially since you have to have a very small type 1 error level. We typically need very large sample sizes unless the effect that we eventually find is massive. We've got a lot of experience in this right now. There are a couple of instances where the effects are kind of massive, but generally the effects are really, really subtle. But the whole idea of a genetic study is you basically take this model, you run it once, you run it a second time, you run it 1,000,000 times. And again, this is why we got computers. And since we're doing this over the whole genome, this is a genomic study. Now, going a level deeper on this, this graph, which I stole from a review from Steve McCarroll and Steve Hyman, shows this is the current time going back in time to 100,000 years, and this shows you the population of humans on the planet in billions. And as you can see, we were really, really rare for a long time, and then around 3,000 years ago with agriculture, the population started to climb, and in very, very recent time, there's been this huge increase in human population. Now, so as it turns out, if there's a genetic variant which is common, it tends to be old. So in other words, if there's a particular genetic variant that half the people in this room have, by and large, that variant will actually have its origin way, way back in time, which means it tends to be found pretty much everywhere. Maybe a little bit more, maybe a little bit less, sometimes not at all, but usually if you see it in one human being and it's common, you're going to find it in many other human beings as well. Now, on the other hand, with this new expansion, if something happened very recently, it's going to be pretty rare. So typically, rare mutations are new, and older mutations or common mutations are rare. And the technology you use to go after these two different types of events depends. So if you're looking for old variation, genotyping with a fixed set of markers is the way to go. That's a relatively effective thing to do. It's cost efficient. If you're looking for something which is really quite rare, maybe present only in one person or in that person and their immediate relatives, they may literally be the only people in human history who have ever had that mutation. And if you want to find that, you simply can't use this method. You have to use a sequencing method. So again, this is sort of the thing that goes behind what we call common variant association studies, looking for things that are old but common, and things that are rare variant associations, where we actually start to look at things that on the frequency spectrum become low frequency, things you might find once in a large sample, things that are private just to like a person and their immediate relatives, and even things that are found literally in one person ever. A couple of examples of this. This was a paper we did in schizophrenia, a common variant study, where we essentially looked at something like 8 million genetic markers in a large sample of 5,000 cases from Sweden and 6,243 controls. And again, when I mentioned coming up with all these replication samples, that was the 5 emails in a couple of days in order to get that data. So that's how that happened. This is an example of looking for rare variation, where we redetermined the bases in the protein coding portions of the genome, the exome, in 2,500 cases from Sweden and 2,500 controls. And again, this is an example of a common variant and a rare variant study. Okay, so why should we care? And my little computer here is to remind me to say that there actually are a couple of clinical uses right now. In effect, this is kind of like where computers were back in 1980 or something, where it was very clear that there were a bunch of uses which were immediately ready for prime time. So for instance, the use of computers for managing payrolls, for running telecommunications systems, for working for air traffic control, that was pretty straightforward, pretty obvious, running payrolls, whatever the case might be. So again, there are some uses which are actually extremely important and fairly straight up. And a lot of this comes down to what is a clinically relevant or actionable sort of variation. And typically, the ones that we're talking about are almost by definition really quite rare and typically have strong effects. And this will be something that'll come up over and over again. That in addition to rare things being new, rare things tend to have a stronger effect. Common things tend to have a more subtle effect. If you have something which has a very strong effect on a phenotype, there's a strong effect on a human trait, it really makes a big difference in terms of the development of the body, the development of the brain, the liability to cancer. Typically, these things don't last that long. They're selected against. And so things that are common tend not to have, at best, a subtle effect. And so it's much more difficult for selection to operate on them. Conversely, things that are quite rare can have extremely strong effects. And the things that may actually work in clinical scenarios right now are a subset of things that typically give you that they're involved in conditions that have relatively early onset, that are relatively severe, and or they have clear inheritance patterns in families. So I'm going to give 3 examples. The first psychiatric disorder that would fit this bill is intellectual disability. There was a moderately sized study that looked at this recently. We really need a much larger, more definitive study that may well be ongoing. But if you actually take 50 children with intellectual disability and throw a lot of genomic technologies at them, one study found that 39% of the kids had a de novo single base pair change. De novo in the sense that the child with intellectual disability had it, but neither parent did. So that de novo, the new variant, happened in the making of that child. And so in terms of a 1 base pair change, typically this is a base pair that happened in a critical exon of a critical protein involved in synaptic function or the development of the brain, and basically that buggered things up and led to intellectual disability. Another sort of variant is a copy number variant. And again, de novo copy number variants. The affected child has it, but neither parent does. And in that instance, this is not just a single base pair change. This is actually the loss or the duplication of a region of the genome. And so occasionally these things happen, not infrequently. We all have many of them. Mostly they don't matter. Occasionally they happen and they're quite large. They can be as much as 500,000 bases, 1 million bases or more, and they can influence many, many genes. There's many examples of these that are out there that are important clinically. And if you add up these 2 portions of the pie, you come up with something in the order of 60% being explained by one or the other. A very small piece of the pie are inherited variants, and in this case, recessives, where the child, by definition, got it from a very unlucky event where 2 people, each of whom had one of the variants, married, and the child had both and had a disorder as part of that. And so, again, this is one example where I think in many places in the world, this technology is often used, one of these technologies is often used to screen children with intellectual disability. Autism, again, is the second example, and again, we're looking at an early onset condition. But the numbers here are a little bit different. So the de novo piece is much smaller, 7%. This is after you remove kids with mental retardation, by the way. So this is just kids with autism that do not have mental retardation. And again, a small fraction of kids have a de novo event. In other words, neither parent had it, but they do. A few of them 3% have an inherited recessive, Fragile X syndrome, Rett syndrome. There's a handful of disorders that have a pretty strong chance of being something like autism, autistic-like, if you actually have a classic sort of protein-changing variation. But again, the vast majority of the pie is unknown. You can change these by selecting people in certain ways. In other words, if you look at children with autism that have sort of a syndromic picture, that they've got sort of signs and symptoms that are reminiscent of a classic Mendelian disorder, that of course will increase your selection as well. But again, in a lot of places in the world, children with autism or developmental delay actually do get these technologies applied to see whether or not they might fit into one of these categories. And the final one we'll talk about is Alzheimer's. And with Alzheimer's, there's 3 genes in particular: amyloid precursor protein, presenilin 1, presenilin 2. There are many examples of families on the planet where they've got a protein-changing mutation in one of these genes, and that leads to typically a dominant early onset form of Alzheimer's. There's a rare protective Nordic variant that Decode discovered, and of course there's the classic and relatively famous the Swedish mutation in APP as well. And again, typically sequencing technology is what used to get this. But here, obviously this is not an early onset disorder, but it is in a relative sense. Individuals with this tend to have early onset of Alzheimer's disease, early onset dementia, and again, it tends to run in a dominant way. People have looked at this in a lot of dominantly acting families, but typically you have to have a relatively large family even to know and to have a really good family history before you can get to this point. Okay, back to our little 1984 Mac again. What's the point of this? I mean, you can argue that for each of the 3 disorders I've mentioned, we can't do anything. right? There's no way right now that we can fix intellectual disability, Alzheimer's. There are some treatments for Alzheimer's, sorry. So intellectual disability and autism, there are some treatments for Alzheimer's, but again, there's nothing that works super well. The first thing, I think, is sort of a medicine 101 thing. From the very first day of medical school, they teach us that you need to call things by the right name. So I think there's certainly something important about having a correct diagnosis. In addition, especially for the childhood onset conditions, the answer kind of matters for a parent if they're thinking of having another child. So in other words, if two parents have inherited a recessive, and that's why their child has intellectual disability, that gives you a chance to actually screen the next child to see whether or not that child has gotten unlucky again and inherited both copies. That would be information that a lot of parents would want to have. I should mention that there's actually a company called Counsel in the United States, where for about $300 US, they will screen both people who are thinking of having a child for something like 80 different Mendelian disorders, ones that have strong effects, just to make sure that there isn't like two people with recessives coming together. Apparently, the yield is very low, but apparently something like 3% of all couples actually in the San Francisco Bay Area actually get this service done. So they actually have a fantastic business model just to sort of cater to people who are actually relatively well-educated and somewhat nervous about having kids. The other reason why I think this is important is, especially, a lot of parents wonder, is this my fault? And information about what exactly is happening and why can be something that can help the person sort of understand, often information helps, is the point I'm trying to make. But there's something actually I think more practical here, which is increasingly, it's important that we know why people have certain things. First, because typically that can get them connected with support and services. So for example, there's a couple of relatively rare conditions. 16p11 is a common copy number variant associated with autism and schizophrenia. 22q11 is another one associated with autism and schizophrenia and other things. And Fragile X, of course, is a relatively rare disorder. And typically, these sort of things, with the internet, with social media, with Facebook, et cetera, there's a lot of places where people can get connected. And a lot of parents have told me they find that profoundly helpful in actually sort of knowing what to do. And occasionally they can actually find the right service, the right person to go to, the right doctor who's expert in a condition, or sort of knowledge about which medicines to try for certain things can actually be quite helpful. But I think even more to the point, though, is there's the emerging therapeutics. So for instance, this paper was in Nature, where they actually came up with a really interesting, at least in mouse, potential treatment for Angelman syndrome. Angelman syndrome is really quite rare. It has sort of mental retardation, maybe even an autistic-like presentation associated with it. If a child with ID has Angelman and you don't know, and if a treatment becomes available, you won't know what to target that person for it. I've been talking with some people from Roche. One of their big models right now is trying to figure out specific treatments for these rare disorders. Again, if we know the correct diagnosis, perhaps in 5 years from now, there will be a therapy that might become available. This is one of my favorite stories right now. This is Deborah Levy from Harvard. And there's a pair of relatives, second-degree relatives, one of whom has schizophrenia, the other of whom has bipolar disorder. They both have a duplication of this gene, glycine decarboxylase. This is a gene that gets rid of the amino acid glycine. Under double-blind conditions, the administration of oral glycine actually makes their symptoms better. So the other way to get this, if we can actually come up with a molecular diagnostic, it might in fact be possible to get very targeted and directed therapeutics for individuals. So that's why I think we should care. Now, so the first part of my talk was talking about some of the clinical uses currently. Now the next part is more the potential. And so again, we're getting off in Yogi Berra's space here, right? Where I'm talking about things that might happen. And there's lots of different ways this might play out. And we don't know, but I think it's important that we work on it. And that's going to be a big role of the group that I'm trying to establish here. Let's talk about schizophrenia. So firstly, schizophrenia, as I think many people here know, has massive morbidity, mortality, personal, familial, societal costs. So just to sort of nail that, the life expectancy of people determined in Sweden, I think is like 10 or 12 years less than everyone else. There was a very good paper on this recently. In the US, the cost per lifetime is something in the order of $1.4 million all told. So again, this is a hugely, and that's the one part of this we can actually put a number on. The cost to an individual and to their family, of course, is incalculable. We've done a lot of different things over the last 100 years to try to understand schizophrenia. And largely, there's been a few insights, but largely we haven't really had things which have tended to replicate or to be clear and consistent over time. And I think for a number of reasons, we have to keep in mind that we're probably dealing with a subtle process. So this is not something which typically hits a child at birth or early on. Typically, people develop more or less normally for a couple of decades of life, and then they develop a relapsing, remitting condition. Even when people are really severely ill, typically they still have preserved sensorium, preserved consciousness, mostly preserved cognition, and certainly preserved basic neurological function. And so I think by definition, we're not talking about it like as an atypical Mendelian disorder that affects the CNS. Once you get onset, typically you get a very quick, relatively rapid, relatively direct progression to seizures, coma, and death. So we have something which almost by definition has to be subtle. Now, in terms of why genetics, this has been a relatively strong etiological clue. A lot of the basic work that is established, in fact, has come from Scandinavia, and a lot of it's come from Sweden. And again, I think we're looking at the data are consistent with genetics having a probabilistic rather than a deterministic role. In other words, it may increase the probability you become ill rather than being a genetic variation which determines that you get ill. I think that's a critical thing. Again, we're looking at a subtle process. The reason I say that is the risk of schizophrenia, if a person has, if you have a first-degree relative with schizophrenia, we determine in Sweden, the risk to the people in your immediate family is like tenfold increased. But since schizophrenia has a prevalence around 1%, a tenfold increase means that only about 10% of the relatives might have it. Again, it's a significant difference, it's an important difference, but by no means is it deterministic. And then the other fact I like too is the risk to co-twins. So if one monozygotic twin has schizophrenia, on average, the risk to the co-twin is only 50-50. So again, on average, what's going on there, of course, is there's more to this than genes are involved. We've done a lot of looking over the years, and I've yet to hear of a convincing single gene report for schizophrenia. And again, all this is consistent with a subtle process. As Paul indicated, we did a lot of genetics, but we were hampered. There's sort of this catch-22 you're in. If you know what the true genetic architecture is, you can design the correct study. But since we don't know the architecture, we can't design the study. And so that's a very difficult situation to be in, not knowing the genetic architecture, not knowing the impact of diagnostic heterogeneity. We don't have any good tests for schizophrenia. There's no pathology which will tell us, yes, this person has schizophrenia, or how much of it they have. We deal with sort of clinical interview, which is reliable certainly, but the validity is not known, and we can't unknowable in fact until we actually have a better idea as to what the etiological processes are. In addition, in genetics, we had incomplete genotyping. A lot of the samples that are out there were actually really, really small, a couple hundred people at most, when in fact that's off by a couple of zeros. And there certainly were indications that some of these studies had multiple different types of bias. So as Paul mentioned, in 2007, we started something called the Psychiatric Genomics Consortium. You can go there to find out more about the PGC. We're open source, and so you can actually download all the results of all the studies we've done, all the public ones. And as soon as things get published, they're up there within a day or so. One of the things we were thinking about when we started this was that we'd all read the history of this field, and we had known that every single time a new technology came along, we had gotten the sample size requirements wrong. And we assumed right from the start that the sample sizes we were going to need were going to be far larger than any one group could get. Hence, cooperation was essential, so we made something that was devoted to being democratic, participatory, open, and rapid, just to really try to maximize our chance of progress. And again, I point this out as sort of an interesting sort of thing like computer. We actually tried to rethink how we did science effectively. How can we do this in a way that's different from what we've done in the past? Competing labs, people at war. How can we actually come together and perhaps achieve more as a group than we could individually? So, cooperative, collaborative, comprehensive, blah, blah, blah. But I think that the cool thing about this is that a lot of the people in this are clinicians. All of us get it. We know why this is so important. And a lot of us have sort of agreed explicitly to put the good of the many ahead of the good of an individual group. The PGC structure is this. There's a coordinating committee which has about 15 people on it. There are 9 different disease working groups, ADHD, anorexia, et cetera, including schizophrenia. We also have some cross-cutting groups that look at sort of connections across these disorders, a copy number variant group, a stat analysis group, and I'll talk about CyChip in a second. In terms of things we've accomplished, we are, as Paul mentioned, the largest consortium in the history of psychiatry. It's a complete pain in the butt to do the author lists for our papers, by the way. One of my colleagues spent, 12 hours typing names into a website, and then he found out he didn't have to afterward. It was horrible. We laughed. I think we're clearly I think we're the largest consortium in the history of psychiatry, largest biological experiment. We are a mega-analysis consortium. We actually, instead of sharing results, we actually share the raw data, and that gives us a lot of flexibility and a lot of speed that other consortia don't have, but it was a decision we made from the very start. We've been fortunate to make some good contributions. Currently have genomic data from 172,000 people actually in analysis right now. So these have been uploaded, run through the same quality control, imputation, and analysis pipeline, and are being analyzed. We're currently adding another 100,000 subjects. And the mad goal that we have is to see if we can get 100,000 cases for each of the 9 disorders. So 1 million people. This is not as bad as it sounds. So major depression is actually one of these disorders, and we're going to be there later this year. Mainly because studies like UK Biobank, we've convinced to ask sensible questions about depression, which means we'll be able to use the 500,000 people that they have, and they'll probably have 60,000 cases along those lines. And to get more cases for other disorders is complicated. Cindy Bulick, who's my partner in crime, genetics, and family life, as well as intellectual stuff, is live tweeting this right now, and I hope she notes that I just embarrassed her. She leads the anorexia group for the PGC, and she's done a great job with using social media and large national register studies, like here in Sweden, for instance, to actually come up with relatively large sample sizes. So they're going to go from having something on the order of a couple of studies of 1,000 people, 1,000 cases, to one monster study, which we hope will have on the order of 20,000 cases before too long, and maybe even more if things work out well. And again, that's by sort of creatively rethinking the process and trying to find ways to do it. A lot of the cases she gets in the States, by the way, are from social media. Curiously, the cases from social media often are more severe than the cases that she gets from clinical sources. So it's not like doing this actually hurts the study. In fact, it probably helps. It taps into a pool of people that you might not get otherwise. Okay, so let's talk about how genetic data can be used. And again, the little Mac guy is up there just to sort of remind us that I guess I could have used the little stupid Windows paperclip man, but that would have been a worse one, wouldn't it? Anyways, the little Mac is there to remind us that this is really kind of off the page. So this is not necessarily established, but I think this is something which is a major idea or major intellectual thrust that people are working on. So there are 3 ways you can do this. The first, let's talk about etiology. And the logic here is that psychiatric drug discovery has almost always been through serendipity. Somebody develops a pre-med for surgery and notice that it calms patients. They try it in people with schizophrenia, and lo and behold, it's an antipsychotic. One of the first antidepressants was developed as a tuberculosis medicine. And they noticed that people in sanitaria who were getting this, they started becoming less depressed. The first antidepressant, okay? This is not a rational process that's driven by biology, that's driven rationally. And so the whole idea first is that we wanted to see if we can use the genetic findings to illuminate biology where we really do not understand much for sure. If we get this, if this gets us into the pathways, into the readouts, then there's a way to do high throughput drug discovery and high throughput evaluation of whether combinations of medicines might actually be better than single drugs. My pharmacology colleagues tell me that one of the main concepts they have right now is the notion of a magic shotgun rather than a magic bullet. So in terms of where we are right now, in terms of the genomic findings, 3 slides real fast. So in terms of the exome sequencing stuff, There's been a couple of studies published, 3 small, 2 large. The 2 large ones are here. This one was from Sweden with Christina Hultman and others. And basically, together as well as separately, no single genes pop up. There certainly are indications of pathways. The pathways which appear to occur more likely than chance have to do with calcium signaling, the post-synaptic density activity-related cytoskeleton complex, NMDA receptors, and genes whose messenger RNAs bind to the Fragile X mental retardation protein. To my knowledge, we've yet to see a Mendelian disorder. I've heard of a couple of things that might be in the review process, but I haven't seen them directly. And in analysis, there's 10,000 cases and 1,800 trios, markedly more than here. And I can tell you in confidence, based upon sort of pretty good analyses, the same general pattern reported in this paper holds. Next, copy number variation. Again, this is change in the gene dosage over sometimes a single gene, sometimes 20 genes, sometimes more. There's 10 rare but potent examples of this. These are things that are quite rare in the population. You know, 3 in 1,000 cases might have one of these. But they increase the risk for schizophrenia markedly by 4-fold or maybe even as much as 20-fold. The problem with these guys is that they often point to multiple genes, not just one. And if they increase risk for schizophrenia, they probably do something for autism, epilepsy, intellectual disability, and or developmental delay. The PGC they've done a very elegant analysis of, I think it's actually, 30,000 cases. And again, the same pattern pretty much holds. Genome-wide association, again, is a way to look for common variation. I won't say anything to these 2 people leaving because I said I wasn't going to abuse people in the front row. So this was the paper that we published last summer, and this was where my friend typed in 12 hours worth of names. Needlessly, we pulled together data from about 37,000 cases, well over 100,000 controls. There were something like 52 studies 52 samples contributed to this thing. So clearly it was a worldwide effort, tons of people. And in the end, we ended up with 128 independent SNPs in 108 genomic regions. A few of the regions actually had 2. This is something called a Manhattan plot, because the Manhattan plot is what you sort of imagine Manhattan looking like from across the East River at night. So you sort of see these lovely peaks corresponding to sort of imaginative skyscrapers. This is a lovely plot. We were so pleased when we saw this for the first time, because many other times we had these basically, Holland plots. We sort of totally flat, maybe a little bit of a windmill, and that was it, but nothing notable. We went at this really hard. The first thing we said when we saw this result was some version, which I won't repeat, of what did we do wrong? And so we actually went back at this for about a year and a half. Large team of people attacked it from every angle we could think of, and in the end, the results held up and we published. And some of the things that came out of this were really kind of cool. So first on the list is the dopamine 2 receptor popped up as having a nearby genome-wide significant association. Dopamine, this receptor, of course, is where most effective antipsychotics actually bind. And so it's been a major player in the field. The NMDA receptor story is a known one. There's a lot of biology in this. I like this one in particular. There's a copy number variant on chromosome 16p. And Nico Katsanis did a cool zebrafish study where he determined that was, in fact, the driver. It was the dosage of that gene which was driving it. And we have a genome-wide significant association like 2,475 bases away. So very, very close. The hypothesis is that if you change the dose of this by 50% or 150%, it has a strong effect on phenotype. If you change it a little bit with a regulatory variant, it has a more mild effect. The calcium channel story is still there, as I mentioned. I'll go over this later if we have time. But the thing that's real interesting is these processes, among others, actually come up in multiple different types of studies. Neuronal calcium signaling, postsynaptic density, the ARC complex, NMDA receptors, and genes who interact with FMRP. And again, this stuff pops up with an exome study from CNV studies and GWAS. The fact that different types of studies of different types of variation tend to implicate the same pathways, I think is a lovely bit of convergence that I think increases the chance that these are true findings. Now, in terms of what we learned about the architecture, I think for interest, I'm going to skip this slide. The site chip is something we're working on right now, and this is something that has been led by the PGC. It covers common variation as well as rare variation and SNPs that we put on it. It's cheap. It's $100, I think, at most. It might even be $80 if you get it right. We're currently genotyping 100,000 samples, and we're hoping that for schizophrenia, the number of cases will get up to around 62,000 later this year. So the published study has 36,000. We're hoping to get up to 62,000 cases this year. Now, the next potential use of genomic data has to do with therapeutics. In particular, the prediction of adverse drug reactions or treatment response. I think the prediction of treatment response is quite hard, but I'd like to talk about adverse drug reaction. I think this is actually kind of a key output of precision medicine. There are some examples where you have genetic effects on common measurable things like warfarin dosing, but I think its real sweet spot might in fact be for things that are rare and very serious. So Stevens-Johnson syndrome is effectively when you become allergic to your skin and it sloughs off. That's obviously a bad thing to have. Drug-induced liver injury, where some people who are genetically unlucky take a typical antibiotic and they kill their liver and have to get a liver transplant. A lot of these things, curiously, actually end up being part of the same gene, HLA-B. There's a story about the anticonvulsant carbamazepine in Stevens-Johnson syndrome in Asia, where this particular variant of HLA-B has a huge odds ratio. In fact, they've actually started to screen for it in Taiwan now, and the incidence of Stevens-Johnson has gone really, really low. Lamotrigine also binds there, but it's a different variant with a strong odds ratio. Abacavir does as well. And so basically what happens is you get off-target binding of a drug to the pocket of HLA-B. It gets presented as an antigen, and it triggers an autoantigen response. That's the general idea. And we found for clozapine, for people, a case control study of granulocytosis, people's white count really, really dropped with exposure to clozapine versus controls, that the same gene popped up. And we actually found through molecular modeling that it probably also fit in the pocket in a similar way. Unfortunately, the connection here between agran wasn't anywhere near as large enough to be something that we'd want for a diagnostic test. This was really interesting and helps us with biology, but it doesn't help us to determine which patients should get this drug and which patients who can take it safely and who are the ones for whom it might be risky. And then the final point about the use of genetic data has to do with stratification. So it's possible to compute for each person the number of schizophrenia risk alleles, common variant alleles that they've inherited. This is something which is very easy to do right now. And so effectively, we could, for each of you with genetic data, we could figure out how many variants you had on average compared to other people. And so for instance, what we see here is this is the curve in a large sample for controls. This is the curve for cases. The difference between the means is enormously significant. It's about 6 standard deviations. If this were height, it would be on the order of like 4 or 5 centimeters. And so this is a typical example where we've got a shift in the mean, but it's not to the point of being diagnostic or anywhere near diagnostic. It's a probabilistic risk factor rather than a deterministic one. But the thing that's kind of interesting is if you drill down, you can say, okay, what's the risk of being in the second decile versus the first? So you take these scores, you divide them into tens. And what's the risk of being the third versus the first, et cetera? And you see that you get this really interesting relationship where, in fact, if you're in the top decile, your risk of schizophrenia is like 15-fold elevated over people in the lowest decile. This is something we found in 2 samples, one from Sweden, one from the US, and the Danes had a looser definition, which is why you don't see as clear of a relationship. But a couple of things become apparent. So if you actually look at people in the lowest decile, we have a whole bunch of cases who are down here in the lowest decile. Why do these people have schizophrenia? They inherited a very low risk of the illness, and yet they did not develop the clinical syndrome. Conversely, up here, we have about 25% of the top decile are controls. These are individuals who inherited a lot of schizophrenia alleles and yet do not have the illness. What's up with that? I know there are 5 people who are in here who actually have converted over to that group. So maybe it's just a delay in some respects. But this also allows us some interesting possibilities for epidemiology. Can we look at what protective factors these individuals might have? What sort of epidemiological risk factors these people have, low genetic risk yet have the disorder, or what protective effects these people have. So there's some interesting possibilities there. But one of the big uses of genomic data is to try to get at new targets. This empty pipeline is meant to remind us that in many respects, the pipeline is dead. There's nothing in it. I think it's gotten a little bit better, but for most of the last 5 years, many drug companies, in fact, have not been actually working actively on CNS drugs. And that's a huge difference than maybe 10 years ago, when in fact, if you looked at the bestseller lists, often many of the top 25 were actually psychoactive drugs, antidepressants and/or antipsychotics. Okay, so let's talk about assumptions in terms of how this might work. So first, I think the one thing we've learned is you have to be really, really strict about genetic evidence. It's very easy, most genes, given that around something like 10% of all genes have a protein that's expressed at the synapse, it's very easy, and about half of all genes are brain expressed. It's pretty easy to sort of convince yourself to make a story for a gene if the genetic finding is equivocal. A lot of genes are interesting in the CNS. We have to be really, really rigorous about the genetic evidence, or else this is going to be just a complete mess. That's one of the things we view as being very important. Especially in terms of what we're seeing from the combined genomic results, it certainly appears that more of schizophrenia is due to regulatory variants than protein coding variants. So it looks like it's more disease of regulation of the amount of proteins rather than the amount of the actual change in the primary structure of a protein. In addition, again, the technology that we may need may not be something that we have right now. And again, the optimal approach of how we actually try to understand the impact of the emerging genomic findings on the development and function of the brain is simply not known right now. So I think we actually do need multiple integrated approaches, and the question arises, how can we do it relatively quickly? And so what we proposed is effectively trying to take the model that the PGC has, about the model of cooperation, about just really working together in a concerted way to try and make maximal progress. We've taken that to the wet lab sort of approach to this too. And so we've managed to put together a whole bunch of people who are really good scientists who are also really interested in working in a collegial way. These people are good eggs. They're fun to go out and drink beer with. And that really is kind of one of the key things, quite frankly. Because if that's the case, then you can do science together. And so we've managed to pull together a team of people who are pharmacology, from genomics. Nico's a developmental neurobiologist who works in zebrafish. Pamela's a psychiatrist who works a lot in omics study, other sorts of omics studies in brain, et cetera. And these individuals have all agreed to come together to work on this. We even have a sort of a complicated workflow as to how this is going to work. And this is something that we're in the process of trying to actually operationalize as part of my work here. I'll go through this in a sec. So the first step, of course, is target selection. So it's difficult. Not all things we find in genomics are going to be actually things we could work on, even if we wanted to. Some are extremely difficult to work on. Some are straightforward. And again, this is the sort of the crazy scale of data that come into this, to the one pixel that we want to do. But the first thing, of course, is we need to gather all the relevant results, process them in a very systematic way, database them, connect them to other findings. So that's something that we've done already, to be honest. And again, this is something we're going to need to update many times, given that there's a lot of active work happening right now. And the things that are easiest are in many ways exon variation that pops up. And so here what we want, the reason why it's relatively straightforward is if a gene, for instance, has a lot more variants in kids with autism, protein-changing variants in kids with autism than controls, that becomes something relatively straightforward to model in many respects. You need to do some work to make sure that you're not getting fooled. There's some complexities where that can be a problem. But typically, the great thing about it is that it connects directly to a gene, and you have a direction of association of the effect. In other words, the variants occur in the gene, and they lead to the loss of function of that protein. And that's something that we can take and model in multiple systems. We don't have anything like this yet for schizophrenia, but there certainly are a good handful of things for autism, and larger studies are in progress. And we've got some ideas about how we can actually get more discovery there as well. The thing, oops, this was supposed to be different. Actually, go back. So one of the things that I think is pretty important, at least in one level, is to try to determine what proteins connect each other. And this is the whole protein-protein interaction piece. This is a slide from one of our colleagues, Mark Vidal, who's at Dana-Farber at Harvard. And Mark has sort of shown that The protein-protein interaction data out here are highly biased and highly focused on a handful of targets. So typically, these are things involved in ubiquitination as well as cell cycle control, because those are the ones that have been studied really heavily. P53, for instance, is a big one here. Whereas you can actually find that a lot of genes that are important in the central nervous system haven't been studied at all. And so what we propose to do is actually use two different sort of complementary technologies to look at protein-protein interactions that are relevant in neural tissue. I don't think I have time to go through the details, but each of these can be done in a relatively high throughput fashion. So one technology can handle 100 different genes per year. The other can handle several hundred. And I think that information will allow us to sort of take a protein that we think is involved and expand out to find out what it connects to. The guys that do this are very good. There's a lot of biases, but they're pretty good at figuring out and cross-checking assumptions. The other technology that we can bring to bear is something called high-content screening. This typically happens in a 96 or 48-well format, where you have a nest of neurons that can be either from a person, they can be from a mouse, they can be from a standardized human neural stem cell population, differentiated in neurons, and then we can manipulate them in many different ways. We can knock genes, we can remove genes in singly or in combination, we can increase the expression of genes, and it's possible to, using automated new microscopy and analysis techniques, to actually come up with a fairly solid list of 40-odd parameters per nest, per manipulation, and again, to do this in a matter of weeks. There's also a very in-depth synaptic approach where we work with a synaptic biologist, Matthias Verhaeghe, in Amsterdam. And he's spent a lot of time and money to come up with a way to look at single neurons and then to characterize them with great depth. So for instance, they can do all the synaptic physiology. They can actually track individual packets of information through the cells. They can look at time course. They can do a lot of different things as well. And again, these can be manipulated as well. So to come back to the pipeline, so the first thing we want to do, of course, is to sort of understand whether there's any patient heterogeneity that might be involved in an exon finding. So we're looking at one specific gene that came up, clearly with very, very strong evidence. We would apply the two proteomic techniques to understand what it binds to and how the variant changes binding. There's a sort of a computer science bioinformatic approach here. And then the point is we'd have about 30 people sitting down, looking at the results and thinking, okay, what's the next direction? If it's something where we think it's worth going further, high content screening, and actually doing single neuronal assays are possible. And if that's the case, we actually have some ways where we could actually screen things at relatively high throughput. And then there's similar pipelines to this for copy number variation. The one that I like in particular are single gene CNVs. So here you've got a situation where cases have more copy number variants that hit one gene than do controls. So again, you've connected to a specific gene, and you have a dosage effect. You know that cases have half the number of copies than controls. You need to do some validation work, et cetera, but typically, this is probably the most actionable class of variants we have for schizophrenia. There's at least 3 of these right now for schizophrenia. We have a 4th in the publication pipeline that we like a lot, and I think it's true. But again, this is something that my neuroscience colleagues can actually model. They can change the doses of a gene in many different ways in many different systems. For the very large CNVs, we need a driver. If some of these won't be doable, 22q11 probably has 3 drivers. 16p11 looks like it has 1, and that might be something where we could focus on that gene to see what's going on. And we're doing some work with Nico Katsanis on this right now. And then the one that's actually tricky is GWAS, because typically the signal is subtle and it's relatively broad. And so the connection to a gene and the direction of effect isn't clear. However, it certainly is possible to make this connection. I've got a folder where I keep track of these, and there's several dozen right now, of where people have done the follow-up work and managed to clearly connect, experimentally, a GWAS finding to a gene and with a direction of association. It's more difficult. It's not just a simple lookup. It's not just an assumption that a loss of function means something. You actually have to do additional work to figure it out. But one of the questions we have is how do you evaluate that at high throughput? And here are 3 associations with schizophrenia that I just wanted to talk through. They're all genome-wide significant. But as you can see, it's quite complicated. So here we've got one of the calcium components, the L-type voltage-gated calcium channel, the neuronal type, CAC and A1C. And all of these are genome-wide significant, even some over here, and they all lie in intron 3. And the question is, of course, how do you connect this to this gene or maybe a different gene? Here's another example where we have a big area of genome-wide significance, but as you can see, there are many, many genes in the region. And here's a third example where we've got an area of genome-wide significance, but these are nothing. There's actually no gene within a couple 100,000 bases either way. And the question is, how do we actually understand what these are doing to change the risk for schizophrenia? And the answer is I think you actually have to bring in different data. But it's possible to create a very logical workflow as to what you might do. I mean, the first thing one would need to do is to do the DNA sequencing to make sure that you've assembled that region correctly, that there isn't something funny going on there, because occasionally there is. Second, you need to do some sequencing in brain to see whether there's any unknown transcripts that are coming off that region. Maybe there is a gene there, it's just not annotated. Next, we want to see whether it's actually differentially expressed. An eQTL study is a way to look at genetic variation and how it affects the amount of transcription. And so there's a bunch of large-scale data sets that have looked at this, increasing numbers in brain as well. ATAC-seq is a way to look for open chromatin. And of course, looking at histone marks, whether the SNP actually has to do, whether it actually changes the repressor, enhancer, et cetera, in the usual way. And then finally, I think something that's really important is that there's a lot of these looping interactions, especially in brain, where you have an intergenic genome-wide significant association, and there's no gene there, but it has a regulatory effect on a gene that's 300,000 bases away. We're seeing increasing examples of this. What we need is we actually need a set of data where this has been done very carefully, relatively large sample sizes in neural tissues. Fortunately, we've done this in a pilot work in my group, and there's actually a bunch of large-scale efforts that are happening right now. We're part of something called Psych-ENCODE, where we're, like the ENCODE project, we're doing this, but unlike ENCODE, which focused nearly exclusively on cancer cell lines and just regular cell lines, we're doing it all in brain. And so there's an open source effort to do this. Pamela Sklar is one of the principals of the Common Mind Consortium, where they're actually doing a lot of sequencing in brains of people with schizophrenia versus controls, as well as a lot of the ENCODE type stuff as well. Okay, so that really comes down to this right there, which is the functional genomic data. So I think it's possible to take a GWAS association, overlay other examples, and come up with a hypothesis as to what's happening. It's also then possible to design a wet bench experiment to prove it, to see whether or not that hypothesis is correct. And that's the next step that I think we need to do. Okay, so final slide. I wanted to thank all kinds of people, to be honest. I've got 2 labs now, and I owe them a huge debt of gratitude because I learn stuff from them every day. I started coming over here back around just before 9/11, actually, to do it with Nancy Pedersen. Expanded out to work with Paul Lichtenstein, Christina Holtman, Patrick Magnuson, Sarah Bergen are all people we've done a lot of work with. And I certainly owe them a great debt because I wouldn't be standing here if they hadn't been so kind to me over the years. The Psych Genomics Consortium, I'd like to thank the cast of hundreds. The Swedish Research Council has been extremely kind as well to allow me to come here. And then we've had many, many funders who have really made critical contributions along the way. And I thank you for your attention. And I think we have time for questions.
And the themes that are there, and they're going to continue to extend around the need for our education, not only ours, but the providers, the clinicians we're collaborating with, and as we transfer all that to our patients. There are definite themes among all of these talks. I'm very happy to start our afternoon session by introducing you to Dr. Janine Austin, who's an associate professor in the departments of psychiatry and medical genetics at Vancouver, British Columbia. She is holding the Canada Research Chair in Translational Psychiatric Genomics there, having done a PhD in neuropsychiatric genetics at Cardiff, which is in Wales, if you're not aware, and then trained as a genetic counselor after her PhD at the Vancouver, British Columbia program. She has a very special interest in psychiatric genetics and genomics, and having lectured now on several continents, bringing awareness to what the genetic counselor can do in this very complex area of medicine, and as more and more genomic information becomes available to us with this complex area. I think we're going to be hearing a lot from her now and forever. She tweets all the time. But she's also, in 2015, chairing a session of the Banbury meeting at Cold Spring Harbor on psychiatric genetic counseling, and has been elected as our new president of the National Society of Genetic Counselors. So we are very happy to have you here, Ms. Janine.
Thank you. So I have the unenviable job of trying to keep you all awake after that delicious lunch. Let's see how I do. Oh, and while we have this technical moment here, I promised some of the students last night who shared fun facts about themselves that I was going to share a fun fact about myself today. So my fun fact is that I am actually a cave diver. So that is spelunking in water-filled caves. It's something I love. I've been doing it for 10 years and I'm not dead, so that's good. Okay, so I'm going to be talking with you for the next little bit about clinical applications of psychiatric genomics, and so specifically applying genetic counselling to improve outcomes for people with psychiatric disorders and their families. I should say, no big disclosures. I don't have any, unfortunately, don't have any financial relationships with any companies that will pay me lots of money. I'm just employed by UBC and have held grants from all the regular kinds of agencies that you would expect. And so when I say that I'm just going to go back to my title slide for a second. So when I say I'm going to be talking about psychiatric disorders, I do want to be really clear. My special interest is primarily in psychiatric disorders of adult onset. So I'm not really going to be focusing on autism today. I'm going to be focusing on things like schizophrenia, bipolar depression. OCD will come in there a little bit perhaps, but I don't do autism because lots of people are doing that very, very well already. I wanted to focus on stuff that isn't really being addressed in the clinical genetics community as much. So 3 learning objectives for the next little bit of time I've got with you, and we're going to start out with this bit. And I'm going to do a terrible disservice to the entire psychiatric genetics community by summarizing an entire field in about 3 slides. But it's doable, if you bear with me. Sorry, this is here for my amusement rather than anything else. So this is a slide that, until very recently, if I was trying to summarize for you what we know about the genetics, genomics of psychiatric disorders, I could have just put this up. And I could have talked around it a bit, but this would kind of summarize it a little bit, that we had a corner piece of this 3 billion piece puzzle, but we didn't really have very much more in the way of specifics than that. However, in the last couple of years, things have been changing quite quickly and quite dramatically, and I now have the immense pleasure of showing you the next slide. So this is only exciting to me, I think, but that's okay. So this is a Manhattan plot for those of you who don't know, and so what you're looking at, so the idea is it's a way of visualizing genome-wide association study results. So across the x-axis there, you've got all of the different chromosomes, and the y-axis represents the p-value. And that little horizontal line you see in red across close to the bottom there, that's the threshold that we use for statistical significance when you're doing a genome-wide association study. So we're looking for signals that appear above that line. And so it's called Manhattan plot because if you've got a good one going on, you should have lots of peaks, so it looks like the skyline of Manhattan. So the running joke in the psychiatric genetics community for the last 10 years or so has been that our Manhattan plots look like Omaha. Because they have looked like Omaha. So there's been really nothing coming above that little red line that you see right there. But as you can see right here, this is no longer true. So this came out just earlier this year. This is the findings of the latest genome-wide association study for schizophrenia. Was just published in Nature, and we've got over 100 new hits all of which surpass that threshold for genome-wide significance that we're looking for. We've got genes in there that include things like zinc finger proteins, calcium channel proteins. The big one that you can see pointing right up there, that's the major histocompatibility complex. There's all sorts of different things in there that we maybe weren't expecting to find. But just to give you an idea, like why did our plots used to look like Omaha and how come they're looking like Manhattan finally? Really, it's a function of sample size. When you're talking about disorders that are as complex as these psychiatric disorders are, the individual variants make very small contributions individually, these kinds of SNPs anyway. And so basically we weren't finding anything because our sample sizes were so small. This study involved over 30,000 cases and 60,000 controls, only possible because of the formation of huge international consortia. This paper actually had 380 authors. So it's a big deal, right? These kinds of studies take an enormous amount of manpower and people power, I should say. Yeah. So that's pretty exciting. And with that, I will move on. So we have to remember, though, of course, that this is just one particular kind of genetic variation. That these are single nucleotide polymorphisms that we're looking at here. And we do know that psychiatric disorders are not just complex in that genes and environment interact together to contribute to the etiology of these conditions, but different types of genetic variation also play a role. So this is just a very brief table for you. Don't get too hung up on the details. What I do want to draw your attention to is how you've got variations here in two different colours. And the ones in blue that you can see at the bottom, those are your single nucleotide polymorphisms. And what I do want to draw your attention to is the odds ratio for those. It's tiny, tiny, tiny. So even that biggest hit that you saw on that last slide, and we're actually just going to go back to it to have a look. So this is only telling us about the p-value. So it says it's very statistically significant, but that doesn't tell us anything at all, remember, about how big of a role that variation plays, right? So what we're seeing is that these kinds of single nucleotide polymorphisms have a very, very small contribution individually. But what you see in purple, though, is copy number variations. And there's been a lot of work on those too. We've got a whole bunch of them now that have been very reliably implicated in increasing a person's vulnerability to psychiatric illness. So we'll apologise. The last slide I showed you was about schizophrenia. I showed you that one because schizophrenia is really the furthest along. But all the others are coming along very well too. Like we've got something similar coming out for bipolar disorder, that kind of thing. But just to give you a sense, I'm focusing on schizophrenia. The other thing I want to bring your attention to here is just those last three lines of the table. And if you can't see it, it's showing three variations that are not just implicated in schizophrenia, they're also implicated in bipolar, ADHD, and autism, and major depressive disorder. So this reflects what we've actually known for a very long time, but it's just very pleasing to see it coming out again in the genome-wide association studies, that the variants that contribute to vulnerability don't have specific or discrete effects, if you like. So it's not that this variant in particular only increases your risk for schizophrenia and you have a different one for bipolar. It seems that most of the variants increase one's vulnerability to mental illness in general, and then it just depends on what specific combination of variants you might have that ultimately leads to the manifestation of whatever. Okay? So that's lovely. So we've talked about SNPs, we've talked about CNVs, fabulous, but just to step back even further for a second, we know that that's not all. We know that these are complex conditions, that there seems to be a role for the environment, as we can tell from looking at the difference between identical and non-identical twin concordance rates. So none of the concordance rates for identical twins for any of these conditions is 100%, and that's obviously what we would expect if the environment is playing an important role here. So not to lose sight of that, I think, is all. So where does all of this leave us with regard to genetic testing? Well, sadly, at the moment, for some of us perhaps, there is no genetic test with which to establish, confirm, or refine a psychiatric diagnosis. Psychiatric diagnosis remains something that's based on clinical interview, so inherent subjectivity to that. And in fact, this second bullet, family history evaluation is more useful but less sexy than SNP testing to predict risk for psychiatric illness. Actually, I haven't updated my slides in the last couple of weeks, and I was actually I maybe wouldn't be quite so strident about that anymore. There's actually a bit of an ongoing discourse in the literature at the moment about whether or not polygenic risk scores might be approaching the utility of family history in the context of psychiatric illness. So polygenic risk scores are just a way of trying to take into account all of those different peaks that we were seeing on the Manhattan plot and sort of add them up together to see what happens, kind of thing. So there's a bunch of different ways that you can evaluate whether a polygenic risk score is useful or not, but the most commonly used idea is the area under the receiver operator characteristic curve. And I know none of this really matters. The only thing to know is that an area under the curve value of about 0.5 is essentially useless. It means like flip of a coin. You're going to be able to distinguish between somebody with a disease and without a disease about, flip of a coin kind of thing. An area under the curve value of about 0.99, diagnostic, pretty much. Saying, you can discriminate between people with and without the disease with really good reliability. But and then an area under the curve value of about 0.75 is where we start thinking that you might be able to use that test as a screening tool. So in the same way as we might use a maternal serum screen as a screening tool, if you've got a test that has an area under the curve 0.75, and some people are arguing that that's what we're getting for schizophrenia. I'm not entirely convinced by these arguments yet, but the point is that this is coming. This field is moving very quickly. So that's exciting, but just a quick timeout for myself, I think, more than anything. But it's important to remember that even if we do get to that point, these tests are still going to be relatively expensive, and so that has to be weighed up with the utility of family histories as well. Quick word about CNV testing. There was a paper that came out last year saying that clinically, quote unquote, clinically significant copy number variations could be found in 8.1% of people with schizophrenia. There's actually, I would actually contest that a little bit, the way they interpreted the data. Very good paper, but it's actually a little bit overstated, I think, in some ways. So actually, CNV testing is still only of the most utility in family members of people known to carry CNVs or in people with psychiatric illness and other problems. And then, of course, we've got this whole area of work that's trying to look at psychotropic medication response according to genetic variations. Okay, so given that I've basically just told you that where we're standing right now, there is no genetic testing that can confirm, predict, refine, blah, blah, for psychiatric disorders, what can we do in genetic counselling for people who have these conditions and their families? Well, so I know you may be reading this and going, really? That's really boring, isn't it? That's really boring. And maybe it is to us, but I think we get stuck in our viewpoint sometimes. So understanding that psychiatric disorders are themselves not usually inherited, but that we can inherit a vulnerability to mental illness, that sounds really mundane to us. This is something that we know so intuitively, and you think, how can that possibly be helpful? I'm going to share with you some stories that illustrate how that can be really helpful, actually. And just even the understanding that genes and environment usually work together to produce illness can be really important and powerful. And I argue very strongly, for those of you that know me or have interacted with me over the last day or two, that counselling around the emotional issues that are attached to the explanations for cause of illness can be absolutely profoundly powerful. And again, I've got a couple of little stories that hopefully illustrate that for you. So let's move into that area then. Let's leave the genetic genomic stuff behind and move into the application part, because that's where I have the most fun, at least. So let's think first about why it's so important to people with psychiatric disorders and their families to understand cause. I'm actually going to take that away to stop you reading it. Sorry about that. It will come back in a second. So if you just think about your own experience, right? As human beings, we are meaning-making machines. If any of you have ever had a bad thing happen to you, and I'm sure we've all had at least one bad thing happen, you fail an exam, you crash your car, one of the first things you want to know is why. Why did that happen? And the reason that you want to know that is because you want to prevent that bad thing from happening again in the future. You crash your car, maybe you think, okay, well, I was texting while I was driving, so maybe if I don't text while I drive in the future, I can reduce my chance for having another episode of that bad thing, i.e. a car crash. Same is absolutely true for things like psychiatric disorders. Bad thing, definitely. And so in the absence of being provided with a really good, coherent explanation for why it happened, people will generate their own based on their experience. And unfortunately, those explanations that people come up with usually invoke things like guilt, shame, stigma, stuff that's absolutely counter to healthy adaptation to managing an illness. Okay, so I've got just a couple of quotes to share with you that I think illustrate some of the ways that those guilt, shame, stigma things can manifest in people's own explanations. So this is a quote from a mother of a child with Asperger's syndrome. She says, "The feeling that we somehow caused this is strong. This happens because we're judged harshly due to our child's behaviours. I was lectured by family members about our parenting skills." So this could be the mother of any child with any psychiatric disorder, just to say that straight. I mean, the word Asperger's is irrelevant here. It just happens that that's what her child had. It could be anything. This is such a common sentiment to hear, that people are told so often, you must be a bad parent, that they kind of internalize it and believe it themselves. So you may be thinking, okay, well then that's great, because all we need to do is tell people that genetics plays an important role and that'll fix this problem, right? No, of course not, because that would be too easy. So this is the flip side. This is a father of a child with bipolar. He says, "It came from my side. I've got the guilt. If I hadn't had him, he wouldn't be like that. If I'd known more at the time, I probably wouldn't have had any children because of what I've seen happen to him. I didn't think about this being passed on when I was 23 years old. You think, this will never happen to me. I would have made different decisions if I'd known." So this father didn't actually have bipolar disorder himself, but when it emerged in his child, literally the first thing he realized, with that terrible sinking feeling out the pit of your stomach, is, "Oh, Aunt Rose had this." Right? So he immediately feels responsible for having passed on the quote-unquote bad genes that have caused this. Yeah, so this is really powerful stuff for families, their understanding of cause. So fundamentally, as we've already discussed, everyone has a explanation for cause. And what I've found over time is that when people have genetics-only explanations, it tends to drive people towards this fatalistic type of approach, like there's nothing I can do, this is just what I have, don't bother trying to help myself, you know, that kind of thing. And it can be really hard to engage those people in non-pharmacological treatment, which can be really helpful. Talk-based therapy, you know, managing sleep more effectively, that kind of thing. And it can also lead to over-vigilance with other family members, so real worries about, you know, thinking everybody else in the family is predestined to have the same problem. Whereas environment-only explanations tend to really get more into this guilt-blame thing. Oh, it's my fault, I'm such a bad parent, such a bad sibling, whatever. Or such a bad person, it was my fault that I smoked all that pot and that's why I have schizophrenia, that kind of thing. And with these people, it can be really hard to engage in pharmacological treatment, because they're like, well, why should I, what sense does this make? If I just tried harder, it would get better. And so real impact on social interactions because of these internalized feelings of guilt and shame. So given that, let's just remember for a second what genetic counselling is. So I know I'm going to do this because I know that sometimes we tend to get stuck on thinking we conflate, even as genetic counsellors, we conflate what we do with genetic testing or with pregnancy and things, but it's not. That's absolutely part of what we do, but that's just the tip of the iceberg. There's a whole lot more to it than that. This is the definition that NSGC adopted in 2006 from the Rester paper. So it's a process of helping people to understand and adapt, that's the bit I really like, and adapt to the medical, psychological, and familial implications of genetic contributions to disease. So when I know that's a pretty long, dry sentence, but if you think about what it means in relation to those two quotes that I just shared with you, then it starts to get powerful, at least for me. So the informational component of what we do as genetic counsellor is really targeted towards the helping people understand bit. And in the context of psychiatric genetic counselling, things like how that relates to recovery and reducing risk for relapse, chances for other family members to be affected, what we know from research about how these illnesses arise in the first place. The support component is really about identifying and addressing the guilt, shame, stigma stuff that often revolves around people's existing perceptions of cause. Okay. So now we're going to get into the funnest bit, which is, oh, hi. Sorry, quick photo. So now we're going to get into the funnest bit, which is about talking about the brand new, well, no, it's not new anymore. It's 2 years old. So in 2012, based on the data that we've been generating from my team's research over the last 10 years or so, I was able to convince our Provincial Health Services Authority to give me money to start up a specialist psychiatric genetic counselling service. This is the first, and no longer only, but it's the first one of its kind in the world, so we're deeply proud of what we're doing. And this was just an article that came out about it in one of our local newspapers. And that's me talking with one of the patients that's been seen in the clinic, whose name is Victoria Maxwell, who's absolutely fantastic. So I'm going to tell you a little bit about that. First of all, just the dry stuff, like what does it actually look like? So we have, it's a province-wide service, so basically so here it would be serving the entire state of South Carolina kind of thing. So it's serving the entire province of BC. And at the moment I have one full-time counsellor who is actually embedded within our general genetics program. And so she's physically located there, but all she does is see people with psychiatric disorders and their families. And we see men and women of all ages. So one of the big problems that we've had in communicating with the population in BC about this service is that people hear genetic counselling as a term, and the automatic association is childbearing decisions. So they're like, where do I refer my pregnant women? I'm like, no. Yes, I mean, we'll see your pregnant women, but it's not just about pregnant women. So men and women of all ages who have a personal or family history of any psychiatric disorder. We accept self and healthcare provider referrals. We're accepting self-referrals because we wanted to make the barriers to access as low as possible. And we know that some people with psychiatric disorders are not going to have those relationships with a healthcare provider that would mean that they'd feel comfortable going to get that referral. So in order to make the barriers low, that's what we started out accepting. We do family history prior to the appointment over the phone. The appointments themselves, when people come in, or we do it by telehealth or telephone or whatever, or outreach, because we are a provincial service, the appointments themselves take about 1 to 2 hours. One of the really cool things about defining a new subdiscipline is that you get to set your own terms. So I've been able to say, literally, I don't think this model of half an hour per patient is really one that's going to work for me. What I want to do is improve patient outcomes. This isn't about get as many people in and out through that door and who cares what the outcomes look like. This is about trying to make a positive difference for people, and if that means that we have to spend an hour and a half talking with someone about what we know from research about the causes of illness and how that relates to what you can do to protect mental health, that's what we'll take. So that's quite a privilege to be able to sort of set those terms for ourselves as we move forward. And we've decided as well that 6 new patients a week is all we can handle. That's full caseload in this context. They are you might imagine, they're quite draining sessions, or at least can be. You're talking about family histories of suicide, abuse, all sorts of stuff can come up. That's one of the reasons we do family history beforehand, so that when people come into the session, they're not already beaten up and, by having just spent 45 minutes talking about terrible family history stuff. They've had time to come down again, and they're able to attend to the conversation that we have together. So also, if people show up at the session, or it turns out in the session that it looks like somebody might have a syndrome, we're not providing testing for that. We're re-referring them back into the program for them to be evaluated by a medical geneticist. And we're not currently ordering any genetic testing because there's nothing that really makes a whole bunch of sense to us at this point in time. So, a couple of cases to share with you. How am I doing time-wise? Will you give me a 10-minute call? Yeah, thank you. So the first case I would like to share with you is, and of course I've changed key details so that she's not identifiable, but yeah. So it's about Anna, who was 10 when we saw her. And when I say we, in this case I'm actually referring to Emily, who I'll show you a picture of at the end. She's the counsellor that I employ as the full-time person in this role at the moment. So Anna came in because she'd recently been diagnosed with an eating disorder. Her parents were super distressed about the diagnosis, and as it turned out, through the conversation that Emily had with them at the beginning, they attributed her condition entirely to genetic factors and were feeling really helpless, like there's nothing that we can do kind of thing. Anna herself felt weird. She said, it's genetic, I'm different kind of thing. And so she's withdrawing from her friends. So what happened in the session was we have this model. I don't know if you've seen it or not, but I'll be giving you information about how to find it at the end if you're not familiar with it. This is the JAR model that we use. It's very, very simple, but you can actually use it to talk about all sorts of very complex things. You can use it to talk about epigenetics. You can use it to talk about CNVs, SNPs, or you can use it at a very simple, superficial, simple level. The concept is, of course, that everyone has a mental illness jar, and that in order to be actively experiencing an episode of illness, the jar has to be filled up to the top. And there are two different kinds of vulnerability factors you can fill your jar up with. The genetic factors are represented by the yellow circles, and environmental factors are represented by the triangles. So anyway, we used this model with Anna and her family, sort of reflecting it back onto her experience and the family history. So it turned out actually that Anna's mother had experienced an eating disorder as well when she was younger, and that was a good source of guilt for her, as you might imagine. So they were asking very specific questions about what genes had been implicated in eating disorders, and we were able to address those questions, but we were really careful to bring it back to the key message, which is that it's not just genes and environment acting together. And we also talked about how lots of these SNPs are super common. Everybody probably has some degree of genetic vulnerability to mental illness. It's just that some of us are going to have more, others are going to have less. Anna absolutely loved this. So she was like, because she came in feeling kind of guilty kind of thing. So she got it really engaged at this point and started talking about what she thought some of her Doritos in the jar might have been. So she thought the triangles looked like Doritos, and which was pretty cute. So we had a really good talk about that. So the other really cool thing about psychiatric disorders is that you don't just get to talk about how people get sick, you also get to talk about how people can recover. And the protective factors in this model are really just rings that are stacking on top of the jar. They're not making a lid, they're simply making the jar taller so that it can accommodate more environmental factors, if you like. And so we talked about that, and Anna's mum got really into it. She started talking about the factors that had helped her recover when she'd experienced an eating disorder when she was younger. And long story short, the family left feeling a good deal less helpless. So both of the parents were actively strategizing with Anna about things that they could do to protect her mental health and to help her recover. Oh, really? 10 minutes? Oh my goodness. Okay. All right, I'm going to whip it. This is the best bit. Okay. Another quick story. This one's about Bob. Bob I counselled. So Bob I saw when he was an inpatient in a psychiatric unit in Vancouver because he'd recently attempted suicide. He was there with his psychiatrist fighting about medications. The psychiatrist was saying, if you want to get better and you want to get out, you have to take these things. And he was like, So there was a lot of headbutting going on and nobody was getting any better, and it was not a good scene. So I sat down with him and asked him about his, chit-chat, get to know you. Turns out he has a master's degree in psychiatric genetics. We had a total geek-out session because my PhD is in psychiatric genetics. So we were like, blah, blah, oh, you were at that conference too, so was I, la, la, la. We didn't know each other then. So I made an assumption at that point that I knew what his answer to my fundamental question was going to be. My fundamental question is always, can you tell me what it is you understand about what caused your illness? I almost didn't ask him because I assumed. But he floored me. He said bad life decisions. I was like, what? Because we were just talking about the whole psychiatric genetics master's degree thing. And he was like, yeah, I understand at a level of the population, genetics is really important. But in my own specific case, it was this that I did, and that I did, and the other thing that I did. So took a family history. Both of his parents had actually been affected, not with schizoaffective disorder. His mum had recurrent depression. His dad had bipolar disorder. So I took this, documented it out, turned it around to show him, using the JAR model, reflecting on his family history, dissolves into a puddle of tears. Was able to explain what was going on by saying that he felt like a weight of guilt had been lifted. Perhaps it wasn't all his fault. So profound. He'd been living with his diagnosis for 20 years. It took me an hour and a half, which makes me feel great and terrible all at the same time. Great, yay me, I made a difference for someone. Terrible because it wasn't that hard. And any one of us sitting in this room could do that, and we haven't. We don't reach out to this population. But it got even better. 1 month later, which is when we typically follow up with people, he was able to explain to me that once he understood there was a biological contribution to his own illness, then a biological treatment started making sense. So he's now taking his psychotropic medications, and his mental health is much better than it had been in years. Like, not a genetic test in sight, not a pregnant person in sight. It's, yeah. A last story, just because I love them too much not to tell you. So Cassie and her sister both had a diagnosis of OCD. John is Cassie's partner. John and Cassie come in together because they want to understand more about the causes of OCD. And John in particular is talking about, he wants to understand why Cassie's sister's diagnosis, why is her OCD so much more severe than his wife's, than Cassie's. He had a perception that environmental factors were a lot more important in the etiology of OCD. So Cassie's sister had experienced a traumatic event before the onset of her symptoms, and that was his explanation as to where it had come from. So, as we're sitting there in the session, and John's going on and on about how her sister's OCD is more severe, Cassie breaks down in tears, completely loses it, and basically explodes at John, saying that she feels he completely minimizes and avoids her diagnosis and pretends it's not real. So she did actually experience her OCD to be very severe. It's just that it wasn't in the overt ways of her sister, who had like really red raw hands from excessive hand washing. So Cassie's stuff was more internal, less visible. So he wasn't appreciating that for her. So turned out John was absolutely terrified of thinking that genetics had anything to do with it, because they wanted to have children. So, and this caused him to deny Cassie's experience. So basically the session was about facilitating dialogue and shared understanding of cause between the two. And they, again, basically both left making a pact to work on protective factors together. So genetic counselling in the context of psychiatric disorders is really walking the fine line between saying, it's not your fault, but there are things you can do to protect your mental health for the future, right? That's a fine and delicate line to walk, but it is walkable. And it's also about moving people from extreme positions, so thinking it's genetics only or environment only, to getting them to a more moderate position that actually reflects what we know from research about the causes of these conditions. And as you might be able to tell, I also find it extremely rewarding. Yes. So just to very quickly talk about the so I've given you 3 lovely narratives, so real life stories about how genetic counselling has affected individuals. We also, if you're more of a numbers person and you're like, yes, whatever, give me the p-values, I also have that for you. And this was a paper that was published just earlier this year in Clinical Genetics, if you want to go and find it. But this is the crux of the paper, I suppose. So we administered 2 scales before and 1 month after genetic counselling in the context of our clinic. So we administered the genetic counselling outcome scale, which for any of you who are not familiar with it, is a fantastic instrument. It basically measures empowerment, and it was designed and validated specifically for use in genetic counselling populations. So we used that to measure empowerment, and we used a self-efficacy scale also. So there's a couple of things here. What you're looking at is pre-genetic counselling, post-genetic counselling as the 2 columns, and the y-axis relates to the score on each of the 2 instruments. The higher the score, the better the level of self-efficacy, or the better the level of empowerment. So higher scores equals good, basically. And so what you can see is lovely p-values, both significant, but what excites me is not so much the p-values, it's more the effect size. Because as you probably know, right, you can find statistical significance for something that has a minuscule effect if you just look at a big enough sample size. But what these d values, that's a value of effect size, and d values of about 0.4, 0.5-ish is a rule of thumb that sort of reflects moderate effect, which is a rule of thumb for sort of clinically significant kind of thing. So I actually didn't know that D values came as big as 1. I had to check that like 3 times. I've only ever seen them in the region of about 0.8 before. So this was incredibly gratifying to me. So not only is it statistically significant, it's also clinically significant, the effect that we're having. And I'm just going to finish with a little bit of evangelizing. So if I've managed to inspire anyone today, and I genuinely hope I have, we've had people visit our clinic from all over the world so far, and we've already got additional clinics started up, one in San Francisco, one in Cardiff in the UK. I don't want to be the only one in Canada, and I would love there to be more than one in the US. So, like, we've got evidence that people want this. We've got evidence that it can help. From the genetic counselling perspective, it's incredibly rewarding. So if anything that I've talked about appeals to you and you'd like to give it a try, well, you actually have some fabulous resources here that you can use. But as well, I'd like to help. So do contact us. And this is a book that Holly PA and I wrote that was published just a few years ago. It's a very practical how-to guide about how to do psychiatric genetic counselling. You'll find that jar model that I showed you today in there. And the counselling aids and patient resource take-home booklets you can find for free download on the NSGC website, multiple languages even. No excuses. And I just want to leave you with a picture of my fantastic team that, without whom none of this stuff would have been possible. And with that, I'll stop and take any questions.
I have a question. Since in the United States we're driven by other factors other than the ease of time spent and whatever. But when you do your initial phone call via family history, what is the average time that is spent on gathering that family history with a phone?
Yeah, no, that's a great question. So doing a detailed 3-generation psychiatric family history can take quite some time. So it takes about 45 minutes, I'd say, on average. And the reason that we put so much effort into it is because we don't actually go off and collect medical records. We don't do that for a whole variety of reasons. The nature of the conditions in question mean that family relationships often break down, so getting records from other family members can be challenging. You can waste a whole lot of time going nowhere using that sort of strategy. Also because they are subjective, there's an element of subjectivity in the diagnoses. They can change over time, yada, yada. So the genetic counsellors in my team are all trained in psychiatric interviewing. So they're very careful. So if they, somebody's saying, I think so-and-so in the family has a history of schizophrenia, they'll be able to ask targeted questions to sort of try and figure out if that's really what's going on or not. Not to make a diagnosis. Obviously, we work with a psychiatrist that we collaborate with on that. But so about 45 minutes.
Where do you get that training?
Come see us. It's so you can get that sort of training through working with psychiatrist colleagues who might be willing to help. That was how I initially got trained, and then I've shared it with my group then over time. But we've had lots of people come visit our group from all over the world to get exactly that kind of training, and so we will, if you want to, just drop me an email and we'll figure something out. Thank you very much.
Thank you, Janine. That was fabulous. And I'm happy to present our next speaker as well, Melissa Bennett, who is also one of our various distinguished alumna. Having done her undergrad at Auburn, she has been out in the field for several years now. I'm thinking 1999, Melissa. She did her first clinical position in perinatal genetics in Fort Worth, and then quickly moved into the laboratory arena, where she started at LabCorp and began through that process there over several years to ascend to become manager of the clinical services there in their molecular biology and pathology departments. From LabCorp, she moved on to Myriad and once again began an ascent up to Director of Medical Affairs, responsible for a lot of the genetic counselors in the workforce for Myriad. And more recently, she's moved over to CareCorps National, where she's director of the laboratory management program, and she's literally working on medical policies for that organization in laboratory utilization management. If you don't know that term yet, laboratory utilization, you need to plug into it, because all of us deal with it every day as we select tests for our patients, and our insurance providers are also pushing back and selecting which laboratories we're using. And so the purpose of today's talk is to help us understand more of that perspective from Melissa Bennett.
This presentation is going to be talking with families about psychiatric illness. Strong scientific data support the assertion that major psychiatric illnesses, including psychotic disorders, mood disorders, anxiety disorders, and autism spectrum disorders, are typically caused by combined influences of both genetic and environmental factors. Research data allow us to provide clients with more information about these contributing factors. Thus, it is important when we interpret and apply such data for clients, we keep in mind all of the implications of the information and services that we provide. Our objectives for today's lecture are to identify terminology that is often used in explaining psychiatric illness to families, provide some examples of what you can say to clients and family members about the genetics illness, provide some examples of client and family issues that might be raised about genetics and psychiatric illness, and finally, provide some basic information on the genetics of 4 mental health disorders. I will not be covering Huntington's disease. If you'd like that, I have another lecture later on. Let's first begin by providing a foundation that would allow you to build on understanding of more complex issues specific to psychiatric illness. What I'm going to do here is define some terminology for you that might be useful in helping explain psychiatric genetics to families. When you begin counseling with families, you must know your terminology. Here are some basic words that you will need to be able to define in order to help families understand psychiatric illness. Deoxyribonucleic acid, the word genome, chromosome, gene, genetic variation or genetic variant, alleles, genetic susceptibility, complex or multifactorial traits and disorders, genetic syndrome. Now let's talk about what you can say to clients and family members about genetics and psychiatric illness. You might say, genes are the instructions that tell the body how to grow, develop, and function. Starting with a brief general introduction to the topic of genetics helps people to better integrate the more complex concepts that may be presented later. Because people learn best when a variety of different educational modalities are used, it can be very helpful to include visual aids when describing that genes are special sequences of DNA that contain instructions that tell the body how it should grow, develop, and function, and that the gene product genetic variations or changes in DNA, and variations in genes can cause changes in how the body grows, develops, functions, and responds to the environment. Helping people to understand the relationship between genetic variations and the body as a whole can be helpful in establishing the foundations of your explanation about how genes and genetic variations affect the brain. It is important to point out that we all have lots of genetic variations, that they can happen randomly, and that having them is normal. Usually, these genetic variations do not have a noticeable impact on how our bodies grow, develop, and function, but occasionally a genetic variation does cause an increased or reduced chance of developing a particular illness or group of illnesses. You might say, some genes instruct the body in how to make chemicals that are important in the brain. If genetic variations occur in these genes, the result may be too much or too little of important brain chemicals, or brain chemicals that don't work adequately, any of which can cause a chemical imbalance in the brain. One of the explanations for the origins of psychiatric illness with which people tend to become quite familiar is that these illnesses are caused by a chemical imbalance in the brain. Relating your explanation of the cause of psychiatric illness to the existing ideas people may have helps engage people in discussion and to accept and retain new knowledge. You might say, genetic variations do not usually cause psychiatric illness, but they can make a person more vulnerable to one. The genes that can make a person more vulnerable to psychiatric, some clients might prefer the term risk or vulnerability, whereas others prefer susceptibility or predisposition to psychiatric illness. Still others respond best to the idea of having a genetically based sensitivity to their environment that makes them more likely to develop psychiatric illness. What is inherited is a vulnerability to psychiatric illness, rather than the psychiatric illness itself. This is another key concept that directly relates to the previous point. Although the distinction here is subtle, it is very important. Psychiatric illnesses arise as a result of the combined effects of several different genetic variations acting together with and in response to the environment. So no one factor itself causes the illness. It can sometimes be helpful to compare the differences between vulnerability arising from risk genes to autosomal dominant or recessive disorders that are caused by a single genetic variation. The greater the genetic risk, the more vulnerable a person is to developing psychiatric illness. It is likely that everyone has some vulnerability to a psychiatric illness. Some people have large amount of genetic risk for mental illness, and others have a small amount of genetic risk. Most people have a moderate vulnerability to psychiatric illness. Now let's talk about some issues that clients and family members might raise about genetics and psychiatric illness. How do you know that genes are important in the development of psychiatric illness? We know that psychiatric illnesses tend to cluster in families. This means that relatives of someone with a psychiatric illness are more likely than relatives of someone who is unaffected to develop a psychiatric illness. On its own, this is not enough to tell us that genes are important in psychiatric illness, because it could be that the family members share an environment that makes them more likely to develop psychiatric illness. But studying twins can help us work out whether genes are important in the development of an illness. There are two different kinds of twins, identical and non-identical. You may have heard them in the terms fraternal when referring to non-identical twins. Identical twins share 100% of their genes, and non-identical twins share 50% of their genes. We can use this difference between identical and non-identical twins to see if genes are important in causing an illness. If genes play an important role in the emergence of a particular illness, we would expect that if one twin has an illness, the other twin will be affected more often if they were identical than if they were fraternal. This is exactly what we see for many psychiatric illnesses, which tells us that genes are important. What if you get the question, how do genetic variations happen? How can you respond? You could respond, genetic variations happen naturally for many different reasons, and each of us has different types of variation in our genes. Because genes are passed through a family from parent to child, genetic variations can be passed on in the same way. How about this one? Everyone says I look the same as my sibling who has a psychiatric illness. Am I more likely to develop a psychiatric illness than our other sibling who looks nothing like him? Actually, the answer is no. The genes that control our appearance are unlikely to be the same ones that contribute to risk for psychiatric illness. We each have 20 to 25,000 genes, and just because you share the similarities in genes that contribute to your appearance, it does not mean that you will share the same risks for genes for psychiatric illness. Can I have genetic testing to tell me whether I will develop psychiatric illness? Studies have found evidence for considerable interest among affected individuals and their family members in the prospect of genetic testing for psychiatric illness. Any genetic testing that may become available for psychiatric illnesses will only be able to provide information about the probability of a person developing a disorder rather than a definitive categorical answer. Interest in genetic testing has been found to be greater when categorical answers, for example, yes or no, are provided than when only probabilistic answers, for example, you have a 30% chance, are given. Experience with other disorders, including Huntington's disease and hereditary breast and ovarian cancer syndromes, suggests that interest in genetic testing for a condition is usually greater when the availability remains hypothetical than when the test actually becomes clinically available. Nevertheless, the important message for people who ask this question is that genetic testing for psychiatric illness is unlikely to ever predict exactly who and who will not develop psychiatric illness, because the environment contributes to the risk too. At the moment, we do not know all of the different risk genes for psychiatric illness, and we do not yet understand how the different risk genes might interact with each other. At this point, genetic testing is not very helpful in predicting who and who will not develop mental illness. Is the risk genes for psychiatric illness that make me or my family members so creative? It is certainly possible that the genes that make a person more vulnerable to developing psychiatric illness may have more positive effects too, like making people more creative. At this point, we cannot definitively answer this question. Another question that you might hear, no one else in my family has this illness, so are genes not important in the psychiatric illness of my family? Here's one way to give family members. This does not mean that genes are not important in the development of these illnesses in these families. In fact, this is what we would expect to see, given the risks genes do not cause psychiatric illness. Remember, just because a person has a large number of risk genes does not mean that he or she will develop a psychiatric illness. Without the contributing environmental factors, he or she may remain healthy. So although other family members may be unaffected, they may still have a large number of risk genes. Alternatively, it is possible that both the parents of an individual who develop the psychiatric illness have an average amount of risk genes, and that neither of them ever developed the illness themselves, but that their child, who went on to develop psychiatric illness, inherited all the risk genes from both parents, and so is much more vulnerable to developing the psychiatric illness than was either parent. Is there any way I could prevent passing on risk genes to my child? It can be very important for people to understand that we have no control over the genes that we pass on to our children. Most parents would like to be able to pass on what they consider to be their good characteristics to their children without passing on the kids. This lack of control can be difficult for some people to accept, but it can be a great relief in other circumstances. For example, when talking with parents of a child who has developed psychiatric illness. To reduce the number of risk genes that we have for psychiatric illness, but it is important to remember that genetic vulnerability alone does not usually cause psychiatric illness. Although we cannot change our genetic vulnerability, there may be things that we can do to reduce our risk for psychiatric illness. I'm going to talk about this topic later. I heard that schizophrenia and bipolar disorder are about 80% genetic. Is that true? One of the most widely misinterpreted concepts related to genetics and mental illness is heritability. Heritability is a statistical measure of the degree to which a particular condition is attributable to genetics at the level of the population. It is true that schizophrenia and bipolar disorder have both been estimated to have heritabilities in the region of 80%, but it's more important to remember that this is a population-based concept. For example, although on average, at the population level, couples may have 2.4 children, no individual couple is actually going to have 2.4 children. In much the same way, although these conditions have population-based heritabilities of about 80%, it is not correct then to assume that an individual with schizophrenia or bipolar disorder has that condition as a result of an 80% contribution of genes and a 20% contribution of the environment. At the level of individuals, there is likely to be a huge range in the relative contribution to the genetics to the development of the disorder. The process of providing counseling about the causes of psychiatric illness and the risk for family members to become affected should begin with contracting and learning about your client's perceptions and objectives. In addition to establishing session boundaries, for example, time, contracting makes explicit both your goals for the session and those of your client. The contracting process should include an investigation of your client's perceptions of his or her illness and the risk of family members becoming affected. We're now going to talk about information about how to include your own goals for counseling in this context and those of your client. The importance of exploring your client or family member's existing perceptions of cause, burden, illness control, and amount of family risk, and the importance of acknowledging our inability to provide certain information about personal cause and risk for family members to become affected. Now let's discuss contracting. The very first thing we're going to do when we begin contracting is to elucidate our client's goals and share our own. Remember that contracting promotes your client's autonomy by increasing their control over the process and the information discussed. The needs of your client will differ based upon their personal and family histories, their age. Some will primarily seek psychological counseling around risk in the family. Some will be most interested in discussion of early symptom identification and early intervention, and some will request a risk assessment. Contracting allows individualization of timing and focus of the counseling process. After you've determined the client's primary goals for the interaction, the contracting process also gives you a forum to explain other reasonable explanations for the encounter or to share what you think might be most important or relevant for each client. Now let's talk about assessing perceptions of illness and family risk. Illness perceptions play an important role of shaping an individual's response to information about the cause of psychiatric illness and the risk of family members becoming affected. Identifying and understanding your client's illness perceptions allows you to provide more targeted and effective counseling. In addition, there is evidence that attempting to provide new information about the causes of psychiatric illness without first exploring existing perceptions is likely to be a relatively unsuccessful strategy. Illness perceptions may also play a role in treatment adherence. Some of the themes that have been shown to play a role in appraisal of risk illness include the burden. Clients who receive more burden as a result of the illness typically not only perceive risk to be higher, but also tend to have less tolerance for risk. Control. Clients who perceive themselves as having less control over the onset of the illness in themselves or their children and or their illness symptoms and course typically not only perceive risk to be higher, but also tend to have less tolerance for risk. Preconceptions about the information about the combination of genetic and environmental risk. And then finally, preconceptions about risk for family members. Clients' preconceptions of risk before the genetic counseling session affect the way in which they receive information about genetic risk that is provided during the risk counseling. Thus, it is important to their own illness, including degree of burden, locus of control, their perceptions and preconceptions of the degree of the illness burden, and the locus of control experienced by the others, including affected relatives as well as at-risk relatives, how likely they think it is going to happen again in the family, and the rationale for their concern or lack of concern about family risk. Gathering this background information will help you tailor your messages to each individual client and anticipate areas of concern and reactions to information about the causes of illness and risk. So how can you get started with this discussion of contracting? One thing you might say is we have an hour, or insert the correct time span there, today to spend talking about some of the issues you're interested in. For example, what causes psychiatric illness and how much risk there is for other family members to be affected. As with many other clinical interventions, identifying up front how much time you can spend with your client discussing these issues on this particular occasion defines the boundaries of a session as an important part of the contracting process. Let the client know if the discussion can continue at a later date. You told me that you were interested in learning more about what caused your illness. Do you have specific questions that you would like to make sure we cover? Or what motivated you to ask me about the causes of your illness? This beginning, or these beginnings, can open a dialogue about your client's goals for the session, which are important to establish at the outset. Moving on from there, you might say, what do you feel was responsible for causing your family's member's psychiatric illness? In addition to providing insight into an individual's existing level of knowledge and allowing you to tailor the level of your discussion to him or her, identify accuracies that you would like to seek to reinforce and affirm, as well as any misconceptions that may need correction. Indeed, if the individual's existing ideas about the cause for psychiatric illness are not addressed in the context of the new explanation that you provide, it is more difficult and less appealing for your client to integrate the new knowledge. If the client responds to this question with, "I don't know," consider reframing the question this way. Tell me about what was going on for you and your family member just around the time you first got sick. Or has anyone in the family ever experienced similar problems? These additional questions can be really helpful in providing an opening for some people that makes it somehow their fault. Many clients and family members think that there was something that they did or did not do that actually caused the psychiatric illness. If you suspect this is the case, opening a dialogue with a statement to this effect can be very helpful. For people who have heard or wondered about the role of family dynamics, personal weakness, or character flaws in using that cause psychiatric illness, it can be helpful to mention that there has been a lot of damaging misinformation over the years and that you will help them sort out the likely causes from the unlikely ones. Sometimes people don't want to take their psychiatric medications because they think that their illness is caused by respond to this one. When affected individuals or their family members do not ask, sometimes this attribution for the cause of illness has observable manifestations or consequences. For affected individuals whose attributions lead them to feel doomed, whether because the illness was fated to occur or so strongly genetic that there was no escape, treatment may be seen futile and the individuals may or may not take their prescribed medication. When they're not taking their prescribed medication, in other words, in situations like this, where the behavioral consequence of an illness attribution is exposed, there's potential for significant shifts in the attitudes following the education gained in the session. In other words, you might act. Before we started talking about the causes of psychiatric illness, you told me that you were interested in talking about the risks that family members will be affected. Now that we've talked about causes, I thought I would check in and see if you were still interested in hearing about the risks. Checking in like this can be an excellent way to see how the preceding discussion about the causes of psychiatric illness was received, in addition to ensuring that you are positioning yourself to meet your client's needs regarding risk assessment. Importantly, this kind of simply hearing that psychiatric illnesses are not entirely attributable to genes is sufficient for an individual to realize that his or her child or other relative would not be affected, which may be sufficient information for the client's needs. Although I will be giving you information about the chances for your children to be affected, you should know, should you decide to have any, I won't tell you what to do. For example, I won't make any statements about whether or not you should have children. I want to give you the best information that I can help you make decisions that are right for you. It can be very helpful to state at the outset of the discussion that you aim to promote decision-making and autonomy and will not instruct your client about which choice to make on such important topics. Depending on your clinical role, clients might expect you to be more directive, especially about decision-making or life planning. It can be useful to establish It can be really hard to worry about the chances for yourself or other family members or your children to develop a psychiatric illness. Adolescent and adult children and siblings of affected individuals often silently harbor worries that they may develop the same illness as their family member. Parents of young people who have been diagnosed with a psychiatric illness are often worried about the risk to their other children. Affected grandparents and grandparents with an affected child are often concerned about the risk to their grandchildren. And finally, diagnosed individuals who have young children are often deeply worried that their children will develop the same illness sometime in the future. These fears are frequently left unarticulated. In fact, in my experience, when such fears are articulated, many clients in this way combat the sense of isolation that people with these worries often face. Do you have a sense of the chance that your child, other relative, will develop its existing attributions of his or her illness prior to your providing new information about the causes of psychiatric illness? In the same way, it is important to inquire about your client's existing perception of risk prior to. It can be really helpful to base discussions about the cause of psychiatric illness or risk to family members on a detailed psychiatric family history. I can use this history to see, hey, if we start by drawing out your family history. In the interest of helping the client understand the content of the session, it can be helpful to obtain permission to ask about potentially sensitive family history when these discussions are initiated and briefly explain the purpose of the family history. Having an established therapeutic relationship with the client may facilitate your ability to obtain a more complete family history while maintaining a high level of client comfort. Now let's move to some client issues that may arise during the contracting stage. Sometimes a client might about the cause of my condition. You're going to want to respond with the following information. Most clients understand that we do not have all the answers about causes of and risk for psychiatric illness. These types of questions allow you to acknowledge the limitations of our understanding about the specific factors that contribute to psychiatric illness. It also provides an opportunity to educate more interested clients about why the answers have been so difficult for researchers to achieve. The discussion allows the groundwork for how clients may use the information provided during etiology and risk counseling. Am I right to be this worried? Can you reassure me? Clients may explicitly or implicitly communicate that they hope to be told that the risk to relatives is less than they had anticipated. At the same time, many clients, especially parents, say that they would appreciate having their concerns confirmed. This can be an empowering experience for parents who know there is a risk in their family, but who have never had their knowledge and experience validated by a professional. Can you tell me why I have this illness? Can you tell me exactly what the chance is that this will happen in my family again? Clients may wish for a definitive, clear-cut answer about causes of illness and degree of risk to family members. They may be disappointed to hear that while we can provide up-to-date information about the causes of psychiatric illness in general terms, it is currently not possible to conclusively identify each one of the specific factors that contributes to the development of illness for any one individual. However, some clients are understanding about scientific limitations and appreciate being the most up-to-date, client-friendly information. I don't want to hear about risk to children because I already know that they are certainly going to have the same illness as me, so I'm not having any children. This kind of statement presents a more difficult but not uncommon situation. The client obviously has a right to know, and indeed is telling you that he or she wishes not to know, but it is his or her decision is based on the assumption that is incorrect. In this situation, exploring with the client the basis for this assumption and its origins, simply asking whether the client has an interest in discussing the causes of an illness will allow conclusions to be revised based on this new information. Now let me give you 2 case examples. Shelley remembered hearing voices when she was 10 years old, but did not know that this was an unusual experience until years later. Shelley was diagnosed with schizophrenia in her teens. Now in her 30s, she came with her husband to see me for genetic counseling. She told the genetic counselor that she wanted to understand that the cause of her illness because she could never comprehend why it was that she got sick and her sister didn't, despite the fact that they had the same experiences. When I asked if there were specific questions she would like to have addressed, Shelley told me about her sister, who was 2 years younger. As children, they had moved homes multiple times, each time changing schools, and then when Shelley was 9, their parents divorced. Shelley found these experiences to be hugely stressful and emotionally difficult, and she attributed her illness entirely to them. However, her sister had the same experiences and had not developed schizophrenia, so Shelley was confused and wanted the genetic counselor to explain why this might be. The genetic counselor offered to try to do this, and then asked if Shelley and her husband were considering a family, and whether she would also be interested in hearing about the chances that her children would be affected. Despite her belief that her illness was the result of her difficult childhood experiences, Shelley had not decided to have children because she felt and they would be certain to develop schizophrenia. She also felt that her illness had profoundly negative personal impact and was very burdensome to the entire family. She didn't want to bring someone else into the world who was bound to suffer and make others suffer. Her husband, on the other hand, was interested in hearing about the risk and asked Shelley if she would mind if the genetic counselor told them more about it. Shelley's attitude was that it couldn't be any worse news than what she had already thought, so she shrugged and agreed to talk about that too, just to accommodate her husband's wishes. Shelley was initially reluctant to discuss family history. She couldn't imagine what use it could be, she told me. No one in the family had schizophrenia. She and her husband agreed that the genetic counselor could draw out their psychiatric family histories after explaining that it would help the genetic counselor talk about the known etiology of schizophrenia and about the risk for family members to become affected. Shelley became increasingly interested in the discussion. Based on the process that was just outlined, a mutually agreed upon agenda for the session was developed, and we found some good ideas about Shelley's perceptions of her illness. Her sense of control over her illness was uncertain, but that she had more definite perceptions of burden and risk for family members to be affected. She gave insight into her understanding of what had caused her illness, and we were able to refer her understandings as I offered explanations about etiology. The psychiatric family history is the cornerstone of activities related to education and counseling about causes of psychiatric illness and risk for family members to be affected. I'm now going to provide some tools and strategies for collecting a useful psychiatric family history. I'm going to begin by outlining the benefits of gathering a targeted psychiatric family history and describe some different options for formats that can be used to document the family history. In addition, I'm going to review the accuracy of the self-reported family history and the to obtaining a psychiatric family history. Risk assessment for psychiatric illness requires a complete family history as possible, and personalized discussion of the causes of psychiatric illness is facilitated by a greater quantity of information. It is important to initiate the discussion of causes and degree of risk in a calm, comfortable location that is free of distractions. Similar to other therapeutic interactions, it is of course important to maintain confidentiality when discussing these topics. It can be easy to lose sight of the private and sensitive nature of this family history data. Some clients are eager and prepared to disclose and discuss their family medical history at the beginning of the risk counseling session. For those clients, after a brief discussion about the goals and outcome of the family history, I tend to initiate the history-taking process near the beginning of the session. I use the family history process as a way to learn more about the client and always incorporate education and psychosocial counseling into this phase. Clients may be less willing to share information, so sometimes it's best to begin the session with some education about causes of psychiatric illness and a careful discussion of how the family history may inform a more targeted discussion about those. From there, I may undertake some psychosocial counseling about the experience of living with a psychiatric illness in the family. Generally, the most relevant family history information comes out as part of this discussion. I then build upon the basic information to flesh out the family history after the client feels more comfortable. Collecting information about at least 3 generations of biological relations is standard, but it's beneficial to collect as much information as possible. The fact that both genetics and environment are involved in contributing to the development of psychiatric illness means that the disorders cluster in families instead of segregating in a predictable fashion. So, unaffected relatives can and will pass susceptibility genes to children, which makes it important to obtain information about close relatives of unaffected family members as well as those of affected family members. When collecting the family history data, I want you to consider taking the following. Begin with the client and his or her first-degree relatives, and then work up to the next generation and out to more distant. Ask about the age of onset, illness course, and treatment for anyone in the family with a psychiatric illness. These factors can inform the personalized discussion of the causes of psychiatric illness and the risk assessment. Don't just ask about psychiatric diagnoses. Instead, ask about mental health in general. For example, whether people have taken medications for their mood or for nerves. Sometimes a client does not know if a particular relative has ever received a psychiatric diagnosis, but then describes the relative as having strange beliefs or behavior. For example, he was such a loner, or the things that she believed were often really bizarre. In such a case, it can be useful to check about developmental history. Has the individual achieved normal milestones for age, such as independent living and employment, and social relationships? For example, does the individual have any close relationships? If your clinical experience leads you to suspect a symptomatic relative that the person, 1, has not been diagnosed because he or she has not sought treatment, 2, did not meet full criteria for a psychiatric disorder but has symptoms related to the disorder that's running in the family, or 3, has been diagnosed but has not shared this with anyone else in the family. Ideally, such relatives should undergo a psychiatric evaluation, although it is most likely that they will decline. Ask about suicide and suicide attempts, which are, of course, very common in each of the major psychiatric illnesses, and conversely, psychiatric illnesses are very common in those who commit suicide. It is especially important to discuss suicide risk with clients who have a strong family history of suicide. There seems to be independent risk factors for suicide that make it more likely that a person will attempt or commit suicide if he or she has a family history of suicide. Ask about substance abuse. Substance misuse is a serious psychiatric issue in its own right and should be documented in the family history and addressed with all clients. Keep in mind that the high rates of comorbidity between major psychiatric illnesses and the substance misuse are Remember to gather information of a specific concern to your client as a psychiatric illness that typically has a childhood onset. The rationale for collecting typically is adult onset, however. For example, if the concern is bipolar disorder, which typically has an onset in late adolescence or the early 20s, collecting information about the presence or absence of attention deficit hyperactivity disorder, ADHD, on younger family members can be important, as it may indicate increased familial risk. Remember to let the disorder as they become more distantly related. If clients do have information about more distant relatives, be aware that diagnoses may be less reliable and that histories of the institutionalizations and nervous breakdowns must be interpreted in light of the social context at the time. Remember also to ask about any birth defects, mental retardation, or learning disabilities and any unusual medical conditions that the family cannot explain. Now let's talk about some barriers to gathering a psychiatric family history. The first is the stigma related to psychiatric illness might make an individual reluctant or unwilling to share a personal or family history. Families tend to hide a history of psychiatric illness, and the client may not be aware of affected individuals or may not know their current diagnosis. Clients or family members might describe their illness with a more socially accepted diagnosis that is perceived as less severe. Highly symptomatic clients may find it difficult or be unwilling to provide family history information. And because of their illness, individuals with psychiatric diagnoses may be more likely to be alienated from their families and have less information. So what do you say when you begin to gather the family history? One example might be, I'm going to ask you questions about your family members' mental health, in general how they are doing, and whether they have any serious medical issues or birth defects with which they were born. This family history information will help me to explain to you what causes psychiatric illness in your family and what the chances are that other people in your family might develop psychiatric illness too. Explaining the purpose and process up front is a good idea. It is also usually very well received if you show the developing family history document to your client during the process. It is also usually appreciated if you provide your client takes varies depending on how large the client's family is and how much information she has about the family members, but it usually doesn't take more than 10 to 15 minutes. Then you might move on with something like, don't worry if you don't know about the mental health status of all your family members. I understand that many families tend not to talk about psychiatric illness. Would you feel more comfortable to approach your family history to find out more? Clients can sometimes get anxious if they feel you are asking a lot of questions that they cannot answer, particularly if like failure. In such situations, you can use statements like these above to attempt to alleviate that worry. People approaching family members for additional information, or if the individual cannot get more information because the family relatives do not know the client's diagnosis, reassure such clients with statements like, no problem, we can work with whatever you know. You might say something like, this family history is generated from information you provided, but I wonder what your perspective is on it now that you're seeing it laid out like this. How are you feeling about it? Even though the family history is generated using information provided by the client, sometimes seeing it clearly laid out can be quite startling. This may be particularly true when individuals who have mental illnesses or they may not have done so in a systematic way. With the goal of identifying areas of genetic overlap between diagnoses in the family, seeing the graphic presentation of the family history may bring the client a teachable moment that cannot be missed, but you should pay attention to the client's affect through this process. Now let's talk about some issues that clients and their families may raise during the taking of the family history. How do you respond with, why do you want to know about how old everyone was when they first got sick? You can respond to such a question by acknowledging that it may seem odd or irrelevant to ask these sorts of questions, but that this kind of information can be useful in approximating what might have caused the illness in the family member, and in helping you to provide them with the best information that you can about risk and relatives that might be similarly affected. How do you respond with, no one else in my family had psychosis. Well, I guess my brother did, but he was taking on drugs when it happened, so it was not related to my diagnosis. This kind of statement is actually very helpful because it provides some insight into how this client perceives the brother's illness experience as arising from a very different set of contributing factors from his own. It would be important for you to ask some follow-up questions such as about the type of drug the brother used at the time of the psychosis and for how long the psychosis lasted. You should note the brother's history in the family history data and explain to the client that it's hard to say for certain whether the history is related to the client's own diagnosis. Can I see what you're writing about my family history? Clients are often very interested in seeing the compiled information about their family histories, especially if you take the family history in a pedigree format. Clients may ask to have copies of the information to bring home. Encourage this curiosity and show clients the developing pedigree and explain what the symbols mean. Developing the family history collaboratively is an excellent rapport building tool and helps engage the clients fully in this process. Now let's talk about a case example. Joanne was a 22-year-old woman with a diagnosis of unipolar major depression. She had recently become engaged, and her engagement caused her to think more about the cause and likely trajectory of her illness, as well as her potential to have children with a mood disorder. After some discussion of the goals of the genetic counseling, the genetic counselor described the importance of the family history. Joanne explained to me that she thought that mood disorders were fairly common in her family, but that her relatives were extremely unwilling to discuss mental illness. She therefore asked her mother whether she could share any information she knew about mood illness in the family. Joanne and her mother engaged in their first frank discussion about Joanne's illness and the history of the family, and Joanne's mother admitted that she had been treated for years for depression herself. This was the first time that Joanne learned of her mother's mood disorder. Joanne's mother also shared the limited information she obtained through rumors she'd heard about other relatives' illnesses. Joanne perceived this information sharing as a major gift that her mother had given to her. Joanne and the counselor spent some time discussing what it was like living in a family with so much secrecy and what it was like to learn about her mother's illnesses. The counselor used the available family history information and Joanne's medical history to provide the best assessment of etiology and risk possible. Joanne asked the counselor if she could summarize the information in a letter because she felt strongly about sharing it with her relatives in an effort to reduce the secrecy and stigma in her family. Now let me present some strategies for understanding and discussing the environmental contributions to psychiatric illness and their complicated interactions with each other as well as with other genetic factors. I'm going to provide some tools and suggestions for presenting the concept that genes and environment work together to contribute to psychiatric illness in this section. Nature versus nurture. Oh, for many years, the causes of psychiatric illnesses were the subjects of heated debate among interested healthcare professionals and academics. In general, those with biomedical training in mental health tended to favor explanations for the origins of psychiatric illness that emphasize biological mechanisms, otherwise known as the medical model, whereas those with training in the humanities tended to favor explanations that emphasized experiences and environment, the social model. The popular press fueled this debate, framing news stories about the origins of psychiatric illness as questions about whether these illnesses are caused by nature or nurture. In recent years, the debate around the subject has subsided considerably because across professionals with disparate training backgrounds, a general consensus of opinion has been reached about the origins of psychiatric illness. There is now widespread support for the idea that psychiatric illnesses are typically caused by neither nature nor nurture alone, but rather, like other common disorders such as cancer, heart disease, etc., they arise as the combined influence of both biological nature and environmental contributions, nurture. There are numerous environmental factors that have been implicated in the development of psychiatric illness. I'm going to describe many of these now. Stress. Although a stressful event of some kind often precedes an episode of psychiatric illness and is often described by families as the trigger for illness onset, it is the difficult pickup of traumatic experiences such as a car crash and financial or work-related crises can certainly be important contributors in the development of psychiatric illness. Those illness-triggering events are not always so recognizable to the observer. Less obvious events that the individual appraises to be stressful can also be important at an illness onset. Stress seems to directly influence how genes instruct the body about how to grow and develop and function, especially in individuals with certain genotypes. Stress has been shown to affect the brain by influencing genes, which in turn cause variation in the amount of certain brain chemicals. It is thought that stress could influence risk for psychiatric illness through this mechanism. Drug use. Certain drugs appear to contribute to the development of some kinds of psychiatric illness, particularly the psychotic disorders in individuals who are genetically vulnerable. In particular, crystal methamphetamine, or crystal meth, can trigger psychosis that persists even after the drug is cleared from the system, and so is of particular concern in terms of use by already vulnerable individuals. When used in adolescence, cannabis, otherwise known as marijuana, also seems to increase the risk in genetic vulnerability for individuals for psychosis in later life. People with existing psychiatric illness who use marijuana have poorer outcome than those who do not use marijuana. Drugs such as marijuana and methamphetamine may increase the risk for psychiatric illness by directly influencing the quantities of certain brain chemicals. Paternal age. One of the most well-replicated findings of contributors to psychiatric disorders is that paternal age at the time of conception is related to relative risk for offspring developing schizophrenia. Similar findings are emerging for bipolar disorder and autism. The highest risk to offspring seem to be seen for those fathers who were 55 or older at the time of conception. When fathers are more than 55 years old at the time of conception, the risk seems to double, to about a 2% risk, as compared to the risk for offsprings of fathers at the youngest age category, about a 1% risk. Although the mechanism by which paternal age affects offspring risk for psychiatric illness is not known with certainty, we do know that the chance for spontaneous genetic variation in sperm increases as man gets older. Thus, it is possible that paternal age influences offspring risk for psychiatric illness because the chance that variations will occur in associated risk genes also increases. Obstetric complications. Complications of both pregnancy, for example, preeclampsia, diabetes, rhesus incompatibility, and delivery, such as lack of oxygen during delivery and emergency cesarean section, are associated with increased risk for a newborn to develop schizophrenia later in life. Individuals whose perinatal period was complicated are 2 to 4 times more likely to develop schizophrenia than those whose perinatal period was not complicated. The evidence regarding risk for mood disorders associated with obstetric complications is clear, but there is some evidence that children who were born prematurely may have somewhat increased risk. The exact mechanisms by which obstetric complications increase risk for psychiatric illness are not well understood. Season of birth. There seems to be a small but significant increase in the rate of schizophrenia among individuals born in the winter or spring as compared to those who are born in the summer or fall. The data for the northern hemisphere are stronger than those in the southern hemisphere. Some evidence also seems to support similar trends for mood disorders. The mechanisms behind this association are not well understood, but some researchers have hypothesized that increased frequency of specific types or infection or low prenatal levels of vitamin D in the winter-spring months as compared to the summer-fall months may contribute to the risk. Head injury. For individuals who are genetically vulnerable, sustaining a head injury seems to increase risk for schizophrenia, by which head injuries contribute to risk for psychiatric illness is not well understood. Urban upbringing. There is good evidence that urban birth or urban upbringing is associated with an increased risk for schizophrenia. Although there are many theories regarding why this might be, including increased frequency of infection and higher levels of pollution and stress in urban areas, there is no conclusive data. Only scant research has been conducted with the aim of investigating the relationship between urban upbringing and mood disorders. Immigration. There is evidence that both first and second generation immigrants have increased risk for schizophrenia. There are few data and inconclusive evidence regarding the relationship between mood disorders and immigration. With respect to schizophrenia, researchers have effectively controlled for many potential confounds in studies investigating this issue, and one of the most interesting theories about the mechanism by which the risk is increased relates to the effects of social adversity often faced by immigrants. Maternal famine. Exposure to famine during pregnancy has been shown to increase the risk for the developing offspring to develop psychiatric illness, including major mood disorders or schizophrenia later in life. The reduced availability of vital nutrients is thought to be responsible for this increased risk. And then finally, infection. There's been considerable interest in the question of whether infection with viral or other agents, such as parasites such as toxoplasmosis, can contribute to risk for psychiatric illness. The results generated to date have not been shown that infection is the major contributor to risk. In summary, there's sufficient heterogeneity of illness causes, features, and outcomes with diagnostic categories. There's no single factor that is the risk factor for any psychiatric illness. Many, if not all, of the factors for psychiatric illness increase risk for more than one disorder, and disorder onset usually requires an intersection of interacting genetic and environmental risk factors. There are differences among individuals in terms of the specific genetic and environmental risk factors that contribute to the development of the illness and in the relative contributions of genes and environment. Clients cannot achieve a holistic understanding of the causes of their illness without learning about the roles of both genetic and environmental factors. Although, in my experience, clients' own intuitive attributions of the cause tend to include both genes and biology, as well as experiences, most express some uncertainty and confusion. Many clients have identified attributes in other relatives that make them more likely to become ill, including personality traits and environmental exposures. Conceptually, though, it can be quite difficult to get a solid grasp on exactly how genes and the environment contribute together to the development of these illnesses. In my interactions with affected individuals and their families, I have used different techniques to convey the key concept of this issue. Now I'm going to present two complementary tools that I have found to be most helpful in this regard: the JAR model and the family history. When using these tools to discuss the roles of gene and environment, I try to incorporate clients' perceptions of environmental factors into the discussion. Earlier, I suggested that you consider asking questions such as, can you tell me a little about what was going on for you about the time that you first developed this illness? This type of question can be a very effective way to identify environmental factors that the client perceives to have been significant in the onset of his or her illness, and can be useful to refer back to previous experiences that clearly highlight that the client highlights while using these tools. I like to use the JAR model. Many people grasp concepts more easily when they're represented visually. I find that this simple visual analogy can be helpful in representing this difficult but important concept that genes and environment both contribute to psychiatric illness. The general concept in this model is that we all have a mental illness jar, representing an overall risk for mental illness, and that the jar has to be filled to the top in order for the person to experience an active episode of mental illness. Two different kinds of factors go into the jar: genetic and environmental. We represent the two kinds of factors with different colored and shaped objects. You can use a visual representation of the jar or a physical jar in your demonstrations. The jar analogy can present both vulnerability and protective factors very concretely. While the amount of genetic vulnerability in the jar remains constant, environmental vulnerability factors can be added to the jar as well as removed. A third element in the JAR model is the protective factor. We use examples of medication and good social support network as protective factors that restrict access of environmental vulnerability factors to the jar. While it is a simple analogy, it accurately captures the core issues in such a way that it can be used to help those who may have cognitive difficulties get a better understanding of the factors that contribute to illness, but it can also be used as a basis for more in-depth discussions for those that are more sophisticated understanding. The psychiatric family history can be very useful in illustrating the complex contributions of factors to the development of psychiatric illness in a very personal and therefore powerful way. Graphic pedigree representations of the family history are by far the most effective way to use the family history as a teaching tool. In the family histories of the large proportion of affected individuals who have no affected family members, you can describe how multiple risk factors had to come together to produce the illness. No one else in the family had sufficient risk factors to cause illness, but it is likely that there are genetic vulnerability factors that the client inherited from both sides of the family. If relevant to the client's needs, you can use the pedigree to illustrate the discussion about how an affected individual can pass genetic vulnerability to children. In addition, gathering information about life events that occurred around the time of illness onset for affected individuals can be a useful way to illustrate the role of environmental factors. With an affected individual who has family members who had other psychiatric diagnoses, symptoms, or questionable histories, you can use the pedigree to illustrate the clustering that occurs in families and the shared genetic vulnerability to mental illness in biological relatives. In less common situations where the client has a complicated multi-generational pedigree, or one where there are many affected individuals in the same generation, you can use the pedigree to show how genetic vulnerability factors have been transmitted from generation to generation, or how risk factors have clustered into one generation and the genetic vulnerability that can to subsequent generations. In this example, there is a sort of a family history you might commonly encounter in the format of a simplified, in other words, I've included no ages or onset or treatment data here in this pedigree. You may find that using the family history and the jar model together creates the most individually relevant and easily grasped explanation. So how can you use the jar model and the family history to help you explain concepts to families? Here's one thing that you might say. It can be difficult to understand how genes and environment work together to cause the development of mental illness. So we can use this jar as a visual analogy to help you understand how mental illness arises. Every one of us has a mental illness jar. In order to have an active episode of mental illness, the jar has to be full. There are two different types of risk factors that can fill up our jars. The balls represent genetic risk factors, and the triangles represent environmental risk factors. Let's use this jar analogy to talk about how someone might develop mental illness over time. By using this explanation, you can explain that the jar on the left represents how much genetic variability or risk this individual inherited from his mom and his dad, and the jar is not full, so this individual is not experiencing an active episode of illness. Additional environmental factors may add up over time. Explain to your client that each of these experiences would perhaps add a few more environmental triangles to his jar, and at some point the jar becomes full and the person then experiences an episode of illness. Note that it can be very effective to use plausible environmental vulnerability factors that the individual mentioned having experienced as examples of adding risk to the jar. Take care, however, to debunk any incorrect beliefs about contributing factors. For example, curses or bad character that your client may have. The amount of genetic vulnerability in the jar stays the same over time. Try to help clients understand that although the amount of environmental vulnerability in our jars can change over time, the same amount of genetic vulnerability with which we are born always stays the same. This jar model can be used to illustrate how different individuals will have different amounts of genetic vulnerability to mental illness. An alternate way to think about it is that the person at the top is less sensitive to his or her environment, and the person at the bottom is more sensitive to his or her environment. Different people will have different amounts of genetic vulnerability in their jars. Again, based on the previous model, you could explain to your client that a person represented in the top row of that figure has less genetic vulnerability in her jar and is less likely to develop mental illness. It is less likely that her jar will fill to the top because there's more space in it. But the person represented in the bottom row is more likely to develop mental illness. There is only a small space at the top of his jar, so it would take very little for it to fill up. If both people go on to develop a mental illness, the top person is likely to be older than the individual at the bottom when he or she first experiences an active episode of mental illness because it takes longer for her jar to fill up. You could then personalize the discussion by asking your client's age at symptom onset and describing what this might indicate about his or her genetic vulnerability. This diagram can be used to help individuals with mental illness feel less biologically different. It depicts the idea that we all have the same amount of genetic vulnerability to mental illness and that within a population the amount of genetic vulnerability is normally distributed. As seen on the previous slide, we could say something like this to explain this model. We all have some amount of genetic vulnerability to a range of disorders, including psychiatric disorders and other types of disorders like heart disease and diabetes. Some of us will have very little genetic vulnerability, others of us will have a lot, but most of us likely have some intermediate amount. You can use this discussion to directly challenge the idea that those with mental illness must be in some way biologically different or other than those who are not affected. You could use the analogy of height to help clients understand the idea of population variability to genetic vulnerability. For example, you could talk about how a relatively small number of people will be shorter than 5'2", and similarly, relatively few will be taller than 6'2". Most people will fall somewhere in between. In the same way, it seems likely that while a few people will either have a very large amount or a very small amount of genetic vulnerability to mental illness, most will have some intermediate amount. You could also explain that it is not just those with mental illness who have genetic vulnerability. We can have a genetic vulnerability to mental illness but never get ill because our experiences never cause the jar to fill to the top. This is an example of that concept. This is an important point that directly challenges any tendencies towards genetic fatalism an individual may have. It seems that what we inherit is a vulnerability to mental illness rather than a vulnerability to such a disorder specifically. This is also another important point. It is quite common to see family histories where there are multiple individuals, each with a different mental illness. Research studies increasingly show that most genetic variants that increase risk seem to increase vulnerability to mental illness in a broadly defined way rather than to a narrowly defined psychiatric diagnosis. Perhaps it is the individual combination of genetic and environmental contributors that determines whether an individual manifests bipolar disorder or schizophrenia, for example. You can tell the affected client that although he or she may have no family members with the exact same diagnosis, the family history of another diagnosis or group of diagnoses makes it very likely that he or she inherited a genetic vulnerability to mental illness in general. Some genes play an important role in how we respond to life events. Genetic variations in those genes may result in less than optimal response patterns in particular circumstances. Be sure to help clients understand that one of the primary roles of genes is to respond to changes in the environment. Certain genetic variants seem to affect how individuals respond to particular environmental stimuli such as drugs or stressful life events. To help the client understand this, you can ask him or her to think of his friends and family members who bounce back easily from serious difficulties, and to think of others who are strongly affected by similar experiences. Genetic differences are part of the reason for this. Experiences, environmental factors do not usually cause psychiatric illness by themselves, but they can make a person more vulnerable to developing mental illness or can trigger an episode. Clients or family members who attribute the development of mental illness entirely to environmental factors may have increased perceptions of guilt or blame. Family members often feel guilty and blame themselves for their relative's mental illness. For example, parents of an affected individual may blame the appearance of the illness on their divorce or the fact that they had to move to a new home. Other families, especially those who have lived with the effects of mental illness for some time, have heard the term or have been exposed to the concept of the schizophrenogenic mother or similar attributions of blame to members of the family. These blaming attributions often have a profoundly negative impact on the family, and learning that mental illness is not usually caused by environmental influences alone can be very helpful. It is also important to stress that many environmental influences are beyond a client's control. For affected individuals who could attribute their illness to life choices such as their own substance abuse, understanding that such environmental factors usually do not cause illness by themselves can also help to alleviate debilitating guilt and self-blame. We also find it useful to talk about how mental illness develops before we talk about how people can recover from an active episode of illness. In this diagram, although the amount of genetic vulnerability in the jar remains the same, some environmental factors are removed from the jar. Also introduce the concept of protective factors at this point. The idea with protective factors is that if they do not function as a lid to the jar, rather they make the opening at the top of the jar narrower, thus harder for environmental factors to get in. Or you can talk about them as making the jar taller, and therefore more environmental factors are needed to fill it up again. We can also use this jar model to explain how people can recover from an active episode of illness. It can be very helpful to understand how the illness arose, but what makes the visual jar model even more compelling is that you can use the same model to demonstrate the mechanism by which recovery occurs, by exerting control over environmental factors and introducing protective factors, such that the symptoms are reduced or disappear. Examples of interventions to reduce risk may include psychiatric care, abstaining from illicit drugs, social support, and good self-care. You can explain how such interventions may be protective, that is, they partially block the opening of the jar so that it is more difficult to add to the overall risk. People who have never experienced an episode of psychiatric illness but who are concerned about their risk because they have an affected family member can also use protective factors. Though they cannot change their level of genetic risk, individuals who have never experienced mental illness can make use of protective factor strategies such as those listed above to reduce their chances of developing symptoms. Now let's talk about some issues that clients may raise when you explain how psychiatric illness happens. But no one else in my family has psychiatric illness. Does this mean that I don't have any genetic vulnerability? This is a really common question to encounter from individuals who have no family history of mental illness. The jar model can be very helpful in addressing this issue. You can explain that the client's parents could have easily had the genetic vulnerability to mental illness, but never developed the illness because their jars never become full. They never had the experiences that would have been necessary to fill the jar to the top. Remind the client that it seems that we all have some genetic vulnerability to mental illness, so even family members who were not affected would likely have had some genetic vulnerability. It could be that both an affected individual's parents each had only a relatively small amount of genetic vulnerability in their jars, but the affected individual inherited all of the genetic vulnerability from both parents, resulting in a significantly greater risk to develop mental illness. How do I know how much genetic vulnerability is in my jar? This is something that most people want to know, and essentially the answer is that at present we cannot know the answer for certain. There are clues that may give some idea about how much genetic vulnerability is in a person's jar, but this is not an exact science. For example, we can expect that someone who has lots of affected family members over several generations is likely to have more genetic vulnerability factors in his or her jar than someone with no affected relatives at all. Similarly, we might imagine that someone who first experienced symptoms of mental illness at an unusually young age might have a more genetic vulnerability than someone who was older when he or she experienced it. These are rough approximations, however, and we cannot at present definitively measure the degree to which an individual is genetically vulnerable to mental illness. I feel like I made some bad choices, for example, using drugs that filled up my mental illness jar. Is it my fault that I developed psychiatric illness? This is a big fear that many people have, though they may not voice it. It is really important to find a way to determine the cause and address this guilt. In this case, the individual is potentially moving towards self-blame for the onset of his illness, which may increase distress. It will be important to remind this person that usually no single factor causes mental illness. Rather, the mental illness usually arises due to a context of our circumstances at the time, and that as circumstances change, so can our decisions. You can attempt to redirect the client toward a more empowering side of this concept because we can exert some control over our mental health. We can protect ourselves for the future. Although the straightforward answer to this two-part question is strictly no, your upbringing alone did not cause your mental illness and is not your fault, a preferable response would be, again, to reflect that it sounds like there is a real discussion about the factors that are known to contribute to the development of psychiatric illness. How did environmental factor, whatever, play a role in causing a particular environmental factor increases risk for or triggers the symptoms of psychiatric illness? It is useful to help clients understand the idea of vulnerability or susceptibility, that is, affected individuals were born with some level of genetic risk, and different environmental exposures add to that baseline level of risk, moving a person closer and closer to illness onset. I have psychiatric illness because my mom had psychiatric illness, right? It is really important to note when an individual is searching for someone or something to blame for his or her illness and to address the issue forthrightly. Blame of self or other is often associated with mental illness. Rather, mental illness usually arises due to a combination of factors. It is also important to point out that it is not possible to control what genetic information we pass on and what we don't. Although most people would like to pass on only the best of their characteristics to their children, this is beyond human control. It comes down to chance. Can I give a psychiatric illness to my children? The most concise, correct answer to this question would be, children of people with psychiatric illnesses have higher chances of developing a psychiatric illness themselves than people whose parents do not have this illness, but the illness is generally not passed directly from parents to children, except in the case of Huntington's disease. Using the jar analogy, you can explain that parents pass along the genetic risk that is in any person's jar, but it would be extremely rare for parents to pass along a full jar, indicating inevitable illness. We encourage you to use this opening to investigate whether it would be a good opportunity to find out more detail. If I have children, do I pass on my environmental source of great concern, particularly to people who have personally experienced active episodes of mental illness? By them, mental risk factors accumulate over time. Some environmental risk factors tend to be shared in successive generations, for example, poor dietary habits. Some research suggests that the most important it is important to appreciate the genetic components of some of the proposed risk factors, for example, stressful life experiences or substance abuse. As we have discussed already, it is likely that the genetic variants affect how people respond to stress and how much people seek out and how they respond to substance use. So while stress per se may not be passed to children, parents may pass on a predisposition to respond poorly to stressful life events. Now let's discuss a case example. Sarah was diagnosed with schizophrenia as a teenager, and one of her parents had the same diagnosis. Now in her early 30s, she had been successfully treated with an antipsychotic and had not experienced an acute illness episode for several years. She had a partner, was studying at a university, and felt settled and stable. Based on her own history and her family history, having an affected parent, she felt almost certain that any child she had would have schizophrenia as well. She expressed to the counselor that she wanted the illness to go no further in her family line than herself. In addition, her experience over years of interaction with healthcare professionals who had overtly or covertly discouraged her from having a family of her own had led her to believe that she should not consider having children. Despite this, a quiet, small hope for a child led her to make sure she had her facts right, and she came in for genetic counseling. When Sarah learned about the mechanisms by which schizophrenia arises and that her potential child's mental health was not a foregone conclusion, although her child would have a higher chance of developing schizophrenia or some other kinds of mental illness than a child of someone who did not have schizophrenia and a family history of schizophrenia, there was also a large chance that the child would not be affected. She was elated. The genetic counselor discussed with Sarah the potential impact of her pregnancy on her own mental health. The genetic counselor also discussed the difficulty of not having control over the illness risk or the ability to prevent a child from getting ill, and raising a child with the natural fears about whether he or she may start to exhibit symptoms. The genetic counselor's goal in this scenario was to ensure that Sarah's understanding of the illness causation was correct and to help her understand that mental illness was not a certainty for her potential child. It was also important that the counselor temper her excitement at this news with realism, that her child would have a greater chance of developing mental illness than children of mothers who did not have schizophrenia. While this point was challenging, the counselor aimed to help Sarah feel empowered to consider and discuss options for childbearing with her partner in light of complete and accurate information. Now let's turn to discussing the actual disorders. I'm going to briefly discuss with you Alzheimer's disease, schizophrenia, bipolar disorder, and depression. Family history is the best predictor of the risk for the development of psychiatric disorder. 100% of psychiatrists and 83% of psychologists indicated that they take a detailed family histories of mental health disorders. Risk estimates have been established based on family studies, and the risk can be evaluated based on family history. The fact that many of these conditions are relatively common and their symptoms often overlap means that it's not uncommon to see multiple conditions diagnosed in the same family or even in the same individual. This can lead to complicated family histories, and it is difficult, even the genetics professional, to evaluate the risk. In clinical sections of a survey, the lowest confidence levels were reported when participants were asked about their ability to estimate the recurrence risk for children of a parent affected with a mental health disorder. Another concept that in a survey professionals found difficult was the idea of heritability, which we discussed earlier. In general, mental health disorders are thought to be highly heritable. All the disorders listed here, except Huntington's disease, are thought to have both genetic and environmental influences that combine and lead to the development of the condition. The heritability of a disorder indicates the percentage of risk of developing that disorder that comes from genetics. These have been established by studying families where the condition is present and by doing genetic studies of people with the condition. A heritability of 75% could mean that there is one gene that contributes 75% of the risk, or there are 75 different genes that each contribute 1% of the risk. The gene for Huntington's disease was discovered in 1993, and it is known that all people with a particular type of change in the gene for Huntington's disease will develop the condition, and there are no environmental influences that lead to the development of the condition. Therefore, Huntington's disease has a heritability of 100%. Other mental health disorders do not follow the single gene inheritance pattern that Huntington's disease does. There are thought to be multiple genetic and environmental influences that contribute to the development of these conditions. Research has indicated there are a large number of genes that interact in the development and function of the human brain, and that the disruption of a number of these genes can influence the risk of developing a psychiatric condition. Some conditions are known to have a greater genetic influence than others. This graph shows the heritability estimates for the 9 disorders that were investigated in a study. Autism and ADHD were thought to be highly heritable, with heritability estimates in the 80s and 90s. Study participants in this survey were asked to estimate the heritability of these 9 conditions. On the following slide, you will see what they found. As you see, the red dots indicated the median response by the mental health professionals surveyed. You can see that for most conditions, the participants indicated a heritability less than what was reported in the medical literature. The greatest differences between the medical literature and the estimates in the study were seen for autism and for ADHD, where estimates were significantly lower than what was reported by the literature. These discrepancies may stem from our population's lack of experience with these conditions, as mentioned before, or from their understanding of the complex research on this topic. Understanding genetic research topics are important. Now let's move on to other disorders. First, let's talk about Alzheimer's disease. Alzheimer's disease, also called Alzheimer's Alzheimer's disease, or simply Alzheimer's, is the most common cause of dementia afflicting 24 million people worldwide. Alzheimer's is a degenerative and terminal disease for which there is currently no known cure. In its most common form, it occurs in people 65 years old, although a less prevalent early onset form also exists. There are more than 5 million people in the United States living with Alzheimer's. Every 72 seconds, someone develops Alzheimer's. The direct and indirect costs of the disease and other dementias account for more than $148 billion annually. Here are the top 10 warning signs for Alzheimer's: memory loss, difficulty performing familiar tasks, problems with language, disorientation to time and place, poor judgment, misplacing things, changes in mood and behavior, changes in personality, and loss of initiative. Here's the difference between age-related memory decline and Alzheimer's disease. In Alzheimer's disease, they forget the entire experience. The age-related change, they forget only part of an experience. Alzheimer's disease is gradually unable to care for self. Age-related dementia is usually able to continue to care for self. Alzheimer's disease rarely remembers later. Age-related, they often remember later. Alzheimer's disease is gradually unable to follow written and spoken directions, and age-related, they're always continuing and able to follow written and spoken instructions. While scientists know Alzheimer's disease involves progressive brain cell failure, they have not yet identified any single reason why cells fail. However, they have identified certain risk factors that increase the likelihood of developing Alzheimer's. The first of these is age. The greatest known risk factor is increasing age. Most individuals with the disease are 65 or older. The likelihood of developing Alzheimer's doubles every 5 years after age 65. After age 85, the risk reaches nearly 50%. Family history also is a risk factor. Research has shown that those who have a parent, brother or sister, or child with Alzheimer's disease are more likely to develop the disease. The risk increases if more than one family member has the illness. When diseases tend to run in family, either genetics or environmental factors, both may play a role. Remember, it's not one or the other. We also know that there are some genes that increase the likelihood of developing a disease, but do not guarantee that it's going to happen. Scientists have so far identified only one Alzheimer's risk gene called APOE-E4. There's actually 4 versions of this gene, and it's just the E4 that causes the early onset. Deterministic genes directly cause a disease, guaranteeing that anyone who inherits them will develop the disorder. Rare genes that directly cause Alzheimer's genes have only been found in a few hundred extended families worldwide, so there's not a whole lot of information on deterministic genes at this point. Again, going back to the APOE gene, it's a gene on chromosome 19 involved in making APOE, a substance that helps carry cholesterol in the bloodstream. The APOE-E4 gene is considered a risk factor for Alzheimer's disease and appears to influence the age of onset of the disease. Again, it seems to occur earlier when this gene is there. Genetic tests are available for both APOE4 and the few rare diseases that directly cause Alzheimer's. However, health professionals really aren't currently recommending genetic testing for Alzheimer's disease. Well, unfortunately, in terms of treatment for Alzheimer's, we don't have a treatment or a cure. There's no known drug that's assured to delay or stop the progression of Alzheimer's disease. And the Federal Drug Administration has approved some drugs, but they only simply temporarily slow down the worsening of symptoms for about 6 to 12 months on average for about half the individuals who take them, so they're not really effective. The good news is that there is support for individuals who have Alzheimer's in their family. Here's a wonderful helpline and the Alzheimer's Association National Office for you to refer to. Now let's talk about major depressive disorder. Major depressive disorder impairs a person's ability to work, sleep, eat, and function as he normally would. Here are some facts about depression. Almost 15 million Americans have major depression. Everyone is prone to clinical depression. It does not matter your age, race, or social class. Some are just more vulnerable than others to develop clinical depression. Okay? Usually it occurs for people between the ages of 20 and 50, although people over the age of 65 are more vulnerable. But again, it does not matter your age, race, or social class. Some people are just more vulnerable to develop clinical depression than others. Or marital status. Women who are happily married, though, seem to have lower rates of depression. Here are the symptoms of a major depressive disorder. The person must have a depressed mood or loss of interest in activities he or she once enjoyed for a major depressive disorder episode to be diagnosed. For children and adolescents, this may be just an irritable mood. So, depressed mood, a significantly reduced level of interest or pleasure in most or all activities, a considerable loss or gain of weight, insomnia or hypersomnia, behavior that is agitated or slowed down, feeling fatigued or diminished energy, thoughts of worthlessness or extreme guilt, ability to think, concentrate, or make decisions is reduced, or frequent thoughts of death or suicide. Many factors contribute to depression. Each of these factors work in conjunction with each other to make the person develop major depressive disorder. These factors include genetics, environmental factors, and biological factors. In terms of genetics, we do know that the serotonin transporter gene is linked to depression. It controls two parts of the brain, the amygdala and the cingulate, and there are two types of this gene, the long and the short version. This gene seems to code for proteins that recycle the chemical messenger after it's been secreted in the synapse, or the gulf between the cells and the neurons. The amygdala is a fear processing center in the brain, and the cingulate is responsible for emotion dampening. It is located near the front part of the brain. This is crucial for a proper and functional mood regulating circuit. This picture shows the difference in one with two copies of the long version versus having the short version. There does seem to be an inherited vulnerability to depression, and it will not necessarily become clinically depressed, but along with other factors, the person is more vulnerable. Also, there's a higher chance of developing bipolar disorder. If either parent has bipolar disorder, then the child has a 25% chance of developing a major depression. Most of depression research has progressed through twin studies. Researchers have compared twins raised together and twins raised apart to see how much they have developed depression. What we do know is that women are twice as likely to develop clinical depression because of hormones. Serotonin has been shown to influence the sleep problems, irritability, and anxiety. Norepinephrine has been shown to regulate the alertness and arousal. This may contribute to the fatigue and depressed mood of the person. This hormone normally would peak in the morning and then decrease as the day goes on, but in people who have depression, the hormone peaks earlier in the day and does not decrease or level off. At this point in time, researchers are unsure if these imbalances cause the disease or they just contribute to the symptoms. Another hormone called cortisol regulates anger, fear, and stress has also been shown to influence. Finally, this graph shows the correlation between environmental stressful events and people with either the long or short version of the serotonin transporter gene. Among people who have inherited two copies of the stress-sensitive short version of the serotonin transporter gene, 43% developed depression following 4 stressful events in life in their early 20s compared to 17% among people with 2 copies of the stress-protective long version. About 70% of the 847 subjects carried 2 copies of the short version, 31% 2 copies of the long version, and 51% 1 copy of each version. Having a depressed person in the family puts a lot of stress on the other family members. Family and friends might become frustrated with the depressed person because they are withdrawing from society. In consequence, the disease begins to feed on itself. In terms of treatment, we do know that selective serotonin reuptake inhibitors affect neurotransmitters such as dopamine and norepinephrine, and these have fewer side effects. Also, there are things called tricyclics and monoamine oxidase inhibitors, or MAOIs. It's important that any individual continue their medication for 4 to 9 months even if they are feeling better or do not think that the medication is working. Antidepressants are not habit-forming, but they should be carefully monitored to see if the correct dosage is being prescribed. For example, you can see Zoloft and Prozac. The downside of these drugs is that many times it takes a long time to find the proper dosage and what medicines will work for what particular person. Patients go through a lot of turmoil to find this proper combination. St. John's wort has also been looked at in treating depression. Research is still being done on this subject. Conflicting reports have been shown. In Europe, studies were done that showed promising results in treating depression with fewer side effects, but other studies here in America have actually shown no results. Researchers in the UK are researching other alternatives to antidepressant drugs such as talk therapies and exercise alternatives. In some studies, the drug did not work much better than the placebo. Some are saying that the drugs are being abused and should only be prescribed for severe depression and for those who have no other alternatives. There are many support groups and hotlines that people can get help from. Also, many therapies are being used to give the patients support and help them through their feelings of guilt and shame if they're there. Also, since the disorder is becoming more recognized as an actual disorder, more help and support is becoming available to patients. The thought used to be that people were just making up their illness and that they needed to get over it. People were becoming more tolerable of having major depression, and now those that truly need help are actually coming forward with confidence. Now let's discuss bipolar disorder. Bipolar disorder used to be known as manic depression. It's a disorder of the brain that causes rapid mood shifts, changes in energy, and ability to function. The good news is that it can be treated. However, the bad news is it's a long-term illness that is monitored and managed throughout a person's life. It cannot be cured. The facts are that it affects more than 5.7 million American adults, which is about 2.6% of the United States population 18 and older in a given year have been diagnosed with bipolar disorder. The facts are that when symptoms appear before age 12, it's actually often confused with ADHD. It affects men and women equally. The average age of onset is early 20s. Worldwide, 7 out of every 1,000 are affected with bipolar disorder, or 0.7% of the world's entire population. Bipolar disorder causes dramatic mood swings. They range from overly high to irritable to sad to hopeless and then back again. There's often periods of normal mood in between, and these periods of highs and lows are called episodes of either mania or depression. Here are some signs and symptoms of mania and depression. Mania, increased energy, extreme irritability, poor judgment, little sleep needed, abuse of drugs. Depression, sad, anxious, empty mood, loss of interest or pleasure in activities, and thoughts of suicide. There are actually 3 types of bipolar disorder. Bipolar 1, which is the classic form of the illness, and it involves recurrent episodes of mania and depression. Bipolar type 2, which is where they never develop the severe mania, but they're still alternating between depression and hypomania. And then rapid cycling bipolar disorder, where there's 4 or more episodes within a 12-month period. It tends to develop later in the course of the illness, is more common around a woman than a man. Like depression, there appear to be biochemical, genetic, and environmental factors that cause bipolar disorder. Research on the genetics of bipolar has been done using twin studies. There are 3 genes that have been identified that seem to predispose a person to this disorder. We do know that the research tells us that the relationship to an individual with bipolar disorder, the lifetime risk is highest for a monozygotic twin, followed by a child of 2 affected parents. The good news is that bipolar disorder is highly treatable once it's been diagnosed. Mood stabilizers such as lithium or in combination with psychosocial treatment or anticonvulsant medications seem to work. Also, electroconvulsive therapy seems to be beneficial for some. Here are some support groups for bipolar disorder. Now let's talk about schizophrenia. Schizophrenia is a brain disorder in which a person experiences a loss of contact with reality which reduces their ability to live a normal life. This disorder is thought to be caused by a mixture of genetics and environment. It affects 1% of the population, and it does not matter your age, race, or social class. Okay? There is no known cure, and symptoms begin to appear around the late teens for men and early 20s for women. Here are some possible symptoms. They may hear voices no one else can hear. This does not necessarily mean your conscience. A lot of people hear what they call their conscience telling them to do things. This is above and beyond that. They may believe someone is reading their mind, controlling their thoughts, or trying to cause them harm. They may not make sense when talking. They may sit for hours and not move. And they may have a hard time holding a job, maintaining and establishing relationships, or have a difficult time taking care of themselves. Positive symptoms are hallucinations, delusions, thought disorder, and disordered movement. Then there's such things as negative symptoms and cognitive symptoms. A Beautiful Mind is a great example of schizophrenia. There's a little scene on YouTube below that you may wish to view. According to DSM-4, in order to be diagnosed with schizophrenia, the following must occur. At least 2 months of the following symptoms, each present for at least a month: delusions, hallucinations, disorganized speech, or negative symptoms, functioning marked below the level achieved prior to onset, continuous signs of the disturbance for at least 6 months, at least 1 month of which includes symptoms in full and active form. Here's what the research tells us about the risks for schizophrenia. As you can see, again, the monozygotic twins studies show that risk is highest. Here's a graph showing the same thing as the chart. Again, if you notice, identical twins have a higher risk of developing schizophrenia than do the general population. Researchers have found possible gene defects linked to schizophrenia on the following chromosomes: 1, 6, 8, 10, 13, 18, 22, and the X chromosome. Seems to be polygenic or caused by a combination of these gene defects. And there seems to be this gene by environment interaction. Treatments for schizophrenia includes antipsychotic medications and psychosocial treatments, such as educating the patient, helping the patient manage their life, substance abuse treatment, rehabilitations, family education, cognitive behavioral therapy, and self-help groups. Now that we've discussed some of the disorders, now let's move on to evaluating and assessing risk. Information about the risk of a disorder occurring in a family member of an affected individual can be expressed in a number of different ways. You could express it as a lifetime absolute risk. Siblings of individuals with schizophrenia have an X or X in 100 chance of also developing schizophrenia. Or you could present it to them in terms of a relative risk. Siblings of individuals with schizophrenia are X times more likely than a person in the general population to develop schizophrenia. It is important to know which statistic is being reported, absolute or relative, for schizophrenia and bipolar I disorder, for each of which the lifetime population prevalence is approximately 1%. There is little difference between the absolute and relative risk, 10 times more likely than someone in the general population to develop schizophrenia. But for other disorders, such as autism or depression, for which population prevalences are considerably different from 1%, there can be a very large difference between lifetime and relative risks. When you are evaluating and assessing risk, there are some important things you need to remember. First, that risk moves through generations. If you recall basic genetic principles, you will remember that a parent passes to a particular version of each gene, perhaps the allele that increases risk, or perhaps the allele that does not increase risk. The shuffling of the alleles in each child helps to ensure variation in our species, and also means that there is a wide range of risk that can be passed from parents to children. Keep in mind that additional genetic susceptibility might be added to each mating from the other parent. The second is that risk is increased for more than one disorder. So far, we have discussed the risk for relatives of an individual with a particular psychiatric illness of individuals with most psychiatric illnesses, including schizophrenia, bipolar disorder, major depression, anxiety disorders, and pervasive developmental disorders. We know that in addition to having higher risks than the general population to develop the same condition as the affected family member, there are additional risks to develop etiologically related spectrum disorders. For example, a first-degree relative of someone with schizophrenia would have additional risk of developing a schizophrenia spectrum disorder, which includes schizoid affective disorder, schizotypal personality disorder, and paranoid personality disorder. In addition, for relatives of individuals with both schizophrenia and bipolar disorder, risks for unipolar depression are also increased. Remember that there are gaps in available empiric risk data. Despite the fact that these empiric recurrence risks are reasonably well established, they are available for only a relatively limited range of relationships: siblings, children, parents. In addition, in most cases, there are no empiric recurrence risks that take into account the following factors: occurrence of multiple distinct psychiatric diagnoses in a single family, individuals with uncertain diagnoses or symptomatic undiagnosed individuals, multiple individuals with the same diagnosis within multiple generations, affected individuals on both sides. Application of empiric recurrence risk is often confounded because one or more of the factors listed above is commonly found in a given family. Indeed, you will need to find that empiric recurrence risk data exactly reflect the presenting family structure only infrequently. Even in the rare cases when a family's client history exactly matches a family structure for which empiric risk data are available, keep in mind the limitations that apply to empirically established risks. These risks are averaged from a research population. They are neither constant over a lifetime nor without variation between individuals. Factors that influence an individual's risk for psychiatric illness and thus allow you to evaluate the fit of risk data include the following. You must consider characteristics of the family members. Consider the age at onset of the illness symptoms. A young age at time of onset could indicate greater genetic vulnerability, and an increased genetic vulnerability in the affected individual could mean that his or her relatives may have more genetic vulnerability and are at a higher risk for illness. Also, evaluate illness severity. Would the illness be classified as on the more severe end of the spectrum compared to others with that diagnosis? This too might indicate greater genetic vulnerability. For example, relatives of individuals with type 1 bipolar disorder with psychosis seem to have higher risk for developing illnesses themselves than relatives of individuals with type 1 bipolar disorder without the psychosis. You also need to consider the characteristics of the individual for whom you are estimating risk. Consider the age of the at-risk individual compared to the typical age at disorder onset. For example, the typical age at onsets for schizophrenia is during the late teens or early 20s. Risk for developing schizophrenia is lower in the first decade of life, increases through the second, and decreases in the third and subsequent decades. Initial onset of schizophrenia after age 50, regardless of whether someone has an affected family member, is very uncommon. Take into account mental health history, where existing symptoms would suggest that an at-risk person is more likely to develop a psychiatric disorder. For example, if you have been asked to provide information about the risk of a bipolar disorder for someone who is a parent with the condition, and you discover this individual has been recently diagnosed with ADHD, or has already had several episodes of depression, his or her risk for developing bipolar disorder may be higher. Consider the sex of the individual for whom you are calculating risk. As you are aware, there are disorders that have skewed male-to-female ratios, such as major depression and autism, and individuals of the more often affected sex are empirically at higher risk. There is some evidence about sex effects in other disorders, but the data are not consistent and the associated risk seems to be relatively low. Don't forget to consider the characteristics of the family. Consider the family structure. Does the individual for whom you are calculating risk have only one affected family member or multiple affected family members? A greater number of affected family members usually indicates greater genetic vulnerability and therefore greater risk to family members of affected individuals. Consider both sides of the family. In situations where both sides of the family are affected, it is impossible to determine if the risk factors on one side are the same as risk factors on the other side, and because the vulnerabilities conferred by each side may be different, it is not clear what the cumulative effect would be. It is reasonable to assume, however, that the risk is increased if the at-risk individual inherits susceptibility from both sides of the family. Here's a case example. The following scenario presents an example of an oversimplified and incorrect application of empiric risk, followed by a correct application. A client with schizophrenia has a sister who is concerned about her risk of developing schizophrenia. You find the empiric risk for a sibling with someone with schizophrenia to develop the same condition is about 8 to 15%. You tell the sister, your risk of developing schizophrenia is 8 to 15%. Now I want you to think about this. Why is this answer incorrect? Remember that empiric risk are population-based risk. Risk for psychiatric illness varies over the lifetime and between individuals, so empiric risk cannot accurately be applied like this to any particular individual. Now think, why is this answer oversimplified? Essentially, without knowing more about the sister, her age, current mental status, and family history, we cannot know whether the empiric risk data are a good fit for her particular situation. Let me illustrate. If the sister is 50 years old when you speak with her and she has had no mental health problems at all in the past, it's very unlikely that she's actually going to develop schizophrenia. Her risk of developing schizophrenia would be considerably less than even the lowest end of the risk range, 8%. So in this case, empiric risks do not fit the family situation very well. Given these additional details, the empiric risk is an overestimate. Consider this. If the sister is 15 years old and has recently been experiencing non-specific lowered mood, anxiety, and social withdrawal, she is likely at considerably greater risk than even the higher end of the risk range, 15%, for developing schizophrenia. Indeed, it could be that she is currently experiencing prodromal symptoms. Given these additional details, the empiric risk is an underestimate. If the sister is 15 years old, and in addition to the single affected sister we know about in the original scenario, another of her 3 older siblings might be affected, as is one of her parents, her risk would again be substantially higher than even the higher end of the risk range, 15%, that was quoted as being her risk. Given these additional details, the empiric risk is an underestimate. Remember that empiric recurrence risk should be seen as foundation for estimating the risk that the disorder may happen again in a particular family. But in order to use these data appropriately, it is important to individualize them as much as possible. In the example above, a better response to this scenario might be the following. If we look at the empiric risk for schizophrenia for someone who is an affected sibling, we can see that, in general, 8 to 15% of siblings of an individual's schizophrenia will develop that condition themselves. But we know that someone is going to develop schizophrenia. The symptoms usually begin during the late teens or early 20s. You are 50 years old, so we could estimate that your risk to develop schizophrenia is probably a lot lower than even the lower end of that risk range. Now let me give you some tips for incorporating risk assessment into practice. We appreciate that risk assessment is complex and may seem overwhelming to many clinicians. Take this process one at a time. At minimum, consider the following, which I can increase risk. Many affected relatives, especially look for multiple affected individuals over several generations, especially when the affected relatives are closely related to each other and to the at-risk individual. Several affected siblings in one family when the at-risk close relatives on both sides of the family who are affected, especially when one or both of the parents of the at-risk individual is affected. Early ages at symptom onset for disorders that typically have an adult onset, especially look for the childhood or adolescent onset. Symptoms in the unaffected at-risk person, which may be early prodromal symptoms, or young age of the at-risk person, has most of his or her important lifetime risk ahead. The most important flag of decreased risk is an at-risk relative who has lived through a good portion of his or her risk, for example, an unaffected individual in her late 40s in a family with bipolar disorder. Again, taking the stepwise approach, it may be useful to consider the following items as you think of how to individualize empiric risk results. Determine who in the family is affected and with which disorders. Consider the number of affected individuals and their characteristics, including age at onset, severity of symptoms, and sex. Determine who is at risk for which disorders. Include characteristics of the at-risk person, including age, sex, and current and past mental status. Put parameters around risk assessment. When evaluating the family history to determine the best estimate of risk for illness, it can be helpful for you to keep in mind the constraints of what may be the upper and lower limits for psychiatric illness. Until we know enough to contribute protective genetic and environmental factors to risk, the lower limit of the risk will always be the population risk for that disorder. For example, approximately 1% for either schizophrenia or bipolar disorder and 10 to 20% for known. But for all but the very highest risk situations, 50% is likely to be a reasonable estimation of an upper limit. It is important to note that in most cases, however, an upper limit of 50% will grossly overestimate the risk. The 50% figure roughly approximates the risk for an identical twin of an individual with a psychiatric illness, an individual who has 2 parents affected with the same psychiatric illness, or an individual who has multiple affected family members in multiple generations, and a young person who has experienced an affected first-degree relative and who has recently started experiencing nonspecific lowered mood and social withdrawal. Remember to ask the key question, given what I know about this individual's personal and family history, how well do I think that the empiric risk estimate the actual magnitude of risk in this case? In many cases, our knowledge is not adequate to allow for accurate quantitative modification of risk for any individual client, but research provides us with ample data with which to qualitatively modify risk to better match the individual history. Think of empiric risk as indicating the approximate magnitude of risk. If the client's data are reasonable match to the medical and family history. If you are less experienced with risk assessment and less knowledgeable about the research literature, you may feel most comfortable explaining to clients whether their risk is less likely to be the empiric risk, similar in magnitude, or more than the empiric risk. More experienced clinicians may wish to go a step further and use the risk assessment. The more general estimate of the lower limit of 1% and an upper limit of 50% to be a more specific estimate of a lower limit of 30% and upper limit of 50% based upon the client and family history. A detailed discussion of how to do this is beyond the scope of this lecture, but referral to a genetic specialist for this type of risk evaluation is appropriate. Once you've thought through risk assessment in a stepwise fashion, explain to the client and family why the empirical risks fit their particular family situation or why the data do not. In other words, explain why you think their specific risk in question is higher or lower than the empiric risk might suggest. We find that it might be useful to walk clients and family members through the thinking in a very similar manner to what was described above. For example, you might say, these are the people who are affected. These are the people who seem to have the most risk. These are the disorders I'm most concerned about in terms of risk. I noticed the following indicators of increased risk in your family history. This is the empirical risk range based on the literature. Here's how well I think the range estimates your family risk. Just when you thought you might be catching on to family risk assessment, let's talk about some genetic syndromes that we mentioned earlier. It is important to remember that even taken together, these syndromes are rare, accounting for probably only a few percent of those affected with major psychiatric illnesses of adult onset. Because they are so rare, it is not important that you memorize the disorders or their features, but rather simply be aware of the red flags that suggest an increased likelihood of a genetic syndrome. If a family or medical history raises suspicions of a genetic syndrome because one or more of these red flags is present, I recommend a referral to a specific genetics specialist for consultation and exam. For example, a specialist with psychiatric genetics. In situations where the psychiatric illness occurs in the context of a genetic syndrome, family risk will often be very different from empirical risk estimates that I've discussed previously. Red flags indicate increased likelihood of the presence of a genetic syndrome include a history of birth defects, such as a cleft lip, cardiac abnormalities, or club foot. A history of learning problems or intellectual disability. A history of unusual health problems. Or a multi-generational family history of psychiatric illness, especially when combined with a history of birth defects or a history of learning problems or intellectual disability or a history of unusual health problems. In addition to the fact that for many of these syndromes, the risk for additional family members to be affected may be quite different than the psychiatric illness that is unrelated to a genetic syndrome, it is important to identify families with genetic syndromes because there may be additional health risk and different treatment recommendations. In addition, a much more accurate genetic risk assessment, and in some cases genetic testing, is available for the syndromes. Now let me turn to a risk assessment when the client has autism. The syndrome red flags are important for all psychiatric disorders, but especially cognizant of the importance of single gene disorders and changes to chromosomes as causes of autism and other pervasive developmental disorders. An underlying genetic cause can currently be found in about 15% of cases of autism seen in clinical genetics clinics, and that number is expected to rise with the advent of new genetic testing capabilities. All clients with autism benefit from an evaluation by a genetics professional to determine whether genetic testing is appropriate and which tests might be most informative. The major challenges in the risk assessment process, including deciding who to count as affected, taking into account the spectrum of illness and the illness age at onset, and the occurrence of psychiatric illness on both sides of the family. I'm about to describe each of these. As I've described, risk assessment depends on evaluating the pattern of both affected and unaffected individuals in the family. However, establishing whether someone is affected or unaffected is not always straightforward for the reasons I'm about to describe. First of all, evaluating a family history to provide a basic risk assessment for an individual who is an affected parent to develop bipolar disorder. You notice that in addition to the affected parent, there are other close biological relatives who have had diagnoses of major depression, ADHD, and schizoid affective disorder. Should these relatives be counted as affected or unaffected for the purposes of risk assessment in this situation? It is important to remember that the factors that can increase vulnerability to bipolar disorder are not thought to be specific to bipolar disorder. Rather, it seems that these factors can increase vulnerability to psychiatric illness more broadly, and that the particular combination of vulnerability factors found in a given individual determines what disorder he or she may eventually manifest. The particular group is closely related etiologically, implying shared factors that can increase the risk of these illnesses in this family. Thus, it may be prudent to explain this to the individual for whom you are calculating risk and err on the side of providing a worst-case scenario risk estimate. by counting all of these individuals as affected. For a history as complex as that described previously, a referral to a genetics professional with a specialization in psychiatric genetics is often appropriate. You might also consider undiagnosed relatives. A similar situation arises when a family history reveals members who did not have psychiatric diagnoses but who had, for example, nervous breakdowns or were described as being strange or as loners. Gathering as much information as possible about such family members can be helpful in deciding whether to consider them as affected or unaffected for the purposes of risk assessment. Substance misuse. Family members who have had issues with substance misuse raise other challenges. Alcoholism is a psychiatric diagnosis in its own right, but also reliance on substances may be a form of self-medication for other concurrent psychiatric conditions. So again, determining whether to consider these individuals as affected or unaffected can be difficult. Whether or not you decide to include someone with a substance misuse is affected, it is important that clients and families understand that risk for substance use may also be increased in the family. Also remember that psychiatric illnesses that tend to have an adolescent or adult onset add an additional element to the risk assessment because of the challenge of determining whether unaffected relatives are truly unaffected or whether they were actually developed, or if they have passed away, would have developed psychiatric illness. Even if they were not affected, they may have had a genetic vulnerability that could be passed to subsequent generations, but it's impossible to know how much. Unaffected individuals with a family with a psychiatric illness will be unaffected possibly from one of the following reasons. They have had enough genetic and environmental risk to fill up the jar and will go on to develop illness in time unless they die from another cause before expressing symptoms. They have a significant genetic risk but insufficient environmental risk to fill the jar and or protective environmental factors that reduce the risk. They have a small moderate amount of risk, and it is very unlikely that they will ever accumulate enough environmental vulnerability factors to fill the jar. Or they have very little genetic risk, and thus it is very unlikely that they will acquire sufficient environmental exposures to fill the jar and cause illness. At-risk individuals described under the first two items have a full complement of genetic risk and may pass this on to the next generation. Recall the random 50% chance of passing each allele to a child. The at-risk individuals described under item 3 have a smaller amount of genetic risk that can be passed along to future generations, and those individuals may manifest illness if they accumulate sufficient additional vulnerability. The at-risk individuals described under the last item will pass along little or no genetic risk to future generations. Individuals on both sides of a family as a result of stochastic factors, the frequency of the disorder, and because of assortative mating, the tendency of individuals with psychiatric illness and or family histories of psychiatric illness to choose one another as mates. At this time, it is not generally possible to determine how to integrate risk from both sides of the family. Empiric risk data that pertains to risk from both sides of the family are at best limited to a child who has two affected parents. We do not have sufficient data to support simply adding the empiric risk from both sides of the family. The combined risk may be additive, but it could also be multiplicative, or some of the risk could cancel out. So what do you say to clients and family members during the risk assessment? You might say, your family history and your own psychiatric history are vital to the risk assessment process. Explaining that these are vital pieces of information is a good way to begin to talk about risks with clients and family members. It can also be helpful to explain that certain aspects of already discussed the factors that contribute to the development of psychiatric illness, which is advisable before discussing risk. You can explain that by looking at a personal and family psychiatric history in the jar, which in turn helps you estimate risks. What psychiatric disorders are you concerned about? It can be helpful to establish whether an individual is concerned about risk for a specific disorder or for psychiatric illness in general. The answer to this question allows you to focus on, that is, that there is a risk for more than one disorder. Many clients are not aware of this, and they will not be able to determine whether they want risk information for psychiatric illness in general until they appreciate the risk for the wider set of the disorders. It is important to remember that almost everyone has some risk for psychiatric illness, even with people with no family history. It is good to remind concerned family members or clients that psychiatric disorders are very common and that empirically everyone is on the same level of risk. We find it useful to bring up this point at the beginning of a risk discussion and to reiterate it often where is needed. Coming up with an estimate of risk for psychiatric illness is not an exact science, but by using these empiric risk numbers together with the information about you and your family, I can give you an estimate of what your risk may be. Explicitly acknowledging the limitations of risk estimation process allows clients and family members to form a more accurate expectation. Now let's discuss some issues that clients and their families may raise when you are discussing, evaluating, and assessing risk. Where do these numbers come from? You can explain to the client that the numbers originate from the research studies that generated empiric risks. Being very upfront about the benefits and limitations of the data, you could discuss or review again how the client's medical and family histories affect the application of empiric risk to his or her situation. Note that such a question might warrant your checking in to evaluate the motivation for the query. It may be that the number you provided was quite different from what the individual was expecting, and the client is finding it difficult to believe. Why do you need to know about how old my relative was when he or she got ill in order to give me an idea of what my risk is to develop psychiatric illness? There are different motivations for this question and therefore different possible options for response. One possibility is that the client is curious and eager to evaluate his or her own family history. You could explain that one of the indicators for increased risk for family members is at an early age to indicate that the individual is finding it difficult emotionally or practically to answer your questions. You could reassure your client that it is not normal to not be able to answer all the questions asked during the family history and suggest that even he or she does not know the exact answer to the questions. Any information can help you personalize the risk estimate as much as possible. You could also remind your client that others in the family may have more information about the family history and encourage him or her to obtain more information when possible to add to the family history at a later visit. Now let's discuss some case examples. In this case, I am providing a risk estimation for the young man, age 15, indicated by the pedigree. I took a detailed, targeted psychiatric family history. There is good information about the psychiatric history on all the family members in the pedigree, and his only affected family member is his older sister, who has been diagnosed with schizophrenia when she was 21 years old, which is actually the typical age of onset. She experienced her first symptoms only months before her diagnosis. The young man himself reports no psychiatric problems, and his family reports no concerns about his mental health. He has friends, is active, does well at school, and is engaged with his family. In this situation, the empiric risk for a sibling and an individual affected with schizophrenia is the best possible fit for this young man with this particular family history, because there are no red flags indicating increased risk or markers for decreased risk. In this case, here again, we're providing risk estimation for the young man indicated by the arrow on the pedigree. As in the previous case, I took a detailed, targeted psychiatric family history. There's good information about the psychiatric history of all the family members in the pedigree, and only affected family members are those identified. He has a sister who was diagnosed with schizophrenia when she was 10 years old, but in retrospect, the family thinks that she had probably been sick, responding to command hallucinations, for 2 years prior to the diagnosis. The father of the young man has a history of recurrent depression for which he takes a selective serotonin reuptake inhibitor, SSRI. The mother of the young man was diagnosed in her 20s with schizoid affective disorder. Like her daughter, she has been stable for many years on Risperdal. The maternal grandfather of the young man was diagnosed with schizophrenia, and he committed suicide at age 60. A male maternal cousin of the young man was also recently diagnosed with schizophrenia and has just started taking quetiapine. The young man himself is quiet and withdrawn and answers questions posed to him with 1 or 2 words while looking at the floor. His parents are frustrated and concerned about him. They just found out that his grades have deteriorated at school and that this last week he's been skipping school. For this particular family history, the empiric risk for this young man to develop schizophrenia is probably a very poor fit because there are many red flags indicating an increased risk. His sister was diagnosed at a very early age. In addition to his affected sister, both of his parents have histories of psychiatric illness, as does his grandfather and cousin, and his behavior indicates that his own mental health may have started to deteriorate. It seems likely that his risk of developing psychiatric illness is considerably higher than would be indicated by the empirical risk. In this circumstance, a mental health evaluation for the young man in question is definitely appropriate, and if the family members remain interested in discussing risk, referral back to you might be appropriate as well. Now let's turn from evaluating risk to explaining risk. We recently discussed how you might evaluate the level of risk in a family in relation to empiric risk and derive an individualized risk estimate based upon characteristics of the family. Now I'm going to focus on how to talk about this risk with your client and their family. The overall goal of family risk counseling is to personalize and effectively convey messages about risk for other family members to be affected. There is a great deal of variation in how individuals understand risk, and comprehension cannot necessarily be assumed based on verbal ability or level of education. It is best to assume that your client or family member needs some assistance with understanding characteristics and statistics because many people often have a very difficult time understanding numbers and numerical concepts. Here are some tips for providing numerical risk to clients or family members. Check in frequently to assess understanding. It is not uncommon for individuals to perceive risk from a dichotomous all-or-nothing perspective. It is or it is not going to happen. It may be difficult for people to process risks that are presented in a numerical fashion. For example, 1 chance in 10 or 10%. Check in frequently to assess understanding and remark it is only natural that clients should think of terms of is going to happen versus is not going to happen, since that is the lived truth of the matter, and that is the unfortunate that we are unable to provide definitive information. Use different methods to present risk, for example, visual aids, analogies, and written materials. Many clients make very simple charts or graphs. Talk through your risk assessment process. When describing risk to clients, provide the population risk as the bottom limit of the empirical risk and discuss it as a comparison point for other risk numbers. Use absolute risk as the basis for your risk discussion. Relative risk may be interpreted incorrectly, especially when the disorder is comparatively rare in the general population, for example, autism. If the client seems to understand the absolute risk, in some cases it may be useful to clarify understanding by comparing the absolute risk to the relative risk. Present risk in several different ways, taking cues from your interactions with the client that inform the approaches you use. I suggest that you begin by presenting the numbers in the form of, out of 100 people with the same level of risk, 25 would become ill, because numbers in this form tend to be best understood by the majority of people. You may also want to state the numerical risk as a percentage, 25%, or as a fraction, 1 in 4 chance. It is important to frame risk from the perspective of negative and positive outcome. For example, after you explain that, if there were 100 people with the same risk level, 25 would become ill, you could say that this is the same as saying 75 of the 100 people would not become ill. Point out explicitly that the majority remains well. There are very few cases where the family history is significant enough that we'd expect a majority of relatives to become ill. If you encounter a case where you suspect a mental illness onset is more likely than not, based on the family history, consider referring the client, again, for a psychiatric genetics consultation. You may also want to give qualitative risk data as well. Some individuals do not wish to be given a risk number, but instead prefer qualitative assessment of risk. It is important to be aware of the potential for misunderstanding that comes when using qualitative risk assessment only. Your own perception of risk level, very low, low, moderate, high, very high, is very subjective and may be quite different from that of the client or family member. Therefore, the use of such qualitative terms introduces the potential for bias and should be approached with care. If you use such phrases, it is vital to evaluate how the client or family member perceives the phrase. We find it useful to approach qualitative discussion of risk by stating that the risk is increased above the general population level and then focus on how relatives are more likely not to be affected. Because most families only have one affected family member, the most appropriate empiric risks are often associated with the 10 to 15% range or lower. For families, qualitative assessment might be posed, as I'm going to describe later. So what do you say to clients and family members about risk assessment? Well, you might say, what do you think you might do with the information about risk for family members? This is another method of encouraging your client to consider what the impact of risk assessment might have for him or her, and allows you to help the client manage the new information. You said that you were interested in hearing about the chances of other family members being affected. Are you interested in hearing specific numbers, or would you like me to talk about it in terms of higher or lower risk than other people? This kind of question allows you to tailor the information you provide more precisely to the needs of the individual client, while again preserving the client's right not to know. What would be an acceptable level of risk to you, and what point would you feel the risk is too high to consider having children? It can be important to establish whether this point is something to which the client has given thought or not. Exploring some of these issues prior to embarking on the risk assessment encourages the client to consider potential reactions in advance of receiving the information. You could say, these risk numbers I am talking about come from research studies that have investigated large groups of relevant relatives and disorder. For example, siblings of individuals with bipolar disorder. These studies examine how often the siblings also develop bipolar disorder. Some of the siblings in the research study developed bipolar disorder, some did not. The empiric risk figure is simply the proportion of people in the research group who develop bipolar disorder, so it is an average. It gives us some good information about overall risk in the research group, but it doesn't tell us exactly what the risk of any one person is or in the group was, and it doesn't tell us exactly what your risk is either. Many clients and family members appreciate understanding the source of risk estimates. This is one way in which you might try to explain the concept of empiric risks. You might also say, your children do have an increased chance of having the disorder in question. However, your children have a much better chance of not having it than having it. Presenting the qualitative information from both angles, both as the chance to be affected and the chance to be unaffected, is an excellent strategy that can be used in a large majority of situations because risk is often estimated at less than 50%. Your children do have an increased chance of having whatever disorder it is, though it is certainly possible that they will be well. For now, let's focus on the possibility that they could be affected and discuss psychological reactions, early intervention, or whatever issue seems to be most salient at the point. For an affected individual who is a family with an unusual number of relatives with psychiatric illness, you might want to consider something like this statement. This kind of statement indicates in broad terms that the chances are likely quite a bit higher than the empirical risk, and it seeks to explore the impact of the information and/or allow some anticipatory guidance in the form of perhaps opening conversation about risk management. You might also want to say something like, because your family history is a little unusual, you have several affected close family members, and you have said that you would like to be the most accurate numerical risk estimate possible. I think it might be best for me to refer you to an expert who is better equipped to provide this for you. How would you feel about that? Raising the possibility of referral to a specialist for genetic risk assessment is certainly appropriate in situations like this one, where the individual has a significant family history and is asking for the most accurate numerical risk assessment possible. It is important to keep these chances in context. The chance for anyone in the general population to develop a psychiatric illness is X in 100. It is always helpful to return to the point that although there is a chance for psychiatric illness may be higher for someone who has affected family members, no one has. Now let's talk about some issues that clients may raise when you explain risk. I'm not worried because in my family, psychiatric illness skips generations, so because I'm affected, my children will be fine. This is a great example of an idea that might be helpful to gently challenge or confront. In some cases, you can review the family history with a client and show that the perception itself is incorrect. Alternatively, it could be so far in this family, it does appear that the psychiatric illness is skipping a generation. Although this is how the condition is manifested so far in the family, there is no guarantee that this will continue to appear this way. Again, going through the JAR model and discussing the causes of psychiatric illness can help demonstrate how illness can skip a generation while the genetic variants that increase risk are nonetheless passed on. The unaffected generation will still have the genetic vulnerability, but until now, by the chance, they have not accumulated sufficient environmental vulnerability to manifest illness. There's always a chance that the children of this individual could in fact develop mental illness. This is not going to be a reassuring point for this individual to hear, but it can be helpful to ensure that he or she has the correct information available. It will also be important to pay attention to the emotions that this type of discussion evokes, and to redirect attention to the fact that the children are more likely to be well than ill. Also point out that this family is well placed to manage any emerging illness well by obtaining the appropriate help and treatment in a timely manner, so to promote the greatest potential for the best possible long-term prognosis. Can you just tell me if I'm right to be worried instead of coming up with a number? No client should be forced to hear a quantitative risk evaluation if they prefer a qualitative approach, and no client should be forced to hear about risk at all if they are not emotionally ready for the discussion. Simply express empathy for the client's concerns and consider that perhaps there's some frustration being expressed, the source of which it might be helpful to identify. Point out that worry depends far more on simple numbers. Instead, it is a product of perceptions regarding severity of illness and accompanying burden along with other factors. Exploring the reasons for worry might reveal previously undiscovered information. For example, perhaps this client is so concerned because she thinks the family member could actually be expressing symptoms of mental illness and that he or she is not disclosed. But it is also very possible that the client is simply not comfortable with the numbers or finds numerical risk assessment too intimidating. I'm not concerned about my child, but my pediatrician tells me not to worry, that he's fine. It could be helpful in this situation to reflect that it's hard to live with the increased chance, to wonder if the behaviors you observe are normal or if they may in fact be the first manifestations of mental illness, and to explore in more depth the reasons for this concern about the child in question. And this further exploration leads you to feel that the concern needs to be more thoroughly addressed. For example, if the pediatrician has no experience with or interest in psychiatric illness and the concern seems to be well founded, you can gently raise the possibility of a referral to a different mental health specialist to evaluate the child. I don't understand the numbers. Normalize this experience. Most people actually struggle with the kind of numbers involved in risk estimation, and praise the client's honesty. Ask what format might be helpful and suggest some alternatives. For example, would it be helpful if I drew a pie and showed you, as fractions of the pie, how much chance there was that the individual being discussed would have a mental illness, and the chance that he or she would not have mental illness? Keep in mind that most clients find natural numbers more accessible than statistics. Why did you tell me that you think my risk to develop psychiatric illness is probably less than the 15% risk I found on the internet? Assuming that the 15% figure is an accurate reflection of the empiric risk data in the literature, explain to the client that the numbers that he or she found on the internet are average risk, and what you have done is to take into consideration the information the client gave you about the personal and psychiatric history to modify that risk for the unique situation. You can explain what exactly it was that made you feel the risk was lower. For example, we know that it is very unusual for people to develop schizophrenia after age 50, regardless of whether they have a family history of the illness. If the 15% figure does not reflect the information you have about empiric recurrence risk for the illness in question, it can be helpful to point out that although there is a lot of information about genetics and psychiatric illness on the internet, not all of it is accurate. Consider looking at your client's source to see if it reflects newer information from a reputable source. Explain where you obtained the empiric risk numbers you provided. Let me give you a case example. Alexandria was a healthy woman in her late 20s who came in for consultation with her husband. She had a brother with schizophrenia. Alexandra had a 5-year-old son, and she was concerned about the risk that her child might develop schizophrenia. She was quite forthcoming about her family history and her concerns about her son, who was exhibiting some mild issues with social maturity. When the counselor asked Alexandra about her own mental health and that of her husband and his family, she reported no concerns. The counselor pressed Alexandra a bit about her own mental health, explaining to her how important her status was in relation to the risk for her son. She was resistant to any discussion that could be touched on her own risk of developing a psychiatric illness. She told the counselor that she would have experienced issues by now if she was going to, and she had not come to discuss her own risk. By gently pointing out again how important her own mental health was to her son's risk, the counselor was able to engage Alexandra in a limited discussion around her risk, although the counselor assured her that she would not provide her with a risk number if she did not want to receive the information. The counselor spent much of the remainder of the session discussing how difficult it was to have an affected sibling and how important her ongoing mental stability was to her own self-image, though for many years she had harbored secret fears about becoming ill. She agreed to hear more about her own level of risk of mental health status informed the risk to her son, and the counselor agreed that she was on a very good, stable track in terms of her own mental health. She ended up being quite pleased by the likely risk that the counselor provided by her son, assuming that she remained well, stating that it was much less than she had expected. Now I'd like to turn to talking to specific populations. It is common public misperception that women with serious psychiatric illnesses do not have children. In fact, most do have children, and many pregnant clients have a variety of concerns about how their illness and treatment may directly or indirectly affect their offspring. I'm going to begin by addressing counseling issues about the causes of psychiatric illness and risk when the individual is pregnant or the individual is planning a pregnancy is personally affected with a psychiatric illness. So what do you say to the woman that is pregnant, or maybe considering pregnancy? I appreciate that you told me that you would like to begin a family soon. Let's make a plan that will give you the best chance to have the healthiest pregnancy possible. Often individuals with psychiatric illness, particularly serious psychiatric illness, are discouraged from having children by clinicians, family members, and friends. It can be a huge relief to have a clinician who responds in a supportive and constructive manner to a disclosure about a desire for children. That does not mean that you should share concerns, but the concerns should be expressed as part of a supportive discussion focused on solving problems and reducing risk. It can be helpful to explore to what extent the individual feels supported in this desire by friends and family, as often their support is a crucial piece of a successful pregnancy and postpartum plan. When planning a pregnancy, it is really important to think about how it might affect your mental health. Would you like to talk about this with me? We could start to make some plans for how we best maintain your mental health during and after a pregnancy. This is a good way to open a discussion that might allow your client to share her wishes and beliefs, and it allows you to address misconceptions. For example, you may find out that the client is not concerned about her mental health during the pregnancy because she has heard that the pregnancy protects against mental health problems, and so she is not planning to take any medications until after delivery. In addition to allowing you to challenge misconceptions and share information, it also opens a space for the introduction of advanced directives. How do you imagine that you would like to handle medications during the pregnancy? As we have seen, decisions around how to manage mental health problems, the risk to the fetus associated with exposure to psychiatric medications must be weighed carefully against the risk to the mother and the fetus of untreated psychiatric illness. Despite this need for careful assessment, many women have instinctive negative reactions to the idea of using psychiatric medications during pregnancy. Exploration of these issues early in the pregnancy or in the process of planning a pregnancy allows you to gather detailed information to evaluate the potential benefits and harms and refer to your client to clinicians with specialist expertise if available and appropriate. All women who are considering pregnancy should take folate supplements. Folate is vitally important during pregnancy for the successful closure of the fetal neural tube and reducing risk for certain types of birth defects. Even though foods such as bread and cereal often have added folate, it is recommended that women planning pregnancy take additional supplements, usually 0.4 milligrams daily. Prenatal vitamins often contain 1 milligram of folate. Women on psychiatric medication should talk to their family doctor or psychiatrist about how much folate to take because the recommendations vary for women taking different medications. For example, women taking valproic acid should take 4 to 5 milligrams of folate daily, an amount. You should make sure, though, that women taking valproic acid do not misinterpret that advice to mean that they should take 4 prenatal vitamins a day. Prenatal vitamins contain many different nutrients. Some of them can have harmful effects if taken in excess. It is best to begin taking folate at least 3 months before conception, but for women who have not been taking it and who are already pregnant, it's still too good to begin as soon as possible. Some people who have been diagnosed with psychiatric illness want to know their chances for their children to develop a psychiatric illness in later life. Is this something that you would be interested in talking about? Many clients will appreciate your bringing up this topic for discussion during family planning situations. Some clients would like to know this information but are not sure how to ask the question, have not articulated that desire for knowledge to themselves, or afraid to ask. It is always appropriate to refer individuals for more genetic counseling if preferred to address family risk. Here are some issues that clients may bring up. Can I be a good parent even though I have a psychiatric illness? You could reinforce the point that people with psychiatric illnesses can be great parents or bad parents, just as those without psychiatric illness can be great or bad parents. Parenting is difficult for everyone, and every parent has his or her own challenges, but there is no reason to think that having been diagnosed with a psychiatric illness would preclude a person from being a good parent. As for all parents, it is good to be aware of and alert to one's own challenges and ready to take action, for example, by having a mental health plan in place, such as a Ulysses agreement. Explore the motivation for this question. For example, has this person recently had a conversation with someone who is or her illness she could not be a good parent? I heard that if you take psychiatric medications during pregnancy, your baby will have birth defects. Is this true? Explain that for many psychiatric medications, there is no evidence of any increased risk of congenital abnormalities or birth defects. For quite a few medications, we do not think there is much or any risk, but we cannot say for sure. A few medications, though, have been shown to increase risk for certain kinds of congenital abnormalities, though the risk of a baby having one of these problems is still quite small. There's a far greater chance that the baby will not have any birth defects, even if the fetus were exposed to psychiatric medications in utero. Some women find it helpful to know that many birth defects can be detected during pregnancy by ultrasound. Follow up by asking if the client is pregnant or thinks she may be pregnant. Some women have terminated pregnancies because they believe it to be a foregone conclusion that the baby would have birth defects, and so it is important to be especially careful that the pregnant clients receive detailed education and counseling so that they make informed choices. It is helpful to empathize with the fear of becoming symptomatic while pregnant for caring for a newborn. Reassure your clients that you will help them plan ahead to develop the best possible plan for a successful and healthy pregnancy and postpartum period. Validate that these are really important things to be thinking about and to offer together to come up with a plan for how these possible situations should be handled. Mary marched into the genetic counseling room and defiantly introduced herself. I am 42, bipolar, single, I don't take medications, and I'm going to get pregnant because I want to have a baby. Mary had been referred by her family doctor. The doctor had strong feelings which he shared with Mary that she should not attempt to become pregnant. She told me that the relationship had deteriorated when she had stopped taking the lithium he had prescribed her, and that he insisted that her illness would not allow her to be a competent mother. She felt he had referred her to me, the genetic counselor, in hope that I would persuade her to relinquish her plans for pregnancy through donor sperm. I reflected that I could imagine that she may feel hurt, angry, and disappointed by this kind of response, feelings she affirmed. We talked about her perceptions of others, reactions to her to wish to become pregnant. While she had caring friends and a family around her, it seemed that they were also not supporting her in this decision. I explained it was not my place to try to persuade her to do so, or not do anything. I told her that I believed that she was the person best able to make the right decision for herself, and I wanted to make sure that she had all the information that she might need. At this, any remaining defensiveness evaporated, and she visibly relaxed. I asked if she had questions about pregnancy-related issues, although initially she had only one question, about her chances of getting pregnant at age 42. As we talked, she came up with more and more questions. She'd heard that lithium causes fetal heart defects, so she was trying to stop her medications before trying to get pregnant. What was the chance she would experience an episode of illness during the pregnancy or afterward? If she made sure that the sperm donor she had used had no family history of psychiatric illness, what would prevent the baby from developing bipolar disorder later in life? Over the next hour and a half, we talked about how it would be more difficult for her to get pregnant at age 42, about the risk to a fetus associated with a mother taking lithium during the pregnancy in relation to the risk of having a baby with a birth defect that all women face, and how risk for some conditions increase as women get older. We also talked about how all women have some risk for developing depression or psychosis during pregnancy or the postpartum period, but how these risks are greater for women who have been previously diagnosed with bipolar disorder, particularly if they are not taking their medications. We discussed single parenthood and the importance of support of friends and family. Based on her family history, we also talked about the chance for a potential child to develop psychiatric illness later in life, assuming she selected a sperm donor with no family history of psychiatric illness. We discussed that family history provided by sperm donors is limited and is not confirmed by a health care provider, and that not even a perfect family history report from the sperm donor did not guarantee that he or his relatives would not later develop mental illness. At regular intervals, I check in with Mary to see how the discussion was making her feel. She told me that it was a lot to take in and that she realized that there were many factors she hadn't considered. When I left, she took several informational booklets and accepted the referral I offered for the local reproductive mental health program. I do not know at what decision Mary arrived in the end, but my goal, as I told her, was to provide all the information that she needed in a supportive environment to enable her to make the best possible decision. And in the end, I believe this is just what she did. Most clients who are concerned about the potential for psychiatric illness in their at-risk relatives, and most at-risk individuals who are concerned about their future mental health, are very interested in risk reduction efforts. They tend to express some hope that environmental modification, new treatments, and early intervention may help the risk or minimize the effects of the disorder, but often tell us that they do not know what concrete steps to take to minimize risk. Being at risk or having children at risk is intensely worrisome for some clients and family members and may lead to chronic monitoring of mental health status. Anticipate that for parents of at-risk children, discussions about risk reduction may be one of their primary goals. I'm now going to focus on the most important and common aspects of these discussions, including parenting in the home environment, early symptom identification, other prevention efforts, lack of control over the illness onset, and timing of risk reduction efforts. Many parents of at-risk children have questions about the optimal timing of risk reduction efforts, and unfortunately these questions are very difficult to answer. You can explain that genetic risk is present from the point of conception and that environmental risk factors may accumulate over time. Different risk reduction interventions may be most effective at particular times in the lifespan. Adolescence tends to be the most difficult time for parents as they try to differentiate normal adolescent changes from abnormal behaviors in children who may be approaching the highest time for risk onset. Parents of at-risk children need to understand that the risk for illness onset peaks and then decreases as the children age. Try to maintain a strong focus on the importance of early intervention, empowering parents and other close relatives to identify and seek treatment for symptoms early. Now let's talk about incorporating risk reduction into your counseling sessions. Ask parents of the at-risk child what they think about the likelihood of risk reduction and what efforts they are making in this area, if any. Be aware that they may not have consciously evaluated their actions as related to risk reduction or not. I have found that it is not unusual for parents of at-risk children to say that they are doing nothing specific to reduce risk, but as our discussion continues, they report a laundry list of ways in which they have modified their parenting approach or home environment. It is important that clinicians reinforce any risk reduction efforts that parents have undertaken, as well as suggesting other possible efforts. The parenting home environment modifications that I hear most often in my counseling sessions include the following: vigilance about the moods and behavior of the at-risk child, reducing exposure to stressful experiences and/or finding more effective strategies for managing stress, encouraging the expression of feelings and concerns, identifying mental health clinicians and other trusted individuals who can help at-risk children who are going through difficult times or who have questions, talking about the importance of abstaining from substance abuse, encouraging good decision-making, and being aware of the child's social network, including whether the child has friends and whether the friends are considered to be positive influences. Expect that families will be quite pragmatic, hoping to reduce risk but understanding that there should be a strong focus on minimizing symptoms should they emerge. It is vital to describe the benefits of early intervention and appropriate treatment and long-term outcomes. For many clients, you need to reinforce this point by contrasting their long road to a diagnosis and appropriate treatment to what might have happened if someone close to them had been in a position to notice and act appropriately on their symptoms. You want to spend a significant amount of time on anticipatory guidance related to the onset of symptoms in an at-risk relative. Such families include those of very high risk, for example, a child with 2 affected parents or multi-generational illness on both sides of the family. In these cases, it is especially important that the family has a plan. Now let's talk about some things that you can say to clients and family members of at-risk children. Here is one. While your child's chance of developing psychiatric illness is higher than the chance for a child who does not have an affected parent, you actually have one significant advantage over a family who has no experience with psychiatric illness. You know what the symptoms look like. That would allow you to recognize the condition quickly if it did emerge and get appropriate help. These important actions can improve the long-term prognosis. Remember, symptom identification and help seeking are important and unrecognized advantages that families with psychiatric illness hold over families who have no such experience. Good evidence is emerging from the literature on early psychosis intervention that duration of the untreated psychosis inversely correlates with the long-term prognosis. For example, the longer the duration of untreated psychosis, the worse the long-term prognosis. In this way, although it may not be possible to prevent the onset of psychiatric illness, it may be possible to respond to emerging psychiatric illness in such a way that the impact is reduced. Watching your teenager and keeping note of how do you think that you'll manage giving him more freedom while still trying to make sure he stays healthy. This is an issue with which most families of at-risk individuals tend to struggle. Opening a dialogue on this topic can allow the client to explore possible plans the family could implement as possible transition for the child into his or her adolescence. Have you ever thought about what you would do if you noticed symptoms in your daughter or son? A statement like this one focuses your discussion on ideas around planning, response, and management strategies if symptoms do emerge, and therefore allows you to provide some anticipatory guidance. Now let's talk about some issues that families of at-risk children may raise. If I am a good parent, can I prevent my child from developing the psychiatric illness that exists in my family? After hearing that the environment is an important factor in determining whether someone develops mental illness, people with personal or family histories of mental illness may become attached to the idea that perfect parenting can prevent their child from developing mental illness. This would be an important perception to expose and address, as there is no known parenting method by which mental illness can be prevented. On the other hand, bad parenting is unlikely to be the sole cause of psychiatric illness, because parenting is the primary aspect through which parents attempt to shape their child's development. Clinicians should anticipate that these messages about parenting are likely to challenge parents' perceptions of control. An important message to convey is that good parenting will be beneficial to a child regardless of his or her psychiatric status, and should a child become ill, he or she is likely to have a better outcome if he has a stable, communicative, and supportive home environment. How can I tell if my teenager is okay? Here you can reflect that it is difficult to determine the difference between normal adolescent behavior and emerging mental illness, and that managing that uncertainty can be really difficult for family members. You could offer counseling for such families to help them achieve a balance between appropriate vigilance and inappropriate boundary setting or anxiety. Review the manner in which psychiatric illnesses for which the young person is at risk may manifest, and to reassure the parent that the teenager is most likely to remain well, if possible and appropriate, given what you know about his or her current mental health status and risk factors. She's just a toddler, but I'm already worried about my daughter. How will I be able to stand watching her grow up, just waiting for her to become ill like me? Validate this client's concerns and ask what he or she perceives the risk to be. Ask if the client would like a better understanding of the risk. Ensure that the client understands that his or her daughter is most likely to remain well, if this is actually true. Understand, however, that his or her concerns may have little to do with the at all for the same disorder with which the client lives. Reflect how difficult it can be to live with uncertainty and help the client determine how he or she will manage it. Explore the client's perceptions on his or her own illness and explain that if the daughter were to become ill, her experience might be quite different than her parents'. Finally, address the guilt that this client seems to be expressing about passing risk on to his or her daughter. We have done everything we can to try to make sure that our daughter or son stays well. Here you want to support the parents in their efforts to maintain their child's mental health. Ask them about those efforts and how effective they perceive them to be. Help them understand and manage their inability to prevent the illness while reinforcing the fact that they may be effectively reducing the risk through their efforts. Clarify that we simply do not know how to significantly reduce risk and reinforce your perspective that their efforts seem constructive, if this is applicable. If I make sure he or she stays away from street drugs, he'll be fine, right? Help clients understand that limiting exposure to potential environmental trigger is helpful, of which we can control like this. We cannot prevent illness. Address how emotionally challenging it is to be unable to control whether an at-risk person will develop a psychiatric illness. Let me give you a case example. Bill was a 75-year-old man diagnosed with bipolar disorder who wanted to know more about the chance that his daughter would be affected with a mood disorder. His daughter was in her early 20s, and Bill described all the efforts that he and his wife had made to ensure that she did not experience any environmental conditions that might further increase her chances of developing bipolar disorder. They tried to create a calm, safe, and supportive home environment, and they taught their daughter how to make good decisions. They tried always to be available to their daughter if she needed to talk about them with their feelings or if she wanted their opinions. Bill had a few symptomatic periods during his daughter's childhood, and though he thought they were undoubtedly disruptive and scary for her, he felt that he and his wife had done a good job as they could with providing age-appropriate information and shielding her from as much as possible. As she got older, Bill had several very general discussions with her about his illness. Bill wanted some guidance about how to have a much more frank and detailed discussion now that she was an adult, including how to talk with her about her own risk for bipolar disorder. He also wanted to know if there was a chance for anything else that he could be doing further to reduce her chance of the disorder. The genetic counselor spent some time describing the etiology and the likely risk, and Bill practiced how he might present the information to his daughter. In terms of prevention efforts, the counselor discussed with Bill how avoiding street drugs would certainly be something that could protect her mental health. The counselor also described the importance of early symptom identification and early intervention. Though Bill could not control his young adult daughter's exposure to risk factors, he could discuss his own experience, model open communication about his own mental health, describe the importance of asking for help, and inform his daughter about the potential effect of street drugs. Bill had not considered how early intervention might impact his family if the worst did happen and his daughter developed bipolar disorder. When the genetic counselor presented this as an option for discussion, it was enthusiastically accepted, and Bill left telling me that he felt somewhat more hopeful about his daughter's future. Having a child develop a major psychiatric disorder is usually an intensely distressing experience for parents, regardless of whether it is childhood onset disorder or a disorder that emerges during adolescence or young adulthood. When children become ill, anticipating and responding to parents' questions about, such as, why my child? What could I have done to prevent this? These require careful attention to both education and psychological support. I'm going to build upon the advice in the previous sections of this lecture and provide some guidance when educating and counseling parents or guardians of affected minors. First, let's discuss autism spectrum disorders. ASDs present specific challenges to mental health clinicians who are providing education about factors that contribute to the development of illness and family risk. As you are probably aware, the rate of diagnosis of ASDs has increased quite significantly over the past 10 years. Recent reports posit that the prevalence of autism spectrum disorders is approximately 1 in 100. The exact cause of this increase is not yet known. Experts disagree about whether the frequency of the condition itself is on the rise or whether the frequency of the condition itself is stable, but a greater proportion of affected children are receiving appropriate diagnoses. The causes of autism have been a matter of intense public interest and debate, and much misinformation exists, including the common misperception that increased vaccine exposure has the increased rates of autism. Several studies, as reviewed by DiStefano and Thompson, have included that vaccines did not cause autism in the large majority of clients. though it is possible that exposure to vaccines does trigger symptoms in a small percentage of genetically vulnerable individuals. As in other major psychiatric illnesses, research suggests that for most affected individuals, genes and environment interact to cause symptoms. As with other major psychiatric illnesses, relatives of individuals with ASDs are at an increased risk for psychiatric disorders. The empiric risk for autism in a child with an affected sibling is approximately 5 to 10%, with risk at the higher end of the range if the affected child is a boy and lower end if the affected child is a girl. If two siblings are affected, the empiric risk for another sibling increases to about 25 to 35%. We have less data for other ASDs, but it is expected that the risks are similar. Data also suggests that relatives of an individual with ASD have an increased risk for a range of ASDs and other milder related manifestations. In addition, mood disorders and anxiety disorders are more common in close relatives. As with other psychiatric illnesses, it is not uncommon to hear of relatives who have some features of the condition but who are able to function completely normally. For example, parents of a child with autism might describe relatives who have awkward social interactions but who are brilliant eccentrics. All children diagnosed with autism should be referred to a pediatric geneticist for an evaluation. Following the tiered evaluation scheme recommended by Schaefer and Mendelsohn, 2008, it is expected that a clinical genetic examination and genetic testing will identify a genetic cause in approximately 15% of affected children, and that this number is expected to increase as new genetic tests become available. When autism is associated with a genetic syndrome, risk for recurrence in close relatives may be quite different than that predicted by empiric risk, and in many cases, prenatal testing or testing in very young siblings is possible. In addition, when autism is associated with a genetic syndrome, there are often other associated health risks, and knowledge of these risks may affect medical management. Finally, the emotional and practical burden of autism and severe developmental disorders make it vital that clinicians attend to parents' feelings about their children and themselves. Many parents feel at least partially responsible for their children's illness. This may be especially true in families where the parent or other close relatives have mild symptoms or mood disorders that the family perceives as related to the illness of the child. Parents who choose to have additional children may have serious concerns about younger siblings and benefit from education about risk and counseling about their uncertainty and anxiety. As I previously described, pediatric or early teen onset of mood or psychotic disorders increases a greater genetic contribution to illness, and risk for close relatives is also to be affected is increased. In this type of situation, it is important to educate parents about factors that contribute to the development of psychiatric illness and reinforce the message that parenting and home environment did not cause the illness. Given the increased rate of illness in close relatives, be aware of the chance that one or both of the parents might have challenges to their own mental health. If there are concerns about the parent's mental health, addressing those concerns might allow the family to better cope with the illness of the child. Pediatric diagnosis of mood and psychotic disorders has also been associated with a more severe illness course. For example, children with bipolar disorder have much higher rates of rapid cycling and increased rates of comorbidity. Children often have secondary developmental problems that result from their illness, including poor peer interactions and delayed social development. Diagnosis and treatment of these disorders are often difficult. Parents, siblings, and other caregivers require a great deal of education and psychological support. Parents who have attempted a stable upbringing should be encouraged to reframe feelings of failure and instead recognize that they provided a strong base that will allow the child and family to more successfully meet the challenges of psychiatric illness. There is some evidence that relapse rates of some psychiatric illnesses are higher in families with high expressed emotion or high levels of criticism, hostility, and emotional over-involvement. Although it is difficult to identify definitively anything other than the correlation between the high expressed emotion and relapse, it makes an intuitive sense that helping families to communicate more effectively and cope better with the illness may support the whole family and potentially reduce the rate of relapse in affected individuals. When a close relative has been diagnosed with child onset of a mood or psychotic disorder, it is important to educate adult relatives about the presentation of emerging symptoms, especially how those symptoms might look different than those in adults. For example, irritability in a depressed child. Early symptom identification and early intervention may reduce the effect that symptoms have on the child's social and emotional development. Finally, I recommend the adoption of a low threshold for referral to genetic specialists when a client has an early onset of an illness. When in doubt, refer. It is always appropriate to consult with a genetics professional when an affected child has developmental delays or birth defects, or a family history of developmental delays or birth defects, or if the child has a strong family history of psychiatric illness. So what can you say to parents of affected children? Can you tell me your thoughts are about what caused this illness in your child, a statement or question along these lines can be really helpful in gauging the parent's views on the causes of the illness in their child. This can serve as an entry into explorations of any feelings of guilt and anger related to origins of illness, and can also lead to discussion about the known factors that contribute to the development of these illnesses. Are you concerned about your other? Will be welcomed by some parents. For those who have more than one child, the thought of the onset in other children may be associated with so much fear that it is very difficult to acknowledge the question, let alone verbalize it. For these individuals, having you pose the question may feel like a real relief. For others, however, it may be too difficult to consider, and the question may be met with an angry or upset response. In these cases, some parents may need more time to adjust to having an affected child before they discuss risks with their other children. And some parents will never be able to or interested in discussing risk to their other children. Nevertheless, the fact that you have broached this topic should indicate to them that you are willing and able to support them around this difficult issue, and so lays the foundation for future discussion. Do you plan to have other children? You should consider addressing this important question still in their childbearing years. The diagnosis of the family member can dramatically impact future childbearing decisions. A parent's perception of risk for additional children to be affected might be quite different from the empiric risks. Estimation of risks for relatives is complicated by the young age at illness onset and the need to rule out genetic syndromes. Dr. Anderson, she is a pediatrician and he is a radiologist, attended genetic counseling because they were expecting their second child, and their first child, Mark, was 4 years old and had been diagnosed with autism. They explained that they were interested in receiving the most up-to-date and comprehensive information possible about the etiology of their son's autism and a quantitative risk assessment to the pregnancy. Their son had recently been evaluated by the Genetics Center's medical geneticist, who was unable to identify a syndromic etiology for the disorder. The couple eloquently described how difficult it was not to latch on to fad explanations for their son's illness because they were so desperate to know what had caused the autism and to reduce some of the guilt that they felt about his illness. They appreciated that their medical backgrounds allowed them to evaluate the literature on possible causes, including vaccinations, but they were very frustrated that the possible explanations seemed like a dead end. A genetic counselor explained the current understanding of the etiology and, given their backgrounds and training, provided the couple with some relevant review articles from the medical literature. The genetic counselor described the parents' natural reaction to feeling guilty that they have affected children. Mrs. Anderson reflected that she often saw this reaction in her pediatric practice, even when there was no blame to be placed in her situation. I later asked them about their risk perceptions for the developing baby. They had a reasonable understanding of the empiric risk for the recurrence of autism. Mr. Anderson told me that pregnancy was not really planned. They had not decided to risk having another child, but both had really wanted another baby, and they were pleased but worried about this pregnancy. The couple was scheduled to have an ultrasound the following month, and Mrs. Anderson expressed her strong hopes that the baby would be a girl. She understood that having a female may reduce but would not negate the increased risk caused by their family history. The genetic counselor continued the session by engaging the couple in discussion about how they would manage the uncertainty about whether the baby would be well for the rest of the pregnancy and early in the baby's life. The couple left the session with a more nuanced understanding of the etiology of autism and the risk to their pregnancy.
lovely introduction. Thank you very much, and thanks very much for the invitation to be here today. I'm deeply impressed that there are so many people here, despite the fact that I think it's probably going to shape up to be nice and sunny today. So I'll try and make it worth a while. We'll see how that goes. All right, let me see what I can put the first slide up. That's good. So, yeah, so I'm going to be talking with you for the first hour this morning. I'm glad I've got you fresh about genetics and mental illness, about what we currently know about how genetics is relevant to mental illness, and about where the research is going in the future. And so I know that really when we think about this stuff, I think what most of you are probably most interested in is what's the cutting edge stuff, what's happening right now, and like what have we got coming around just around the corner kind of thing. But it's kind of really hard to make some of that make sense without doing a little bit of the background about where all this is coming from and what has the research to date shown us. So I'm actually going to do a little bit of a kind of a history of psychiatric genetics, if you like. But I know that I have a big, wide range of different levels of knowledge, so I'm going to try and make it accessible to everyone. It's a bit of a tall order, but I'll see what I can do. And I would like to welcome questions as we go along. So if there are things that are pressing as I speak, please do just raise your hand, shout out, whatever, catch my attention somehow, and I'll try and address it. Okay. So So it's nice that you've got these slides popping up there, because I don't have them on my screen, but I can read them there, so that's good. So anyway, I thought that what we would have as an objective for this morning is to basically discuss what it is that we currently know about genetics and mental illness, and what this means, most importantly, I suppose. What does that mean for me if I have a mental illness? What does that mean for me if I'm a family member of someone with mental illness? What if I'm a doctor who's trying to treat my patients who have mental illness? So those are the sorts of things that I want to try and tackle this morning. So, before this is nasty, don't bother trying to read it. I'll read you the bits that are important. And I know the text is probably too small. But I think as we get started, as we just start thinking about genetics as it relates to mental illness, it's really important to acknowledge the environment in which we're living. You are all living in the genomic era. Right? The first draft of the Human Genome Project was completed in the year 2000, and what you're looking at on the screen here is a press release from the White House, which is announcing with great fanfare, hey guys, we just completed the first draft of the Human Genome Project, this is great. The bit that's underlined in red says that basically this will bring in an era that will bring new ways to prevent, diagnose, treat, and cure disease. And yeah, wow, that's right. So this was really, I think, the sentiment in around 2000. There was so much hope and expectation around what this draft of the human genome would be able to tell us, the promise that it held for being able to diagnose, treat, cure disease. It sounded amazing. So there was a lot of hype around this. You know, the media picked up on it, and suddenly the lay press is reporting all these genes that have been found to be associated with various different conditions. And so what happened was that the public became quite interested. The public got quite savvy. Has anybody seen or heard of this group before? They're called 23andMe. Yeah, there's a few people that have it. So this is a really interesting company. It's an online company. And basically what they do is they will, if you connect with them, and they had a special on earlier this year, so for $99, they will send you a little kit, and you spit in a little tube, and you seal up the tube, stick it in the mail, and they will use your cells that they extract from your saliva, from the spit, and look at your DNA. And they will send you a report back telling you about your risk to develop all sorts of different kinds of conditions based on having looked at your DNA. Okay? It's very fast. Good deal, huh? I don't know, actually. So the interesting thing about companies like this is that because they're internet-based, there's actually very poor regulation around them about the sorts of testing that they do and the sorts of information that they give back to people. So while some of the genetic differences that they look at in the context of their testing are actually, they're tests that most clinicians would agree are reasonable tests, although perhaps we shouldn't be offering them online, perhaps your doctor should be offering them to you. Some of the other genetic differences they look at are less well agreed upon. And I'll just leave it at that for now. We'll come back to this concept though later on. So, I should say that some of the conditions that they offer testing for include schizophrenia and bipolar disorder. Yeah. Anyway, so this brings us back to the broader question is, okay, So this is the genomic era. There are companies out there that for $99 will look at the DNA in my skin and tell me about my risk of schizophrenia or bipolar disorder. Is this even legitimate, really? Is it? So is genetics even a relevant consideration when we're talking about mental illness? And so that's where I'm going to start. I'm going to start from the beginning, from how we started looking at this, and I'm going to bring you right up to date, and then we're going to have a little peek into the future about where we're going with all the genetics research and mental illness. So actually, the first people to start asking questions about whether genetics was relevant to mental illness were about 500 years ago. There's an amazing guy called Ariel, what was his name? Paracelsus Bombastus or something. I envy his name, it's quite great. So he was the first one to notice that mental illnesses tended to cluster in families. And so more recently, we've been looking at this question in a more sophisticated kind of way, I suppose, by doing a family study. And really what you're looking at here is a graphic that just shows a family study. So in this picture, each of the two little clusters represents a family, and the squares represent males, and the circles represent females. And people who are shaded in because they have a mental illness of a particular kind. So the idea behind a family study is that we look to see how often the close family members of the person on the left are affected with the same mental illness as compared to the close family members of the person on the right who does not have mental illness. And what we always see when we look at families that have one member with a mental illness is that there's a higher rate of mental illness amongst their biological family members. Doesn't mean that their family members are guaranteed to get the same illness, not by any stretch of the imagination, but there's a higher rate. Okay? So you may think, well, that tells us for sure then that genetics is important, right? It causes mental illness? No, not at all actually. So because families actually, it could be that we see a higher rate because perhaps families share an environment in some way that makes them more vulnerable as a group to developing mental illness. So it doesn't really, it's an interesting observation, but it doesn't really help us to understand whether it's genes or environment that contributed to the development of these conditions. So back in the day, we used to talk about this as nature versus nurture. Is it nature or is it nurture? People want black or white answers to these things. And I think my problem with the word nurture is it sounds like, anything that isn't genetic must be the parent's fault. So I don't like the word nurture, and you'll hear how I talk about it instead a little bit later on. So anyway, family studies were there. They showed a higher rate of mental illness amongst the close biological family members, but we can't really tell why. Is it because of shared genes, or is it because of shared environment? And so this is where twin studies come in. Twin studies are super cool. So, as you probably know, there are two different kinds of twins, right? There are twins that are identical to each other, and they share all of their genetic information, 100% of it, nature's very own clones. And then on the other hand, there are non-identical twins who share about the same amount of genetic information with each other as a regular brother or sister would, so about 50%. So, what we can do is we can do a study where we look to see if one of a pair of twins has a mental illness, what's the chance that their co-twin will have the same condition? Okay? And then we can compare the rate with which those two twins are both ill at the same time between twins who are identical and share all of their genetic information and twins who are not identical and sharing about 50% of the information. So for a condition that's caused entirely by genes, if one of a pair of identical twins is affected, the other twin will have the same condition 100% of the time. Okay? Does that make sense? Yeah, great, okay, cool. And so let's look at that first column there, the one that says identical twins. None of those numbers in that column say 100%, right? So that tells us straight away that these conditions are not conditions that are caused entirely by genes. Okay, straight away, that's what we can see. And I should also say that these studies, this isn't just one study that's been done, these are like dozens of studies over decades have been done, and they all show the same thing. The other thing that we can see from looking at this table is that the number in the identical twins column is always higher than the number in the non-identical twins column. And that's a really key piece that geneticists look for. It tells us that although the number in that identical twins column is not 100%, so it means that genes aren't the only thing that's going on, because the number for identical twins is higher than for non-identical twins, it means that genes must play an important role. Okay? So that's really what we can summarize from this table. So, and from this we can derive what are called heritability estimates. And the reason I bring this up is because sometimes I will see people in genetic counselling who will say, well, I've read that schizophrenia is 85% genetic, so that must mean that my son developed schizophrenia as a result of an 85% contribution of his genes and a 15% contribution of other stuff, environment. Is that right? And so I think it's a really important concept to talk about. So heritability is actually it's a very complex statistical concept. And I think the best way to think about it is we've all seen these, news reports, StatsCan surveys, whatever, that talk about how, on average in the Canadian population, people have 2.4 or 2.7 children. I don't remember how many it is. Is it 2.4? 2.4 sounds good. Let's go with that. Okay. So on average in Canada, it's about 2.4 kids that people have. Does anyone know an individual who has 2.4 kids? No. And so it's talking about across the population. So the concept is that it's an average. The same is true for heritability estimates. So 85% of schizophrenia as a heritability is a good population average, but we can't tell from that number how much my schizophrenia might have resulted from genes as compared to environment. Just the same as I can't tell from the StatsCan data that on average people have 2.4 kids, whether I have 1 child or 7 children or no children. Right? Does that make sense? Great. You're doing really well. So, what we know, basically, in general terms then, is that mental illnesses, no matter which one we're talking about, so you'll hear me mentioning schizophrenia a lot, and I'll use that probably as my primary example, but everything that I say today holds true for all of the other mental illnesses that we could possibly be talking about. It's just that the most research has been done in the context of schizophrenia, so I'll use that as my go-to example quite a bit. So what we know in general terms then is that mental illnesses are caused by genes and environment. That's how I prefer to talk about it, instead of nature and nurture. So environment, so our experiences, things that happen to us. And because of this, we can't really talk about the genetic differences that contribute to mental illness as causing. They don't cause. They contribute to vulnerability, or they increase our predisposition or our susceptibility, if you like. But they don't cause mental illness in and of themselves, because there's this other piece. The environment that's so important as well. And then the second point there says that in general, mental illness is not inherited, but what we can inherit is a vulnerability to mental illness. And that's a subtle but very, very important difference that I hope makes some kind of sense. Okay. So that's very nice to talk about it in words. Genes and environment, lovely, I kind of get that. But for me, I'm a visual learner. I like to have pictures that represent things. And this is a tool that Angela and I use in our genetic counselling service to help people understand in a different way more about those words. So what do we really mean by that? So the analogy that we use is that we each have a mental illness jar. And in order to be actively experiencing an episode of mental illness, that jar has to be filled up to the top. And there are 2 different kinds of vulnerability factor one can use to fill one's jar with. There are these circles, which represent the genetic vulnerability factors, and then there's triangles, which represent our life experiences or environmental vulnerability factors. And so as you can see, like we're born with a certain amount of genetic vulnerability, and that doesn't change over time. But what can change over time is the amount of environmental vulnerability in that jar. So you can see in the middle, the jar has a little bit of environmental vulnerability in it, but it's not full up to the top still, so the person is not actively experiencing an episode of illness. But on the far right-hand side, there's been enough experiential things have accumulated such that the jar is now filled, and the person experiences their first active episode. So obviously, we're not all going to be born with the same amount of genetic vulnerability to mental illness. And so, we also use these 2 pictures. The top one sort of shows somebody who starts out with relatively little genetic vulnerability, and the one at the bottom talks about somebody who started out with quite a bit more, I suppose. And we might imagine that the person at the bottom is more likely to develop mental illness than the person at the top. Perhaps they might develop mental illness at a younger age, just because there's less space in the top of that jar to be filled. But also, if we just look at the 2 jars on the very right-hand side of the picture, this talks about how there are different ways in which one's jar can fill up. You can have a small contribution of genes and a large contribution of the environment, or a large contribution of genes and a small contribution of the environment, and so on. So, as many of you are aware, 2 people can have the same diagnosis, schizophrenia, but their clinical symptoms might actually look quite different from each other. Why is that? We think that it's probably because of this, the underlying differences in the nature of the factors that contributed to the development of the condition in the first place. Does that make sense? Yes. Excellent. Yeah. Okay. So that's all very nice, but I'm supposed to be talking with you this morning about genetics specifically. Like, this is lovely about the jars, great, whatever. But what can you tell us about genetic research? So we're now going to focus in on these little yellow circles in this jar. What are these genetic factors specifically? Okay, that's what's next. So essentially, what the genetic factors are that I'm representing in as little circles in this jar is there differences in our genetic code, in our DNA. And we all have differences between individuals. And there are several different types of genetic difference that we can think about. And I'm going to go through each one of them in turn with you. Okay? So, does anybody recognize this from like biology in high school or anything like that? Yeah. Cool. So basically what you're looking at is a picture of human DNA. This is what it really genuinely looks like if you take a photo of it down a microscope. So it has to feel nicely arranged. Of course, usually it's a big blobby mess, but if you separate them out, this is what it can look like. And so, when we think about DNA, so the genetic code that tells our bodies how to grow and develop is called DNA. And almost all of the cells in our bodies have this DNA inside them. And usually we think of that DNA as being one big long string inside a cell. It isn't. It's actually 46 little strings, and each of the strings is called a chromosome. Okay, so that's what you're looking at. So each one of these little wormy things is a string of DNA, and it's called a chromosome. Okay? So we have, as you can see, they're arranged in pairs, and that's because we get one copy of each chromosome from each one of our parents. The first 22 have numbers because we're not very imaginative in genetics. And the last two, these are the really important ones that determine whether or not whether we're male or female. So in this, does anybody know whether this is a male or a female? Female? Good guess? Male? Okay. It is actually a male. So with this, we can tell because that very last pair on the bottom right, there's an X and a Y. So that means it's a boy. If it's a girl, it's going to be two X's and no Y. Okay? So that's what we can tell from looking at this. So it first became possible to look at our chromosomes down a microscope in the 1950s, so quite recently. And so the first thing that people did when they wanted to know, hey, what causes this condition? It doesn't matter what the condition is right now, it could be anything. What causes this condition? I wonder if it's genetics. Let's look down the microscope and see if we can see any differences in the DNA between somebody who has the condition I'm interested in and somebody who doesn't have the condition. So that was a super helpful technique for understanding what caused Down syndrome. So Down syndrome is actually caused by having an entire extra copy of chromosome number 21. So instead of having 46 chromosomes like here, an individual with Down syndrome will have 47, and the extra one is a copy of 21. So scientists can look down the microscope and see, aha, there's an extra chromosome, that's what causes this condition. So they did that for mental illness. They looked down the microscope, couldn't see anything different, nothing at all. No extra chromosomes, no missing chromosomes, kind of like, doesn't help us. So, but there are some other things that you can tell just from looking down the microscope of chromosomes. You can also sometimes see rearrangements. And what I mean by rearrangement is that you can have a chunk of a chromosome that's broken off, and a chunk of another chromosome that's broken off, and they kind of switch places. Okay, that happens, relatively commonly. And so people look for those in the context of mental illness. Is it these swapping bits of material that makes a difference? And in about 5 decades worth of research, we've found 4 families in the world that have mental illness as a result of those kinds of translocations between different chromosomes. So we are not talking about something that's responsible for mental illness in the vast majority of cases. So this still wasn't really very helpful. So what do we do next? Well, so a little bit of time goes by and technology improves, and now we become able to more closely look at the DNA. So instead of just looking at the gross structure of the chromosomes, now we're able to actually look at the sequence of the DNA itself. Okay, so basically what you're looking at here is a cartoon of a chromosome. And this is probably something you've come across before, that DNA looks like a twisted ladder. So the thing to know is that basically just all genes is there a section of that string of DNA that tells the body how to make a particular protein, right? Like a dopamine transporter, serotonin transporter, something like that, proteins. Okay. And the genes, so you can see that each one of the rungs of the ladder has 2 parts. And each one of those parts, this is just extra information for those of you who may be interested, are called nucleotides. And I'm going to come back to the concept of nucleotides in a little bit. But yeah, so that's really all you're looking at. It's the picture of how the DNA and the gene relate to the chromosome. Okay? So it became possible to look at the sequence of the rungs on the ladder. So that's what people started doing next. So this is probably around, let's say, early 1980s, well, 1970s perhaps a little, but it really started picking up in the 1980s that this became possible. So what people did then was they started looking for chunks of the chromosomes that were transmitted together with the disease through multiple generations of a family. Okay? So what you're looking at here again is that males are squares, females are circles, everybody who's shaded in has got schizophrenia, let's say, and the little lines by each of the symbols, they're cartoons of chromosomes. Okay? So, so a region transmitted together with the disease. Can anybody spot a region of one of the cartoon chromosomes that's being transmitted together with the disease? The green bit, yeah. There it is, there it is, there it is. Yeah, exactly. So this is the purpose of the linkage study. It's looking for chunks of the chromosome that are transmitted together with the disease through multiple generations of the family. This sort of technique is spectacularly helpful when a condition is caused by genes. So conditions like Huntington's disease or cystic fibrosis, everybody who has one of those conditions has the same genetic kind of states going on. And for those kinds of conditions, this linkage study was supremely helpful. We used it in the context of mental illness. Kind of equivocal. So one group would say, hey, we found this region of chromosome number 5 is linked to schizophrenia. And another group would try and look at the same thing and they say, no, we didn't find that. And then somebody else would say, how about chromosome 2? I found linkage of chromosome 2. Another group would try and say, no, we didn't find that. So lots and lots of ambiguity, I suppose. We couldn't figure out how to interpret the results. And in the context of illnesses like this, we really need to be to have replication, so multiple people finding the same thing, in order for us to be confident that a finding is real and not just random chance or a fluke. So that really wasn't happening. So people started getting disillusioned. They thought, okay, well perhaps it's because the genetic differences that contribute to the development of mental illnesses, they're not like the ones that cause Huntington's disease or cystic fibrosis. Perhaps instead the genes have small effect. So let's use a different technique, this one, called the association study. And the ideal thing about this is that the association study is much better at detecting genetic differences that have small effect. So linkage studies are good for finding genes that have big effect. Association studies are good for finding genes of small effect. So, and it's really simple. All you're doing is you have a population of people with schizophrenia and a population of people who do not have schizophrenia, and you identify a genetic variation and you look to see how frequently it occurs in each of those two populations. And if it occurs more frequently amongst the population of people with schizophrenia, then we say it's associated with schizophrenia, and we think that perhaps it might play a role in some way in making people more vulnerable to developing the condition. Okay? So not causing, but making people more vulnerable. So, and in fact, this was what I did for my entire PhD. Like, I spent my entire PhD looking for these little genetic variations by doing association studies. And this was, and I finished my PhD, just to give you a sense of the timeline where we are, I finished my PhD in 2001. So we're kind of up to around 2000 at this stage. And so, again, I found some stuff, I published some papers saying, hey, I found that there's a variation in a gene called neuroregulin that's, neurotensin, that's more frequent in people with schizophrenia than people who do not have schizophrenia. Nobody else can find it. Other people published papers saying, hey, I found it's in this gene, there's a variation that makes people more vulnerable. I tried it in my sample, I couldn't find it. So, again, lots of conflicting results. So we weren't getting that replication that we needed to be sure that these were true findings. However, things carry on, and after a couple of decades worth of trying linkage and association studies, the psychiatric genetics community had kind of felt like, yeah, okay, we've got mixed results, but there are these genes that we're relatively confident that they seem to play some sort of role in mental illness. And so geneticists, I'm afraid, love acronyms. So NRG1 just stands for neuroregulin. DTNBP1 is disappointed. It doesn't really matter. I think what does matter is to notice that middle column. And the middle column shows that most of the genetic variations that were identified using these two old school techniques, so linkage and association studies, they showed that these differences did not seem to increase a person's vulnerability specifically to schizophrenia alone. For the most part, the same variations were also implicated in bipolar disorder. And I didn't put it on the slide because it would have gotten too busy, but also, for many cases, depression. So these differences don't seem to increase a person's vulnerability to one specific kind of mental illness. It seems to be mental illness broadly. Okay? The other thing I want to bring your attention to is that third column, one that says relative risk. And what that means is, how important is this gene? How much does it increase a person's risk? Okay? So if we think about the general population risk of schizophrenia being 1%, if you have that variation in the top row there, in the irregular, then your risk doubles, which sounds terrifying until you realise that that means your risk has gone up to 2%. Right? So nothing really that's going to be clinically useful. So nothing that people were offering genetic tests for. Also bear in mind that, you know, there was kind of broad consensus that maybe these do something, but there have been negative findings associated with these genes as well. So it wasn't that everybody was like, absolutely, it's a sure thing, that's certainly associated with this condition. It was like, yeah, it probably is, probably, you know, on the way to the strength of the evidence that we got, looks like it. So this is where we were at by about 2003, I suppose. Okay, so this is the list of the best genes that we had at that point. And then technology develops further. And so now what became possible was it became possible to instead, I talked about association studies, right, where you took one genetic variation and you looked to see if it was more common in a population of people with schizophrenia than it was in a population of people without schizophrenia. Now we have the concept of the genome-wide association study. And basically what that involves is doing 500,000 association studies at the same time. Yeah, ooh, exactly, it blew lots of people's minds. And so really, I'm going to show you this picture again. Okay, so a genome-wide association study looks at tiny differences in the DNA. So remember, this is the twisted ladder, this is just a cartoon of the DNA again. And so a single nucleotide polymorphism is so a nucleotide is just half of the ladder rung thing that we were talking about earlier. It just means that one of the pairs of rungs on the ladder has been changed for another one, and that's what you can see in this cartoon. And this is what we use in genome-wide association studies. These occur really commonly. We all have loads of them. Okay. Most of the time, they don't do anything negative to our health at all. They occur about 1 every 1,000 rungs on the ladder. And, but we look at 500,000 of these differences at the same time in a population of people with schizophrenia and a population of people without schizophrenia, or a population of people with bipolar and a population of people without bipolar. And we're looking to see some of these more frequent in the population of people who are affected. Okay, that's the idea. So, the problem though, you might imagine, is if you're doing 500,000 of these tests at the same time, aren't you going to get some of them come back as showing association just like your charts? Absolutely, yes you are. So you need to have really stringent levels and thresholds for statistical significance. And you need to use absurdly huge sample sizes. So when I was doing my PhD, which I finished, as I said, in 2001, I had a sample size of 500 people with schizophrenia and 500 controls. And I really thought I was the dog's doodah because my sample size was huge compared to everybody else's. But with genome-wide association studies, you need thousands, so not hundreds, thousands of people in each of the two groups in order to be able to have enough power to tell whether or not the variations that you're seeing are really different or not. So, 2006 is about where we're at right now, maybe 2007. So this is the list of genes that came back from the first genome-wide association studies that were done in the context of schizophrenia. Does anybody recognize any of those acronyms from that other slide I showed you? No. Me neither. Yeah. So this was actually a pretty devastating blow for the psychiatric genetics community. Because really, we thought, okay, we've been using these old school techniques, linkage and association, and yeah, okay, conflicting results, but the list of genes I showed you a moment ago, everybody was pretty confident that they were going to be playing an important role. And so everybody was confident going into the genome-wide association studies that they would basically confirm our suspicions and solidify our confidence in those variations. And that did not happen. So basically what happened was that psychiatric geneticists en masse had a hissy fit, or a tantrum, I suppose. And it really did cause quite a significant crisis of confidence. I mean, remember, it had been several years by this stage since the announcement of the first draft of the Human Genome Project, right? And we're still waiting for ways to prevent, treat, we're still looking for all of that stuff, and we can't even find the genetic differences that make people more vulnerable. So it was quite a profound, awful time. So, in fact, it sparked cartoons like this one, which I really enjoy. For those of you that can't oh, good, I'm glad you all enjoy it as much as I do. eulogy, I suppose, I mean, in academic circles. This was a paper that was published in Nature, so a very good journal, and it's talking about, oh my goodness, we know that these conditions, they have a genetic component, but there's all this missing heritability. We can't find the genes, the actual specific genetic differences that make people more vulnerable. So it was a bad time. What came out of it was essentially two camps. So people took this blow and they went in two different directions. And I'm going to tell you about each of the two different directions. One group said, okay, we know for sure that it's going to be that these are conditions that are caused by multiple genes, each one of which has only a small effect on whether or not somebody develops mental illness. Okay? So we're using the right strategies, the genome-wide association study. The problem is that our sample sizes aren't big enough. So we need to have even more people in our studies in order to have enough power to detect these differences. Okay? The other group, they basically just laughed and they said, come on guys, we're going to need to recruit everybody in the world who has schizophrenia and everybody in the world who does not have schizophrenia to have enough power to find these genes that you think exist. We disagree. We think that instead, we've been using the wrong strategy. Although the linkage studies, which are good for finding genes of big effect, we've been using them in the wrong way. So we think that actually schizophrenia and bipolar disorder and major depression are all going to be conditions that are caused by genes that each have big effect, but not in a population, in a family. Okay? So in my family, it might be one gene in particular that causes a big effect, and in your family, it might be a different gene. So when we look at the population as a large, it all washes out, and we don't see anything. So you have to look at individual families to find those differences. So these were the two schools of thought that emerged in around 2007. Okay? So I'm going to bring you up to date with where each of those two groups has gone. So, the group that said, okay, the genome-wide association study, looking for these genes that small effect, lots of them, where they went was really go big or go home, was really what they decided was the strategy. So, remember we had a good laughter that said, you need to recruit everybody in the world with schizophrenia. They did a pretty good job, actually. So, they formed an international collaboration and did a study, and this one was published in, I think, oh no, this was 2011. This was in Nature in 2011, like towards the end of last year. And it involved 52,000 people. Huge, huge. I mean, you know, let's think back to my PhD at the beginning of the 2000s, where I had 500, and I was like, yes. And they've got 52,000. So it really quite remarkable. So 52,000 people, and what did they find? Basically, what they found was they found 5 genetic differences that actually met the really stringent levels that we were setting for statistical significance. So, usually if you do a statistical test, if any of you have a stats background, you'll use a threshold of P, the statistical term, has to measure less than 0.05, right? And when we do these tests, we use a threshold of P has to be less than 5 times 10 to the minus 8. So in this study, they had the best genetic variation that they found came back with a p-value of 5 times 10 to the minus 11, which is really even smaller than 10 to the minus 8. So it means it's a really good candidate. So it looks really good at this point. Okay. So they were kind of yeah, told you so. Into the other group. They're very childish. Meanwhile, and this is actually another paper that was published at the end of last year from a group based out of Montreal, so a Canadian paper in Canada. And what they decided to do was look for just the genetic differences that occur in the regions of the DNA that codes for proteins. So not all of our DNA codes for proteins. In fact, only about 2% of it codes for proteins. Which is kind of bizarre as an idea, but anyway, they have it. And so what they did was that they looked at all of the DNA in the entire genome of 14 people with schizophrenia that code for proteins and their parents. Okay? And what they found was that the people who had schizophrenia had a higher rate of brand new mutations or differences in their DNA than they would have expected. So they weren't inheriting these differences from their parents. They kind of spontaneously happened somehow. Okay? Really interesting. But, okay, so I should hold that thought for a second. I'm going to go back to that first study that I was just telling you about with the P10 to the minus 11 thing. So, although it was very strongly significant, it still has a tiny effect size. So again, if in the general population your chance to develop schizophrenia is 1%, if you have this variation that they've identified in this study last year, it's 2%. So it's nothing that we're going to be saying, hey, everybody should be getting genetic testing for this, because it doesn't make any difference to how anybody we choose clinically, right? So, sorry, to bring us back to that second study I was just mentioning again. So they found that people who had schizophrenia had a higher rate of brand new differences in their genes that they had not inherited from parents. But we all have brand new genetic differences that we've not inherited from our parents, and there's a lot of overlap between the two groups. So it's not that we can say people who have schizophrenia have more than 10 brand new differences that they've not inherited from their parents, and if you do not have schizophrenia, you have less than 10. No. There's a lot of grey murk in between. And even if this holds up, and even if it was a discrete thing like that, we still don't really know what it means, because it's not telling us about what specific differences are. Do you know what I mean? So it's still not something that can be useful as a test. Okay? So that was the one group. That was the common variant, common disease group. Let's talk about the other group, the group that laughed and said, you guys just, that's just silly. We're going to go the route of trying to identify genetic differences that are rare in the general population, but within a specific family, they make a big difference to whether or not somebody develops mental illness. And so the approach that they took was to use a model of a condition that we know of very well. It's called 22q deletion syndrome. And the picture that you're looking at here is with permission, a picture of a man who has this condition. Okay? And so really, all that this condition, so it's called 22q deletion syndrome because if you remember that picture of the chromosomes that I showed you, there's a chunk of chromosome number 22 that's deleted, hence the name, 22q deletion syndrome. So the interesting thing about this condition is that 30% of the individuals with it will develop some form of psychotic illness. So schizophrenia or bipolar disorder with psychosis. Okay? And that's a really big elevated risk over what we would expect based on the general population. So it's perhaps something about this deleted chunk of DNA that these people have that somehow increases their risk. So this other group of researchers decided, okay, we're going to look for lots more of those kinds of genetic differences because we think there's going to be loads more of them than we currently know about, and they're going to exist in discrete families. So that was what they were doing. So their question was basically, are there lots of these rare, small, missing or extra chunks of DNA that increase a person's vulnerability quite substantially? So where did they get to? So basically, they started finding lots and lots and lots of extra or missing chunks of DNA in people's genomes. They found them in people with schizophrenia, for sure, yeah, they did. They found them in people with bipolar, for sure, absolutely. But they also found them in the genomes of everybody, like whether or not they have schizophrenia or bipolar disorder. And this was actually quite a shock. We didn't realise that this was going to be the case. We thought that to have missing or extra chunks of DNA of that kind of size would for sure cause some kind of health problems that we could see. Doesn't seem to be true. So how do we interpret the fact that, yes, there were these missing or extra chunks of DNA in people with schizophrenia or bipolar disorder, but we also all have them anyway? So it's kind of, again, we're not in a place where we can say this is a good test that we can offer. Okay? Yeah, question? Yeah, two questions. I'll go for this one first and then that one.
How does epigenetics play?
Oh, you preempted my last slides. That was your question too, right? Yes. Okay, good. I put the right slides in then, sorry. So just to repeat the question for those that didn't hear it, the question was, where does epigenetics play into this? And so that's going to come right at the end, because that's more of a future thing. So we're just on the cusp of starting to look at that stuff in the context of psychiatric disorders. I'll get to it in a sec. So, where are we right now? So that kind of brings us up to date as far as those two groups. So one group with a common disease, common variant, each one a tiny small effect, and the other group looking for the rare variants, each one a big effect, but in different families. So to me, frankly, it felt a bit like the whole nature-nurture debate all over again. Is it nature or is it nurture? Is it these common variants or is it these rare variants? And happily, after this period of infighting, sense prevailed, I suppose, and people have realised finally that it seems that there is a role, perhaps, for both of these mechanisms in mental illnesses. Perhaps in some families, it is going to be multiple genes, each one a small effect, and for other families, there's going to be these big insertions or deletions of DNA that have bigger contributions to whether or not someone develops an illness. So there's going to be a lot of differences between individuals who have these conditions. And so just as you remember at the beginning, I was telling you about how you can have jars that are filled in different ways, large contribution of environment, small contribution of genetics, or the other way around. It's also going to be the nature of the little circles in that jar that differ between individuals. They might be these big missing or extra chunks of DNA, or they might be just these tiny changes in the rungs on the ladder. It's going to be different for different people. Okay? So, just to summarise what it is that we know so far before we get into the epigenetics that apparently there's a lot of enthusiasm about in the room. Good. So what we know so far is that there are a few very rare families as a result of chromosomal changes. Remember I showed you that picture of looking at chromosomes down a microscope, and I told you that there's about 4 families in the world where we found they have translocations, so chunks of chromosomes have moved around, and that seems to go together with the illness in some families. About 4, very rare. And there are a few rare, small missing or extra chunks of DNA that seem to make substantial contributions to increased risk of schizophrenia or bipolar disorder, like the 22q deletion syndrome I was telling you about, the picture of the man that I showed you. So some single nucleotide variations do, so those changes in the rungs on the ladder that I was showing you, do seem to make small contributions for increased risk for schizophrenia. So remember that the variation I was telling you about that had the significance value of 5 times 10 to the minus 11, you know, that's pretty good. But they only make small contributions. And there's some evidence emerging that people with schizophrenia may have a larger number of genetic variations than people without schizophrenia. But again, it's not a black and white thing. There's lots of overlap, and we don't really know what that means yet. So, actually, you know what? I'm not going to talk about that, I think, in the interest of time. Yes, so let's talk instead about what it is that we don't know. So we've done all this work, but what we don't know still is how to interpret the clinical importance of most of these missing or extra chunks of DNA that we could be finding. As I said, we're finding them in the general population all over the place. It was unexpected, but there it is. So how do we interpret this stuff? We don't know. We pretty much for sure don't know the full list of all of the different variations that could potentially contribute to the development of schizophrenia or bipolar disorder. And we don't know how the different variations combine together to increase a person's vulnerability. We don't know the mechanism by which these variations increase a person's risk. And we don't know either why it is that lots of the genetic differences that have been found don't seem to increase risk for just schizophrenia alone. They seem to increase risk for schizophrenia or bipolar disorder or depression. And perhaps it just depends what specific combination of factors that you have that determines what the eventual manifestation of one's illness might be. So, a lot that we don't know. So you may be thinking, okay, and then what was the point of all this? I didn't think that's something I could do. But no, so I think that the fundamental thing to remember is that although we don't have all of the details down, i.e. we don't know the nature of the specific genetic variations yet with absolute certainty, and we don't have the full range of them, we do know absolutely, there's no disagreement really, I think, no matter what your academic discipline. There's no sort of dissent about the evidence that it seems from the twin studies like that I showed you at the very beginning that we know that genetics plays a role in these conditions. Okay, so we're still kind of back in that place that I told you about just as the genome-wide association study data came out for the first time, and everybody was like, oh, I don't know what to do. So we don't have all of the details about the specific variations, but we still know that genetics is an important contributor. So what does this mean right now? Where are we at for doctors and people with mental illness in their family as well? Fundamentally, there is no genetic test with which we can diagnose mental illness. Okay? And at present, there is no evidence to suggest that any genetic test can offer any kind of meaningful information about a person's risk of developmental illness. I would say, aside from that 22q deletion syndrome thing I showed you earlier, where if you have that deletion, you have a 30% chance to develop mental illness. But I should also say, if you have that deletion, then you have a whole host of other medical problems as well. So it's not just that it increases risk for mental illness. So let's get back to that 23andMe. Let's get back to that. So basically, the genetic testing that they're offering. So think of all the complexity that we've just talked about in the last little while. These guys are offering genetic testing for schizophrenia that's based on 2 single nucleotide polymorphisms, so those little changes in the rungs on the ladder, And they're not even any of the ones that I showed you in my slides. Okay? Hopefully that gives you some sort of sense of the credibility. That's all I'm going to say. Because it's a big thing. Okay, so that's where we are at right now. Let's talk about where we're going. And so there's been some preemptive questions about epigenetics, so you'll be delighted to see that it's right there. Look. So I think what's next? So we've been talking about changes at the level of individual rungs on that DNA ladder. And I've been also talking with you about missing or extra chunks of DNA. And to date, the size of the missing or extra chunks of DNA that we've been looking at have been in the realm of thousands of these rungs on the ladder, chunks of DNA that are thousands of rungs on the ladder long. Okay? So in between this size of difference and the individual rung on the ladder size difference, there has to be a whole whackload of smaller missing or extra chunks of DNA that we haven't even begun to look for yet because we haven't really had the technology until very recently. So I think that one of the next things that will happen is that people will start looking for these. So the missing or extra chunks of DNA that are like a couple of dozen bases. There's a question up there. Yeah.
In my immediate family, two have suffered severe depression. My sister has, both parents had severe depression.
Yeah.
Is it suggested that that's not genetic?
Okay, so I think the thing to remember is that, actually, let me go on to my next. So so my message to you today, if you take away nothing else, is that genetics is absolutely relevant to mental illness. Although we do not have the specific variations pinned down, we know that this model is correct. Okay? But we know, for the most part, that despite the family history that you describe, it's not going to be the case that there is one single genetic variation that causes depression in those members of your family. Actually, that pattern of family history is exactly the sort of thing that we would expect to see if we accept this model, that it's genes and environment acting together, and that in order to experience an active episode of illness, the jar has to be filled up to the top. So I wouldn't describe mental illnesses as genetic conditions. They're what we call complex or multifactorial conditions. So they arise as a result of the combined effects of genes and environment acting together. Okay, so yes, I would say that genes are important, but I wouldn't necessarily say it was genetic. I hope that subtle difference makes sense.
It came down the male line.
Right, yeah.
The female.
Yeah, and so if that's something that you're interested in talking about, I'll refer you to my colleague Angela over there. That's exactly the sort of thing we can talk about in genetic counselling. But actually for now, I'd just like to get back onto the, to where we're going in the future, I suppose, as far as genetic research in this area goes. So that's the smaller missing or extra chunks of DNA. We talked about that. The other two pieces are, I've written them down here as microRNAs and epigenetics. But really, I think what summarises both of them is that to date, we've been very busy looking at the sequence of the DNA letters. Okay? There is more to our genomes than just the sequence of the letters. So, just to give you an example, human beings have 25,000 genes. Who thinks that sounds like a large number? Some people think that sounds. I thought it sounded like a large number. And then I heard that rice has 55,000 genes. And that's a true story, I'm not making this up. So rice has 55,000 genes. Human beings have 25,000 genes. But we are, well, conscious for the most part. We have sequenced our genome. And as far as we know, rice has not achieved any of these things. So what's the difference between, so how can we be so much more or we like to think that we're so much more complex than rice, but we have fewer genes? Like, how does that work? Well, what we're increasingly coming to understand that it's not the number of genes, it's the subtlety and the detail with which they're regulated. Okay, and that's where epigenetics comes in. So epigenetics is just talking about the epi means upon or beside. So upon or beside the genetics piece. So not the sequence of the A's and C's and G's and T's, the sequence of the rungs on the ladder, but there's like little marks that can be made on the DNA that determine whether or not there's more or less of a particular protein produced. Okay, and that's what gene is, something that produces the protein. And so microRNAs basically do the same thing. it's chunks of DNA that code for something that isn't a protein. It's a different kind of molecule, but what it does is it regulates how much protein one's DNA produces. So, so basically, I think where we're going is understanding that, you know, so for example, as twins, identical twins, they're born with exactly the same sequence of rung on the ladder, for the most part. But as they age, their epigenetic marks, so how their proteins are produced, diverges from each other. So we actually see, you know, as twins, you know, one twin will develop schizophrenia, the other twin will not. How come? How come? Well, perhaps it's because of how our genetics interacts with our environment. Okay? So we know that environmental influences can alter the way in which our DNA is expressed, I suppose. I know this is really complicated, I'm ever so sorry, but I do think it's important to know where it's going. So it's looking, in some ways, just looking more at the subtleties, not in the sequence of the DNA bases, but in terms of what the effect is of that. Yeah. Yeah, question.
So would adoption twin studies then be another area to explore?
Oh, adoptions. Yes, adoption twin studies, right. So, I didn't talk about adoption studies of mental illness today, and I didn't because of time, and because I wanted to get onto the meat in terms of, you know, what it is that we're doing in genetics now, kind of thing. But there have been adoption studies done. So adoption studies, you can do them in a number of different ways, where you look at young people who have been adopted away, who, for example, later on develop mental illness, and you look to see how often their biological and their adoptive parents are affected with mental illness. You're talking about twin-based adoption studies. Very, very, very difficult to do, because ethically we cannot say, aha, you had twins, we're going to adopt them away so that we can see what happens, and, you know, you know what I mean? So from a scientific point of view, it would be wonderful to have that information, but there really is very little data about that, just because it's naturalistically very, very difficult to find those samples. And, you know, with lots of things like that, we can use animal models, but we really don't. There's no animal researchers in here. I don't think we have any really good sort of, how many? 5, okay. We don't really have any good animal models of mental illness, but we can try taking rats away from an affected mother, for example, and see what happens. So really great question, but it's very difficult to do those studies, and there's very little data. Yeah. Okay, yeah, question at the back.
Is there any research on environmental factors that might be considered in a prevention program?
Absolutely. Come to see our genetic counselling service. We'll tell you all about it. So I'm just going to finish up with my slides, because there's another cartoon that you might find amusing. I don't know. But I think when we think about genetics and how it relates to mental illness, I think what we hope for is, you know, what I showed you on the press release from the White House. We hope for prevention, cure, treatment. And so gene therapy is, I think, what people think of a lot of the time. And not to put a dampener on things, but we've known about the gene. There's one gene that causes cystic fibrosis. We've known about it since 1984. Okay? Cystic fibrosis is a condition that affects people's very thick, sticky mucus in their lungs, makes it hard to get enough oxygen, blah, blah, blah. Nasty, nasty condition. There's one gene that causes it in everybody that has the condition. The environment is not important, it's just genetics. Okay? It's the lungs that are affected. So we can access our lungs. I'm accessing my lungs, right? I breathe in, breathe out, and that's my lungs that I'm accessing. So we know the gene, the organ is accessible, and we don't have any good gene therapy even for cystic fibrosis after more than 20 years of knowing what the gene is that's responsible. If we think about mental illness, we're talking about, it's been really complex what we talked about today, multiple different genes, okay? The organ that's affected is very, very well protected inside my skull and by my blood-brain barrier on the inside. So I think there's going to have to be some significant advances in how we think about gene therapy before I think this is going to become realistic in the context of mental illness. But the reason I mention that now is because I do want to come back to the point that you mentioned. So, okay, if we can't do gene therapy, what can we do to protect ourselves against developing mental illness, or to reduce our chance of having a relapse if we've already experienced the condition? And so that's something that I often talk about in other presentations that I do, but it is actually a major focus of what we talk about in the context of genetic counselling, and we talk about it in a very personalised way. So we learn about your personal family history and about your own experiences with mental illness, and then we talk about protective factors in that context. So if that is something that you're interested in, I would encourage you to come and speak with myself or with Angela, and we can set up an appointment.